ವೆರಿ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಟು ಒನ್ ಆ್ಯಂಡ್ ಆಲ್ ಪ್ರೆಸೆಂಟ್ ಹಿಯರ್. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು ಆ್ಯಂಡ್ ಬೆಳಗಿನ ಶುಭೋದಯ. ಐ ರಿಕ್ವೆಸ್ಟ್ ಟುಡೇಸ್ ಗೆಸ್ಟ್ ಆಫ್ ದಿ ಪ್ರೋಗ್ರಾಮ್, ಪ್ಲೀಸ್ ಕಮ್ ಆನ್ ಟು ದಿ ಡಯಾಸ್. Professor Nagaraj sir, Founder Director Base and Prayoga, Professor Shiv Prasad sir, Professor Girija madam, and Dr. Anand sir, Dr. Ramesh sir. So we are today to celebrate National Science Day in the memory of Dr. C.V. Raman sir, great scientist of the era of India. So first of all, I request all of you, please rise up for Nadagite. Thank you. Now we have Karnataka Vijnana Science and Technology Academy Vijnana Geete. It's written by Professor Dodda Rangegoda and sung by M.D. Pallavi.
ಕರುನಾಡನ್ನು
Now I request Dr. A. M. Ramesh sir, CEO, KSTA, to welcome the guest and all of you for this wonderful function.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಗುಡ್ ಮಾರ್ನಿಂಗ್. ವೇದಿಕೆ ಮೇಲೆ ಆಸೀನರಾಗಿರುವ ನನ್ನ ಗುರುಗಳು, ಅಕಾಡೆಮಿಯ ಹಿಂದಿನ ಹಣಕಾಸು ಸಮಿತಿ ಅಧ್ಯಕ್ಷರು ಹಾಗೂ ಪ್ರಯೋಗ್ ಸಂಸ್ಥೆಯ ಮಹಾಪೋಷಕರು ಆದ ಡಾಕ್ಟರ್ ಎಚ್ ಎಸ್ ನಾಗರಾಜ್ ಸರ್, ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಆಗಮಿಸಿರುವ ನಮ್ಮ ಹಿಂದಿನ ಅಧ್ಯಕ್ಷರಾದ ಡಾಕ್ಟರ್ ಎಸ್ ಕೆ ಶಿವಕುಮಾರ್ ಅವರ ಧರ್ಮಪತ್ನಿ ಹಾಗೂ ರಸಾಯನಶಾಸ್ತ್ರದ ಪ್ರಾಧ್ಯಾಪಕರಾಗಿ ನಿವೃತ್ತರಾಗಿರ್ತಕ್ಕಂಥ ಡಾಕ್ಟರ್ ಸಿ ಆರ್ ಗಿರಿಜಾ ಮೇಡಮ್, ನಮ್ಮ ಪಕ್ಕದಲ್ಲೇ ಇರುವ ನ್ಯಾಷನಲ್ ಸೆಂಟರ್ ಫಾರ್ ಬಯಾಲಜಿಕಲ್ ಸೈನ್ಸ್ನ ಅಸೋಸಿಯೇಟ್ ಡೀನ್ ಅವರಾದಂತಹ ಡಾಕ್ಟರ್ ಪಿ ವಿ ಶಿವಪ್ರಸಾದ್ ಸರ್, ನನ್ನ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರಾದ ಡಾಕ್ಟರ್ ಆನಂದ್ ಮಾದೇಗೌಡ, ಶ್ರೀನಿವಾಸ್, ದಿಲೀಪ್ ಹಾಗೂ ರಾಜ್ಯದ ವಿವಿಧ ಭಾಗಗಳಿಂದ ಬಂದಿರತಕ್ಕಂತಹ ಪ್ರಶಸ್ತಿ ವಿಜೇತರೆ. Today, as you know, today is the very important day, National Science Day. National Science Day was started celebrating in the year 1987. Government of India took a decision to celebrate National Science Day on the recommendation of Department of Science and Technology in 1986. Since then, we have been organizing National Science Day regularly. This is the 38th year that we are celebrating National Science Day. As you may be aware, this year's National Science Day's theme is Indigenous Technology for Viksit Bharat. In fact, our Honorable Minister was supposed to be here to inaugurate this National Science Day, but due to our Honorable Chief Minister's program in Jawaharlal Nehru Planetarium, our Minister has to be there, so he has conveyed his greetings to this function. And our Honorable Minister also has congratulated all the award winners in 3 categories. Dr. S. K. Shivakumar Sir Award for UG students, Professor U.R. Rao Award for PG students, and KSTA Award for general public and also teaching faculty. In fact, KSTA was started in the year 2005 under the chairmanship of Padma Vibhushan Professor U.R. Rao. Under his guidance, KSTA worked for nearly 12 years. We conducted many programs across the state. After that, another great space scientist, Dr. S. K. Shivakumar Sir, was our chairman for about 2 years. And recently, Professor S. Ayyappan, former Director General of ICR, was our chairman for 3 years. And we had many eminent personalities as members of KSTA. Dr. H. S. Nagaraj Sir was also one of the members of KSTA for more than 6 years. And he was also the chairman of our finance committee. Under their guidance, this beautiful campus has been built. And Chief Executive Officer and Member Secretary of KSTA was Dr. H. Honegoda for more than 15 years. Under his guidance, many programs were successfully implemented, not only in Bangalore but across the state. It is a matter of pride to say that we have conducted more than 35 conferences across the state, only 8 of them in Bangalore. Rest in Gulbarga, Bijapur, Raichur, Mysore, you name any district, we have conducted a program. I mean to say, we have reached to every district, but we have not reached to every taluk. Our ambition is to not only to reach the taluks, but also to villages. Of course, it depends on the grant and support from people like you. You should always be an ambassador for academy programs. We have only one office here in Bangalore. We have only 15 employees. 4 of them are scientific officers, including me. So it is very difficult to cover every part of the state. We depend on collaborators. In fact, KSTA has MOU with 60 organizations so far, universities, educational institutes. In association with them, we organize many programs. We cover entire educational pyramid, right from high school to the university level. We conduct many programs, and these programs have been very successful. And in fact, last year on this day, I was in New Delhi to receive National Award for Science in New Delhi to receive National Award for Science Communication for outstanding effort. So that's all because of people like you who have been encouraging KSTA to do more. I request you to be great support for us in future as well. And Viksit Bharat, in fact, it was started in 70s itself. In fact, ISRO is the pioneer, I must say. When Professor U.R. Rao was the head of the Aryabhatta satellite mission, he used to say average age of persons, technicians, engineers who were involved in the launch of Aryabhatta on 19th April 1975, it was just 25 years it seems. So youngsters had a lot in Aryabhatta's success. So Professor U.R. Rao always used to give encouragement to youngsters, and that's why KSTA started awards for youngsters, like for PG students, UG students, and also to young researchers, including teaching faculty. In fact, I must say there were 91 entries, including 15 for PG award, for Dr. S.K. Shivkumar award, 35 from UG students, and KSTA award, 37, including general public and teaching faculty. And we should get at least more than 200 next year, because you all should propagate the information to your own colleagues and students, so that we will get the award will go to the deserving people. Not only selecting only 5 from 15 is not a great thing. We should select 5 from 200. Only then the award will get more weightage. So I request all of you to kindly help KSTA in this regard. And as I told, Professor U.R. Rao was the architect of satellite mission in India with the launch of Aryabhatta. At the same time, our Dr. S.K. Shivkumar was the architect of big antenna, 32-meter antenna you all must have be knowing. It's established in Byalalu. It is a 32-meter antenna through which we get information from Mangalayaan, Chandrayaan, and so many other, even Aditya mission. All these are received at Byalalu antenna. The architect was Dr. S.K. Shivkumar, Padma Shri awardee, and he is known as the father of deep space antenna network in India. So under their guidance, KSTA blossomed into a good organization. We are very grateful to these two doyens of science, space science and technology. And we have with us Dr. H.S. Nagaraj sir, as I told. Sir is the founder of BASE Institute. Some of you must be aware. After PU, students would be aiming to go for JEE and other competitive exams. In fact, in 80s, there was no educational institute to train for JEE or other competitive exams. When I completed my PU in 92, I used to go to Vijay College. Sir was my teacher. We used to listen at the college and study ourselves. But now it is not the case. After maybe 90s, the requirement was that a good educational institute which trains students to go for JEE or CET or NET and so many other competitions. Sir is the pioneer of such an institute in Karnataka. Now, it is a matter of pride that Sir has started an institute, educational institute, research institute called Prayoga. It is in Kanakpura Road. Recently, Professor U.R. Rao Research Lab was inaugurated. It is truly world class. I urge all of you to once visit Prayoga, then you will understand the pain and the enthusiasm with which Sir has started Prayoga. It is free for everybody. Anybody can take their students with prior permission from high school to university level. They can go and work. And there are physics, chemistry, biology, and earth sciences lab. Anybody interested could contact me. I will give the contact address. You can experience a world-class lab there. On behalf of Karnataka Science and Technology Academy, I welcome sir to this program. Hearty welcome to you, sir. As they say, for behind every successful man, there is a woman. Madam is the woman behind Dr. S.K. Shivkumar sir's success, Padma Shri awardee. He was the Kstacademy director for more than 10 years. He was also the director of URSC for more than 4 years. Under sir's guidance, KSTA organized many programs. In fact, we lost him very early, just 1.5 years. He was with us. He was very friendly, humble, and even now we remember his humbleness. On behalf of all of you, Professor of Chemistry from Government Science College, Madam is retired now. On behalf of all of you, welcome, hearty welcome to you, Madam. Now we have with us Dr. P.V. Shivaprasad. He is the Associate Dean of Faculty, National Center for Biological Sciences. In fact, I had invited Director of NCBS. He was busy. Sir suggested Dr. Shivaprasad to be invited. When I sent them an email, I got instant reply from him that he will be attending. We are very grateful for accepting our invitation, sir. Sir, Dr. Shivaprasad has done MSc in Applied Botany from Mangalore University. After that, he got CSIR fellowship to pursue his PhD in biotechnology from Madurai Kamaraj University under the guidance of a renowned educationist, Dr. K. Velu Thambi, and later sir pursued postdoctoral research at the very renowned University of Basel, Switzerland. Then second postdoctoral on gene silencing in tomato under the guidance of Professor Sir David Blocom's lab, University of Cambridge, UK. With this brief introduction, I once again welcome you, sir. We have with us senior faculty, Dr. Manja Nayak and Dr. Vinay and others. I welcome all of you to this function. I welcome all the prize winners and all the participants who have come to participate in the painting competition, and also some of the family members of the prize winners have come. I welcome you all to this function. After the inaugural, Professor P.V. Shivaprasad will deliver a talk. He is known to be a very good speaker. I request all of you to be present even after the prize distribution. You can listen to his speech, and after that you can have lunch and disperse. And I also welcome all my colleagues, Dr. Anand, Madhav, Dilip, Srinivas, Shantraj, Ramesh, Umesh, and our cameraman Bharat, and all of you welcome. Once again, I am sure this Science Day celebration will be very meaningful and all of you will enjoy. Thank you.
Indeed, thank you very much, sir, for overviewing about this program, and also RT welcome to you, sir. Without your guidance and support, could not able to organize this kind of programs. Welcome you, sir. Now I request Professor H.S. Nagaraja, sir, mentor Prayoga, formally inaugurate the program by watering the plant, and also dignitaries, please join hands. Thank you all. Now I request Professor Nagaraja sir to address the gathering.
ಎಲ್ಲರಿಗೂ ಶುಭೋದಯ. ಮುಂಜಾನೆಯ ನನ್ನ ನಮಸ್ಕಾರಗಳು. ವೇದಿಕೆ ಮೇಲೆ ಇರ್ತಕ್ಕಂಥ ಎಲ್ಲ ಮಿತ್ರರೇ, ಶಿವಪ್ರಸಾದ್ ಅವರು, ರಮೇಶ್, ಡಾಕ್ಟರ್ ಗಿರಿಜಾ ಅವರು, ಆನಂದ್ ಅವರು, ಇದು ತುಂಬ ಸಂತೋಷವಾದ ದಿನ ನನಗೆ. ಏಕೆಂದರೆ 2-3 ಕಾರಣಗಳಿವೆ. ನಾನು ನ್ಯಾಷನಲ್ ಸೈನ್ಸ್ ಡೇ ವಿಷಯಕ್ಕೆ ಇನ್ನೊಂದು 2 ನಿಮಿಷ ಬಿಟ್ಟು ಬರ್ತೇನೆ. ಐ ವಾಸ್ ಲಕ್ಕಿ ಟು ಬಿ ಪಾರ್ಟ್ ಆಫ್ ಮೈ ಜರ್ನಿ ಇನ್ ಲೈಫ್ ವಿತ್ ಪ್ರೊಫೆಸರ್ ಯು ಆರ್ ರಾವ್ ಫಾರ್ 6 years, ಥ್ಯಾಂಕ್ಸ್ ಟು KSTA ಇಲ್ಲದೆ ಹೋಗಿ ನಾನು ಅವರೊಟ್ಟಿಗೆ ಕೆಲಸ ಮಾಡೋವಂಥ ಅವಕಾಶ ಸಿಗದೆ ಹೋದರೆ ಈಗ ನನಗೆ ಗೊತ್ತು ತುಂಬ ದೊಡ್ಡ ಒಂದು ಜೀವನದಲ್ಲಿ ಕಳ್ಕೊತಾ ಇದ್ದೆ ಒಂದನ್ನು. ತುಂಬ ಸಂತೋಷ ಪಡುವಂಥ ಅವಕಾಶ ಹೊರಟೋಗ್ತಿತ್ತು. ಶಿವಕುಮಾರ್ ಅವ್ರ ಜೊತೆನೂ ಹತ್ರ 2 ವರ್ಷ ಇಲ್ಲಿ ಕೆಲಸ ಮಾಡೋ ಅವಕಾಶ ಸಿಕ್ತು. ನನಗೆ ಹಲವಾರು ಮಿತ್ರರು ಇಸ್ರೋದಲ್ಲಿ ಕೆಲಸ ಮಾಡಿದವರು ಇದ್ದರು, ಮೈ ಓನ್ ಕ್ಲಾಸ್ಮೇಟ್ಸ್. ರಾವ್ ಇವರೆಲ್ಲ ಎಂಥವ್ರು ಅಂದರೆ ದೇ ಹ್ಯಾಡ್ ದಿ ಬೆಸ್ಟ್ ಪೊಸಿಷನ್ಸ್, ಟೀಚಿಂಗ್ ಪೊಸಿಷನ್ಸ್, ರಿಸರ್ಚ್ ಪೊಸಿಷನ್ಸ್ ಇನ್ US. ಸಾರಾಭಾಯ್ ಅವರು, ಇವರ ಒಂದು ಸರ್ಕಲ್ ಏನಿತ್ತು, ಅವರು ಬಹುಶಃ ನಾಸಾಯಿಂದ ಹಿಡಿದು ಎಲ್ಲಿ ಬೇಕಾದರೂ ಏನು ಕೇಳ್ದಲೇನೂ ಇವರಿಗೆಲ್ಲ ಸವಲತ್ತು ಸೌಕರ್ಯ, ದಿ ಹೈ ಎಂಡ್ ಆಫ್ ಲೈಫ್ ಅಂತಾರಲ್ಲ, ಆ ಥರದ್ದು ಸೋಷಿಯಲಿ ಎಲ್ಲ ಸಿಗ್ತಾ ಇದ್ದಂಥ ಕಾಲ. ಭಾರತಕ್ಕೆ ಬಂದರು. ಯಾತಕ್ಕೆ ಬಂದರು? ಈ ದೇಶ ಬೆಳಿಬೇಕು, ನಾವು ಹುಟ್ಟಿದ ನೆಲ ಜಲ ಮಣ್ಣು ಇಲ್ಲಿ ಹುಟ್ಟಿರ್ತಕ್ಕಂಥ ಮಕ್ಕಳುಗಳು ವಿಶ್ವದ ಪ್ರಜೆಗಳಾಗಬೇಕು, ಒಳ್ಳೆ ಸೈಂಟಿಸ್ಟ್ ಆಗಬೇಕು ಅಂತ ಬಂದವರು ಇವರೆಲ್ಲ. ಅಂಥವ್ರ ಜೊತೆ ಕೆಲಸ ಮಾಡೋದೇ ನಮಗೊಂದು ಅದೃಷ್ಟ, ಪುಣ್ಯ, ನೀವು ಅದೇನಾರು ಹೇಳಿ. ಆ ದೃಷ್ಟಿಯಿಂದ KSTA ಮತ್ತು ಈ ವ್ಯವಸ್ಥೆ ಏನಿದೆ, KSTಗೆ ಸ್ವಂತ ಬಿಲ್ಡಿಂಗ್ ಇರಲಿಲ್ಲ. ಇದೆಲ್ಲ ಬರಲಿಕ್ಕೆ ಯು ಆರ್ ರಾವ್ ಅವರವರು ಭಾಳ ಶ್ರಮ ವಹಿಸಿದ್ರು. ಎಷ್ಟು ಕಾಳಜಿ ಇತ್ತು ಅಂದರೆ ಸರ್ಕಾರಿ ಆಫೀಸಲ್ಲಿ ಅಪಾರ್ಥ ಇಲ್ಲ, ಇದರಲ್ಲಿ ಕಷ್ಟ ಇದೆ ಕೆಲಸ ಮಾಡಿಸ್ಕೊಂಡು ಬರೋದು. ಅದೆಲ್ಲವನ್ನು ಬೇಜಾರಿಲ್ಲದೆ ಮಾಡ್ತಾಯಿದ್ರು. ಇನ್ ದ್ಯಾಟ್ ಸೆನ್ಸ್ ಐ ಆಮ್ ಗ್ರೇಟ್ಫುಲ್ ಟು KSTA ಆ್ಯಂಡ್ ಸೋ ಹ್ಯಾಪಿ ಟು ಬಿ ಹಿಯರ್ ಟುಡೇ. ಎರಡನೇದು, ನಾವೆಲ್ಲ ಇವಾಗ ತುಂಬ ಸೌಕರ್ಯ ಸವಲತ್ತು ಇದೆ. ಯಾವುದೇ ಒಂದು ರಿಸರ್ಚ್ ಮಾಡಬೇಕಾದ್ರೆ ಬೇಕಾದಷ್ಟು ಅವಕಾಶ ಇದೆ. ಯು ಆರ್ ರಾವ್ ಅವರು ನೇರವಾಗಿ ನಮ್ಮಗಳ ಜೊತೆ ಊಟದ ಟೈಮಲ್ಲೋ ಬಾಕಿ ಮೀಟಿಂಗ್ ಮುಂಚೆನೋ ಅವಾಗ ಇವಾಗ ಮಾತಾಡೋರು. ಅವರು ಫಸ್ಟು ಒಂದು ಇಲ್ಲಿ ಪೀಣ್ಯದಲ್ಲಿ ಅವರಿಗೆ ಜಾಗ ಕೊಟ್ಟಾಗ, ಯಾವುದಕ್ಕೆ? ಸ್ಯಾಟಲೈಟ್ ಟೆಕ್ನಾಲಜಿ ಡೆವಲಪ್ ಮಾಡೋದಕ್ಕೆ. ಈ ವರ್ಷದ ಥೀಮ್ ಏನಿದೆ ನ್ಯಾಷನಲ್ ಸೈನ್ಸ್ ಡೇದು, ಇಂಡಿಜಿನಸ್ ಟೆಕ್ನಾಲಜಿ ಫಾರ್ ವಿಕಸಿತ್ ಭಾರತ್ ಅಂತ. ಇದರ ನಿಜವಾದ ಬುನಾದಿಯನ್ನು ಹಾಕಿದವರು ರಾವ್ ಅಂತವರು, ಇನ್ನೂ ಹಲವಾರು ಜನ, ಸಾರಾಭಾಯ್ ಇರ್ಬೋದು, ಇವರು ಮತ್ತು ಬಾಬಾ ಇರ್ಬೋದು, ಇವರುಗಳೆಲ್ಲ ದೇಶಕ್ಕಾಗಿ ಕೆಲಸ ಮಾಡಿದವರು. ಅಲ್ಲಿ ಒಂದು ಶೆಡ್ ಕೊಟ್ಟಿದ್ದರು. ಇವತ್ತಿನ AC ರೂಮ್ಗಳಲ್ಲ, ಒಂದು ಇಂಡಸ್ಟ್ರಿಯಲ್ ಶೆಡ್ಡು, ಶೀಟ್ ಮೆಟಲ್ ವರ್ಕ್ ಮಾಡೋ ಅಂಥದ್ದು. ಇವತ್ತು ಮಾಡರ್ನ್ ಇಂಡಸ್ಟ್ರಿಯಲ್ ಶೆಡ್ಸ್ ಆರ್ ಆಲ್ಸೋ ಬೆಟರ್. ಅಂಥದ್ದೊಂದು ಅಲ್ಲಿ 3 ಜನ ಲೇಡೀಸ್ ಸ್ಟಾಫ್ ಇದ್ದರು. 2 ಲೇಡೀಸ್ ಟಾಯ್ಲೆಟ್ಗಳಿತ್ತಂತೆ. ಇವರಿಗೆ ಎಷ್ಟು ಅನಾನುಕೂಲದ ಜಾಗದಲ್ಲಿ ಕೆಲಸ ಮಾಡಿದರೆ. ಲೇಡೀಸ್ ಟಾಯ್ಲೆಟ್ 2 ಇತ್ತು. ಅಯ್ಯೋ 3 ಜನ ಸ್ಟಾಫು 2 ಲೇಡೀಸ್ ಟಾಯ್ಲೆಟ್ ಹಾಕಿ ನಮಗೆ ಜಾಗ ಇಲ್ವಲ್ಲ ಅಂತ ಆ ಟಾಯ್ಲೆಟ್ಗೆ ಸ್ಯಾಂಡ್ ಬ್ಯಾಗ್ನ ಹಾಕಿ ಒಂದು ಶೀಟ್ ಕವರ್ ಮಾಡಿ ಸ್ಟೋರೇಜ್ ರ್ಯಾಕ್ ಮಾಡಿಕೊಂಡು ಸ್ಟೋರ್ ರೂಮ್ ಮಾಡ್ಕೊಂಡಿದ್ರು ಟಾಯ್ಲೆಟ್ನ. ನಾವುಗಳ ಅಂಥ ಆಫೀಸಲ್ಲಿ ಅಂಥ ಪರಿಸ್ಥಿತಿಯಲ್ಲಿ ಕೆಲಸ ಮಾಡೋ ಒಂದು ಪರಿಸ್ಥಿತಿ ನಮಗಿಲ್ಲ. ವಿ ಆರ್ ಆಲ್ ಲಕ್ಕಿ. ಅಂದರೆ ಯು ಕ್ಯಾನ್ ಇಮ್ಯಾಜಿನ್, ಅಂತಹ ಒಂದು ವ್ಯವಸ್ಥೆಯಲ್ಲಿ ಮರದ ಶೆಲ್ಫ್ಗಳನ್ನ ಮಾಡಿಸ್ಕೊಂಡು ಅಲ್ಲಿ ಸ್ಟೋರೇಜ್ ಮಾಡಿಕೊಂಡು ಸ್ಯಾಟಲೈಟ್ ಟೆಕ್ನಾಲಜಿಗೆ, ಸೌಂಡಿಂಗ್ ರಾಕೆಟ್ಸ್ ಬಗ್ಗೆ ಅವ್ರು ಹೇಳಿದ್ದೆಲ್ಲ ಹೇಳೋದಕ್ಕೆ 2 ಗಂಟೆ ಬೇಕು ನನಗೆ. ಅಂಥ ಪರಿಸ್ಥಿತಿನಲ್ಲಿ ಇವತ್ತು ಭಾರತ ಇಟ್ಸ್ ಒನ್ ಆಫ್ ದಿ ಲೀಡಿಂಗ್ countries for space technology. What other countries have not been able to do, India has demonstrated. right from cryogenic engine to several other technologies, if India has progressed, it should be because of these indigenous technologies. So, ಈ ವರ್ಷದ ಥೀಮ್ ಏನಿದೆ, ಇಂಡಿಜಿನಸ್ ಟೆಕ್ನಾಲಜೀಸ್ ಫಾರ್ ವಿಕಸಿತ್ ಭಾರತ್, ಅದರ ಫೌಂಡೇಶನ್ ಸ್ಟೋನ್ಸ್ ಹಾಕಿದವರು these great scientists, and we are here in that place where those people had worked for their dreams. ಅಂತ ಒಂದು ಸುದಿನ ಇದು. Why is this celebrated? We all keep telling C.V. Raman's name. We are all proud. Every Indian should be proud. C.V. Raman ಅವರು ಒಂದು ರೌಂಡ್ ಬಾಟಮ್ ಫ್ಲಾಸ್ಕ್ ಇಟ್ಕೊಂಡು, ಬೆನ್ಸಿನ್ ಇಟ್ಕೊಂಡು, ಮರ್ಕ್ಯೂರಿ ಆರ್ಕ್ ಲ್ಯಾಂಪ್ ಇಟ್ಕೊಂಡು, ಇವತ್ತಿನ ಲೈಟ್ಗಳಿರಲಿಲ್ಲ, ಪವರ್ಫುಲ್ ಲೈಟ್ಸ್. ಎಕ್ಸ್ಪೋಜರ್ ಟೈಮ್ ಯೂಸ್ ಟು ಬಿ ಸಮಥಿಂಗ್ ಲೈಕ್ 40 hours. ಆ ಫಿಲ್ಮ್ ಮೇಲೆ ಒಂದು ಫೈಂಟ್ ಒಂದು ಇಮೇಜ್ ಬರಬೇಕಾದ್ರೆ 40 ಗಂಟೆ ಎಕ್ಸ್ಪೋಜರ್ ಟೈಮ್. ಇವತ್ತು ವಿತಿನ್ ಎ ಮಿಲಿ ಸೆಕೆಂಡ್ ಯು ವಿಲ್ ಗೆಟ್ ದಿ ಇಂಪ್ರೆಷನ್. ಆ ಕಾಲದಲ್ಲಿ ವರ್ಕ್ ಮಾಡಿ, ಇಟ್ ಈಸ್ ನಾಟ್ ಜಸ್ಟ್ ದಿ ಎಕ್ವಿಪ್ಮೆಂಟ್, ಬಟ್ ಇಟ್ ಈಸ್ ಯುವರ್ ಬ್ರೈನ್, ಯುವರ್ ಪ್ಯಾಷನ್, ಯುವರ್ ಕಮಿಟ್ಮೆಂಟ್ ಅಂತ ತೋರಿಸಿದಂಥ ಮಹಾನ್ ವಿಜ್ಞಾನಿ C.V. ರಾಮನ್ ಅವರು. ಅವರಿಗೆ ನೊಬೆಲ್ ಪ್ರೈಸ್ ಬಂದ ಸಂದರ್ಭದಲ್ಲಿ ಬಂದು ಇವತ್ತಿಗೆ 1930, ಎಷ್ಟು ಇನ್ನು 6 ವರ್ಷ ಆದರೆ ಒಂದು ಶತಮಾನ ಆಗುತ್ತೆ. ನೋವಿನ ಪಾರ್ಟ್ ಏನು ಅಂದರೆ 94 ವರ್ಷ ಆದರೂ ಇನ್ನೂ C.V. ರಾಮನ್ ಹೆಸರು ಅಷ್ಟೇ ಇಟ್ಕೊಂಡಿದ್ದೀವಿ. ಅದಿರಬೇಕು ಯಾವಾಗಲೂ, ಬಟ್ ಅದೊಂದೇ ಹೆಸರಾಗ್ಬಿಟ್ಟಿದೆ. ಆಮೇಲೆ ಚಂದ್ರ ಇವರಿರ್ಬೋದು, ವೆಂಕಿ ಇರ್ಬೋದು, ರಾಮಕೃಷ್ಣ ಇರ್ಬೋದು, ಕೊರೋನಾ ಇರ್ಬೋದು, ಅವ್ರು ಯಾರೂ ಭಾರತದಲ್ಲಿದ್ದು ಕೆಲಸ ಮಾಡಿ ನೊಬೆಲ್ ಪ್ರೈಸ್ ತೊಗೊಳ್ಳಿಲ್ಲ. ಹಲವಾರು ಹೆಸರುಗಳು ಕೆಲಸ ನೊಬೆಲ್ ಪ್ಯಾನಲ್ಗೆ ಹೋಗಿದೆ, IAS ಇಂದ ಹೋಗಿದೆ. ಸೊ ಇಂಡಿಯಾ ಈಸ್ ಎ ಗ್ರೇಟ್ ಕಂಟ್ರಿ ಅನ್ನೋದ್ರ ಬಗ್ಗೆ ಡೌಟ್ ಇಲ್ಲ. ಆದರೆ ವಿ ಕ್ಯಾನಾಟ್ ಲೀವ್ ಆ್ಯಂಡ್ ಕಂಟಿನ್ಯೂ ಟು ಬಾಸ್ಕ್ ಇನ್ ದಿ ಗ್ಲೋರಿ ಆಫ್ ದಿ ಪಾಸ್ಟ್. ನೀವು ಯಂಗರ್ ಜನ್ರೇಷನ್ಗೆ ನನ್ನ ಕೋರಿಕೆ ಏನು ಅಂದರೆ, ಯು ಶುಡ್ ಆಲ್ ಬಿಕಮ್ ಸೈಂಟಿಫಿಕ್ ಇನ್ ಯುವರ್ ಥಿಂಕಿಂಗ್. ವೆನ್ ಐ ಸೇ ಬಿಕಮ್ ಸೈಂಟಿಫಿಕ್ ಇನ್ ಯುವರ್ ಥಿಂಕಿಂಗ್, ಐ ಆಮ್ ನಾಟ್ ಟೆಲಿಂಗ್ ಗಿವ್ ಅಪ್ ಹ್ಯುಮ್ಯಾನಿಟಿ, ಹ್ಯೂಮನ್ ವ್ಯಾಲ್ಯೂಸ್ ಬಿಡಿ ಅಂತ ಅರ್ಥ ಅಲ್ಲ. ದಿ ಹೋಲ್ ಲೈಫ್ should be taken in the right perspective as a wholesome thing. But if you don't have the scientific thinking, we are going to remain a slave for some technologically advanced countries. ಈ ಹ್ಯೂಮನ್ ಹಿಸ್ಟ್ರಿ ನೋಡೋಣ. ಹ್ಯೂಮನ್ ಹಿಸ್ಟ್ರಿ ನೋಡಿದ್ರೆ ಒಂದು ಕಾಲದಲ್ಲಿ ಶಕ್ತಿ ಇದ್ದವನು ಬೇರೆ ದೇಶಕ್ಕೆ ನುಗ್ಗಿ ಲೂಟಿ ಮಾಡಿ ಎಲ್ಲವನ್ನು ಆಕ್ರಮಿಸ್ಕೊಂಡು ಬದುಕ್ತಿದ್ದ ಕಾಲ ಇತ್ತು. ಆಗಿದೆ, ಎಲ್ಲ ದೇಶಗಳಲ್ಲೂ ಇದನ್ನು ನಾವು ನೋಡಿದ್ದೀವಿ. ಸೊ ಫಿಸಿಕಲ್ ಮೈಟ್ ವಾಸ್ ದಿ ರೈಟ್. ಅದಾದಮೇಲೆ ಏನಾಯಿತು, ಯಾರು ಸ್ವಲ್ಪ ಟೆಕ್ನಾಲಜಿ ಡೆವಲಪ್ ಮಾಡಿದ್ರೋ, ಇಂಡಸ್ಟ್ರಿಯಲೈಸ್ ಮಾಡಿದ್ರೋ, ದಟ್ ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್ ಆ್ಯಂಡ್ ಟೆಕ್ನಾಲಜಿಕಲ್ ರೆವಲ್ಯೂಷನ್ ಸ್ಟಾರ್ಟೆಡ್ ಕಂಟ್ರೋಲಿಂಗ್. ಅಂದರೆ ಟೆಕ್ನಾಲಜಿ ಇದ್ದರೆ, ಇವಾಗ ಇಂಟರ್ನೆಟ್ ಈಸ್ ಎ ಗ್ರೇಟ್ ಲೆವೆಲರ್, ಆದರೆ ದಿ ಸೇಮ್ ಇಂಟರ್ನೆಟ್ ವಿತ್ ಆಲ್ ದಿ ಆ್ಯಪ್ಸ್ ಆ್ಯಂಡ್ ಆಲ್ ದ್ಯಾಟ್, ಪೀಪಲ್ ಸಿಟ್ಟಿಂಗ್ ಎನಿವೇರ್ ಕ್ಯಾನ್ ಕಂಟ್ರೋಲ್ ಎನಿಬಡಿ ಎಲ್ಸ್. ಇವತ್ತು ನೀವು ಮೊಬೈಲ್ ಇದ್ದರೆ ನೀವೇನು ಮಾಡ್ತೀರಾ, ನಮ್ಗೆ ಏನಿಷ್ಟ ಇಲ್ಲ ಎಲ್ಲರಿಗೂ ಗೊತ್ತಿದೆ. ಅಲ್ಲೆಲ್ಲ ಗೂಗಲಲ್ಲಿ ಕೂತ್ಕೊಂಡವ್ರಿಗೆ ದೇ ಕ್ಯಾನ್ ಟೆಲ್ ವಾಟ್ ಐ ಲೈಕ್, ವಾಟ್ ಐ ಡೋಂಟ್ ಲೈಕ್. ನೀವೆಲ್ಲ ನೋಡಿರ್ತೀರ, ಒಂದು ನಿಮಿಷ ಯಾವುದೋ ಮಾತಾಡ್ತಾ ಇದ್ದರೆ ಆ ಅಡ್ವರ್ಟೈಸ್ಮೆಂಟ್ ಬಂದಿರುತ್ತೆ 2 ನಿಮಿಷಕ್ಕೆ. ನೀವು ಯಾವುದೋ ಬಟ್ಟೆ ವಿಷಯ ಮಾತಾಡ್ತಾರೆ, 10 ಗಾರ್ಮೆಂಟ್ ಇಂಡಸ್ಟ್ರಿ ಫ್ಯಾಕ್ಟ್ರಿ ಆಫೀಸ್ಗಳಿಂದ ಅಥವಾ ಅಂಗಡಿಗಳಿಂದ ಮೆಸೇಜ್ ಬರುತ್ತೆ ನಿಮಗೆ. ಅಂದರೆ ಆ ಪ್ರೈವಸಿ ಎಲ್ಲ ಹೊರಟೋಗಿದೆ. ಇದೆಲ್ಲ ಯಾರೋ ಕೂತ್ಕೊಂಡು ಕಂಟ್ರೋಲ್ ಮಾಡ್ತಾಯಿದ್ರೆ ಭಾರತದಲ್ಲಿ ಅಥವಾ ನಮ್ಮ ದೇಶದಲ್ಲಿ, ನಮ್ಮ ಇಂಡಿಯನ್ನೆಸ್ ಏನು ಉಳಿಯಲ್ಲ. ದೇ ಕ್ಯಾನ್ ವರ್ಚುವಲಿ ಕಂಟ್ರೋಲ್ ಅಸ್. ಯು ಹ್ಯಾವ್ ಎ ಚಿಪ್ ಇನ್ ಯುವರ್ ಬ್ರೈನ್ ಟುಡೇ. ರೀಸೆಂಟ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟು ಏನಾಗ್ತಾ ಇದೆ, ವರ್ಚುವಲಿ ಮ್ಯಾನ್ ವಿಲ್ ನೋ ಮೋರ್ ಬಿ ಎ ಪ್ಯೂರ್ಲಿ ಬಯೋಲಾಜಿಕಲ್ ಅನಿಮಲ್. ದೇ ವಿಲ್ ಬಿ ಲಾಟ್ ಆಫ್, ಎಷ್ಟು ಚಿಪ್ಪುಗಳಿರುತ್ತೋ, ಎಷ್ಟು ಕಂಟ್ರೋಲ್ ಸಿಸ್ಟಮ್ಸ್ ಓಡಾಡ್ತಿರುತ್ತೋ, ಸ್ಕ್ಯಾನಿಂಗ್ ಸಿಸ್ಟಮ್ಸ್ ಒಳಗಡೆ ಮೈಕ್ರೋಮೋಟರ್ಸು ಕ್ಯಾಮರಾಸ್ ಓಡಾಡ್ತಿರ್ತೋ ಗೊತ್ತಿಲ್ಲ ಇನ್ನು 10 ವರ್ಷಕ್ಕೆ. ಅಂದರೆ ಸಂಬಡಿ ಕ್ಯಾನ್ ಕಂಟ್ರೋಲ್ ಸಂಬಡಿ ಎಲ್ಸ್ ಇನ್ ಅ ಮಚ್ ಮೋರ್ ಎಫೆಕ್ಟಿವ್ ವೇ ದ್ಯಾನ್ ವೆನ್ ದೇ ಡಿಡ್ ಇಟ್ ವಿತ್ ಸೋರ್ಡ್ಸ್ ಆ್ಯಂಡ್ ಗನ್ಸ್. ಬಂದೂಕ ಮತ್ತು ಕತ್ತಿ ಇಟ್ಕೊಂಡು ಮಾಡಿದ್ದಕ್ಕಿಂತ ಇನ್ನೂ ಸುಲಭವಾಗಿ ಕಂಟ್ರೋಲ್ ಮಾಡೋ ಕಾಲ ಬರ್ತಾಯಿದೆ. ಇಫ್ ವಿ ವಾಂಟ್ ಟು ಪ್ರೊಟೆಕ್ಟ್ ಅವರ್ ಸೆಲ್ಸ್, ಈವನ್ ದ್ಯಾಟ್ ಮಿನಿಮಮ್ ವಿ ಹ್ಯಾವ್ ಟು ಬಿ ಬೆಟರ್ ಇನ್ ಟೆಕ್ನಾಲಜಿ. ಸೊ ಆದ್ದರಿಂದ ಈ ದೇಶ ನಿಜವಾಗ್ಲೂ ಒಂದು ಶಕ್ತ ದೇಶ ಆಗಬೇಕಾದ್ರೆ, ಒಂದು ಕಡೆ ಐ ಆಮ್ ನಾಟ್ ಟಾಕಿಂಗ್ ಪಾಲಿಟಿಕ್ಸ್, ಆಯಿತು ಇವತ್ತು 5th ಲಾರ್ಜೆಸ್ಟ್ ಎಕಾನಮಿ ಇರ್ಬೋದು, 3rd ಆಗ್ಬೋದು, 2nd ಬರ್ಬೋದು, ಇದು ಏನೇ ಆಗಬೇಕಾದ್ರೂ ಅದಕ್ಕಿಂತ ಇನ್ನೂ ಹೆಚ್ಚು ಮೂಲಭೂತವಾಗಿ ವೈಜ್ಞಾನಿಕವಾಗಿ ಯೋಚನೆ ಮಾಡೋದು. ನೀವೆಲ್ಲ ಪರೀಕ್ಷೆ ಪಾಸ್ ಮಾಡಿದ್ರೆ ಬಂತು, ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಬಂದರೆ ಕೆಲಸ ಸಿಗುತ್ತೆ, ಡಿಸ್ಟಿಂಕ್ಷನ್ ಬಂದರೆ ಕೆಲಸ ಸಿಗುತ್ತೆ. ನಿಜ, ನಮ್ಮ ಕಾಲದಲ್ಲಿ ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಇದ್ದರೆ ನೋಡ್ತಿರ್ಲಿಲ್ಲ ಅವನ ಮಕಾನ. ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಇದ್ದವ್ರಿಗೆಲ್ಲ ಅಪಾಯಿಂಟ್ಮೆಂಟ್ ಲೆಟರ್ ಕೊಡ್ರಿ ಇನ್ನೋರು. ಇವಾಗ ಈವನ್ ಇಫ್ ಯು ಆರ್ ದಿ ಟಾಪರ್ ಫ್ರಮ್ MIT, ಸ್ಟ್ಯಾನ್ಫರ್ಡ್, IISc, IIT, NCBS ಯಾವುದಾದ್ರು ಆಗಲಿ 10 ಲೆವೆಲ್ ಇಂಟರ್ವ್ಯೂ ಇರುತ್ತೆ. ಅಂದರೆ ಪೀಪಲ್ ಡೋಂಟ್ ವಾಂಟ್ ಟು ಸಿ ಯುವರ್ ಮಾರ್ಕ್ಸ್ ಆ್ಯಂಡ್ ಮಾರ್ಕ್ಸ್ ಕಾರ್ಡ್ಸ್, ಪೀಪಲ್ ವಾಂಟ್ ಟು ಸಿ ಯುವರ್ ಕಾಂಪಿಟೆನ್ಸಿ. ನೀವು ಎಸ್ ಯಂಗ್ಸ್ಟರ್ಸ್, ಐ ವಾಂಟ್ ಟು ಸೇ, get those marks. They are not anything meaningful according to me. What is important is how you think and evolve and grow. ಈ ದೇಶ ಸುಭದ್ರವಾಗಿರಬೇಕಾದ್ರೆ, ಸಶಕ್ತವಾಗಿರಬೇಕಾದ್ರೆ, ಯು ಶುಡ್ ಆಲ್ ಇನ್ನೋವೇಟ್. ವಿಷಯ ತಿಳ್ಕೊಳ್ಳೋದು ಬೇರೆ, ವಿಷಯದ ಬಗ್ಗೆ ತಿಳ್ಕೊಳ್ಳೋದು ಬೇರೆ. ಐ ನೋ ಅಬೌಟ್ ದಿಸ್ ಅನ್ನೋದು ಬೇರೆ, ಐ ನೋ ದಿಸ್ ಅನ್ನೋದು ಬೇರೆ. ಸೊ ಇಟ್ಸ್ ಎ ಗ್ರೇಟ್ ಆಪರ್ಚುನಿಟಿ. ಈ ದೇಶದಲ್ಲಿ ಇರೋ ಪಾಪ್ಯುಲೇಷನಲ್ಲಿ ಎಷ್ಟು ಜನಕ್ಕೆ ಒಳ್ಳೆಯ ವಿದ್ಯಾಭ್ಯಾಸದ ವ್ಯವಸ್ಥೆ ಸಿಗುತ್ತೆ. ನೀವೆಲ್ಲ ಅದೃಷ್ಟಶಾಲಿಗಳು. ಅದೃಷ್ಟಶಾಲಿಗಳು ಅಂದರೆ ಇದು ಮಾತ್ರ ಈ ಸವಲತ್ತು ನಮಗೆ ಅಂತ ಅನ್ಕೋಬಾರ್ದು. ನಮ್ಗೆ ಅವಕಾಶ ಸಿಕ್ಕಿದೆ, ನಾವು ಜವಾಬ್ದಾರಿ ತೊಗೊಂಡು ಇಡೀ ದೇಶವನ್ನು ಸೈಂಟಿಫಿಕ್ ಟೆಂಪರ್ ಇಟ್ಕೊಂಡು ಯೋಚನೆ ಮಾಡಿ ಟೆಕ್ನಾಲಜಿ ದೇಶವನ್ನು ಮಾಡೋದ್ರೆ ಮಾತ್ರನೇ ಈ ದೇಶ ಪ್ರೋಗ್ರೆಸ್ ಆಗುತ್ತೆ. ಒಂದು ನೊಬೆಲ್ ಪ್ರೈಸ್ಗೆ ನಾವು ತೃಪ್ತಿ ಪಟ್ಕೋಬಾರ್ದು. ರಾಮನ್, ಹ್ಯಾವಿಂಗ್ ನೋನ್ ರಾಮನ್, ಐ ಹ್ಯಾಡ್ ಹರ್ಡ್ ಹಿಸ್ ಲೆಕ್ಚರ್ಸ್, ಐ ವಾಸ್ ಲಕ್ಕಿ. ಅವರೇನಾದ್ರು ಇವತ್ತು ಬದುಕಿದ್ರೆ ನನ್ನ ಹೆಸರು ಹೇಳಿದ್ರೆ ನಿಮಗೆ ಬಡ್ದು ಹಾಕ್ತೀನಿ ಅಂತ ಕೋಪ ಮಾಡ್ಕೊಳ್ಳೋರು. ಹಿ ವಾಸ್ ಸಚ್ ಎ ಟಫ್ ಮ್ಯಾನ್ ಇನ್ ದಿ ಸೆನ್ಸ್, ಯು ವುಂಟ್ ಟಾಲರೇಟ್ ಕೋಟಿಂಗ್ ಹಿಮ್ ಆಲ್ ದಿ ಟೈಮ್. ಎಷ್ಟರ ಮಟ್ಟಿಗೆ ಅಂದರೆ ಒಂದು 7th ಇಂಟರ್ನ್ಯಾಷನಲ್ ಕಾನ್ಫರೆನ್ಸ್ ಇನ್ ರಾಮನ್ ಸ್ಪೆಕ್ಟ್ರೋಸ್ಕೋಪಿ ನಡೀತಿಲ್ಲೇನೆ, ಸಾರಿ 7th ಇಂಟರ್ನ್ಯಾಷನಲ್ ಕಾನ್ಫರೆನ್ಸ್ ಇನ್ ರಾಮನ್ ಸ್ಪೆಕ್ಟ್ರೋಸ್ಕೋಪಿ ನಡೀತು. ಅವ್ರನ್ನ ದಯವಿಟ್ಟು ನೀವು ಬಂದು ಕೀನೋಟ್ ಅಡ್ರೆಸ್ ಕೊಡಿ ಅಂತ ಕೇಳಿದ್ರು. ರಾಮನ್ ಹೇಳಿದ್ರು, ಐ ಡೋಂಟ್ ವಾಂಟ್ ಟು ಕಮ್ ಅಂದರು. ಎಲ್ರಿಗೂ ಆಶ್ಚರ್ಯ, ರಾಮನ್ ಸ್ಪೆಕ್ಟ್ರೋಸ್ಕೋಪಿ ಬಗ್ಗೆ ಕಾನ್ಫರೆನ್ಸು, ನಾನು ಬರಲ್ಲ ಅಂತಲ್ಲ. ಅವ್ರು ಹೇಳಿದ್ರು, ನನಗೆ ಅರ್ಥ ಆಗಲ್ಲ ಇವತ್ತು, ನಾನು ಮಾಡಿದ ರಾಮನ್ ಸ್ಪೆಕ್ಟ್ರೋಸ್ಕೋಪಿ ಎಷ್ಟು ಫಂಡಮೆಂಟಲ್ ಅಂತ ಇವಾಗ ಗೊತ್ತಾಗ್ತಾ ಇದೆ. ಶಾರ್ಟ್ ಲೀವ್ಡ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಇರುತ್ತವಲ್ಲ, ಲೈಫ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಆ್ಯಂಡ್ ಸೆವೆರಲ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ವಿಚ್ ಕೀಪ್ ಆನ್ ಕಾನ್ಸ್ಟೆಂಟ್ಲಿ ಅಂಡರ್ಗೋಯಿಂಗ್ ಚೇಂಜ್, ಫೇಸ್ ಟ್ರಾನ್ಸಿಷನ್ಸ್ ಇರುತ್ತಲ್ಲ, ಪರ್ಟಿಕ್ಯುಲರ್ಲಿ ಲೈಫ್ ಸೈನ್ಸಸ್ಸಲ್ಲಿ ಅಥವಾ ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್, ದೇರ್ ಲೈಫ್ ಸ್ಪ್ಯಾನ್ ಈಸ್ ಸೋ ಶಾರ್ಟ್ ಅನ್ಲೆಸ್ ಯು ಹ್ಯಾವ್ ಫಾಸ್ಟ್ ಇನ್ಫಾರ್ಮೇಷನ್ ಗ್ಯಾದರಿಂಗ್ ಟೆಕ್ನಾಲಜೀಸ್. ಅಂದರೆ ಲೇಸರ್ಸು ಅದನ್ನು ಯೂಸ್ ಮಾಡಿದ್ರೆ ಮಾತ್ರ ಆ ಡೇಟಾ ತೊಗೊಳಕ್ಕೆ ಸಾಧ್ಯ. ಆ ಥರ ಟೆಕ್ನಾಲಜೀಸ್ ಎಲ್ಲ ಬಂದಿದೆ. ನೀವು ಕಾನ್ಫರೆನ್ಸಲ್ಲಿ ಮಾತಾಡಿದ್ದು ಅರ್ಧ ನನಗೆ ಅರ್ಥ ಆಗಲ್ಲ. ರಾಮನ್ ವುಡ್ ನಾಟ್ ಬಿ ಎ ಪರ್ಸನ್ ಆಫ್ ದಟ್ ಟೈಪ್, ಬಟ್ ಈ ಸೆಡ್ ನನಗೆ ಅರ್ಥವಾಗಲ್ಲ, ನನಗೆ ಮುಗಿಸಿರಾಗತ್ತೆ ಅಂತ. ಬಟ್ ಫೈನಲಿ ಹಿ ಕೇಮ್. ವೈ ಐ ಸೇ ದಿಸ್ ಇಸ್, change is the only constant thing. If you don't change fast, the nature will change you. You will become irrelevant. ಅದರಿಂದ I don't want to take too much time. Actually we are running short of time. We are behind the schedule. ಇಷ್ಟನ್ನು ಹೇಳಿ, ಉಜ್ವಲವಾದ ಭವಿಷ್ಯ ಇದೆ. Next century belongs to India. Next few decades, the best will happen in life sciences. Best will happen in life sciences, take it from me. ಒಂದು ಗ್ರೌಂಡ್ ಬ್ರೇಕಿಂಗ್ ಟೆಕ್ನಾಲಜಿ, ಇವಾಗ ಸೆಮಿಕಂಡಕ್ಟರ್ಸ್ ಬಂತಲ್ಲ, ಯಾರೋ ಒಂದು ಸೆಮಿಕಂಡಕ್ಟರ್ ತೊಗೊಂಡು ಎ ಮೆಟೀರಿಯಲ್ ವಿಚ್ ವಾಸ್ ಕನ್ಸಿಡರ್ಡ್ ಯೂಸ್ಲೆಸ್ ಬಿಕೇಮ್ ದಿ ಸೆಂಟರ್ ಫಾರ್ ಆಲ್ ಅವರ್ ನ್ಯೂ ಟೆಕ್ನಾಲಜೀಸ್. ದಟ್ ಈಸ್ ದಿ ಸೆಮಿಕಂಡಕ್ಟರ್ ಟೆಕ್ನಾಲಜಿ ಬಂದಾಗ. ಇನ್ನೊಂದು ಬರಬೇಕಾದ್ರೆ ಒಂದು 50 ವರ್ಷ ಬೇಕೇನೋ. ಬಟ್ ಇವಾಗ ಬಯಾಲಜಿ ಈಸ್ ಬೇಯಿಂಗ್ ಲುಕ್ಡ್ ವೆರಿ ಡಿಫರೆಂಟ್ಲಿ. Biology requires mathematics, biology requires chemistry. The modern biology is something very different. ದಯವಿಟ್ಟು dedicate your life for your happiness. Your happiness means enjoy what you learn, and India will be in one of these conditions in the next 10 years. ಏನಂದರೆ a country or a bunch of countries which will be working at the latest of technologies. Latest of technologies at terrible speeds. Another set of countries which can only absorb or digest and use those technologies. There will be a third layer of countries in the world which will be more or less like illiterates. They can't even absorb because of economic reasons and poor scientific manpower. ಈ 3 layer guarantee. Those who innovate, create, those who can understand, digest, those who will be deprived. We should not be in that bottom layer. At least we should be in the mid layer. But as future citizens of this country, you should make your efforts to be in the top layer. Thank you very much. I hope all of you will see a glorious technology era in this country. And I thank Mr. Ramesh and his team for inviting me. Thank Dr. Shiva Prasad, Girija and Anand for giving me this opportunity to be here. Thank you very much.
Thank you very much, sir, for your motivational speech. Still, we are waiting some more time to discuss. So, as sir told, we must fit in the knowledge-based era. We should definitely think over it. Thank you, sir, once again for your encouraging thoughts of the day. Now I request Dr. Girija Madam, former professor of chemistry from Gas College. Please share your thoughts on this occasion.
Dr. Ramesh introduce ಮಾಡುವಾಗ ನನ್ನನ್ನು ವಿಮೆನ್ ಬಿಹೈಂಡ್ ದ ಸಕ್ಸಸ್ಫುಲ್ ಮ್ಯಾನ್ ಅಂತ ಹೇಳಿದರು. ಐ ವಾಂಟ್ ಟು ಕರೆಕ್ಟ್ ಇಟ್, ವಿಮೆನ್ ಬಿಸೈಡ್ ದ ಸಕ್ಸಸ್ಫುಲ್ ಮ್ಯಾನ್ ಅಂತ. ಜಸ್ಟ್ ಐ ವಾಸ್ ಜೋಕಿಂಗ್. ನಾವು ಹೆಣ್ಮಕ್ಳು ಸ್ಟ್ರಾಂಗು ಗುರು ಅಂತ ಹೇಳೋಣ ಅಂತ, ಬಿಕಾಸ್ ಐ ವಾಂಟ್ ಟು ಟೆಲ್ ಆಲ್ ದ ಲೇಡೀಸ್ ಇನ್ ದ ಗ್ರೂಪ್ ಆ್ಯಂಡ್ ಗರ್ಲ್ಸ್ ಇನ್ ದ ಗ್ರೂಪ್, ವಿ ಆರ್ ನೋ ವೇ ಇನ್ಫೀರಿಯರ್ ಟು ಮೆನ್ ಅಂತ ಹೇಳೋಣ ಅಂತ ಅನ್ಕೊಂಡೆ. Yes, I was behind and also beside my illustrious husband. So, ಇವತ್ತಿನ ಥೀಮ್ ಇಂಡಿಜಿನಿಯಸ್ ಅಂತ ಹೇಳುವಾಗ ಐ ರಿಮೆಂಬರ್ಡ್ ದ ಪೇನ್ ಆ್ಯಂಡ್ ದ ಸ್ಟ್ರಗಲ್ ದೇ ಆಲ್ ವೆಂಟ್ ಥ್ರೂ. ಸೊ ಒಂದು ಚಿಕ್ಕದ ವಿಷಯ ಹೇಳಿ ಆಮೇಲೆ ನ್ಯಾಷನಲ್ ಸೈನ್ಸ್ ಡೇಗೆ ಬರ್ತೀನಿ. ದೇ ವರ್ ಗಿವನ್ ದ ಟಾಸ್ಕ್ ಆಫ್ ಡೂಯಿಂಗ್ 32 meter antenna indigenously. So my husband and his team took up this challenge. It was not easy, 360 crore project, and I know the way they struggled. Months together or manege barta irlilla. I think whenever I wanted to see my husband, I used to see antenna ಮೇಲ್ ಕೂತಿರೋದು, ಆಮೇಲೆ ಕೆಳಗೇನೋ ಕೆಲಸ ಮಾಡ್ತಾ ಇರೋದು, ಆ ಥರ. ನನಗೆ ಭಯ ಆಗಿತ್ತು, ಒಂದು ತಿಂಗಳು ಎಲ್ಲಿ ಹೋದ್ರಪ್ಪ ಇವರು, ಬ್ಯಾಗ್ ಇಡ್ತಿದ್ರು ಹೋಗ್ತಿದ್ರು. ಆ್ಯಂಡ್ ಐ ವಾಸ್ ರಿಯಲಿ ವರೀಡ್. ಆಲ್ ದ ಟೀಮ್ ದೇ ವರ್ ಆಲ್ ಸ್ಟ್ರಗ್ಲಿಂಗ್, ಅಂದರೆ ಇಯರ್ಸ್ ಮಾಡಿದ್ದಾರೆ. ಆ್ಯಂಡ್ ಇಂಡಿಜಿನಿಯಸ್ ಅಂದರೆ ಲೋಕಲ್ ಟ್ಯಾಲೆಂಟ್ನ ತೊಗೊಂಡು, ಇಂಡಿಯಾದಲ್ಲಿರೋ ಲೋಕಲ್ ಟ್ಯಾಲೆಂಟ್ ತೊಗೊಂಡು ಚಂದ್ರ ಮಂಗಳ ಗ್ರಹದಿಂದ ಬರೋ ಸಿಗ್ನಲ್ಸ್ನ ಈ ಒಂದು ಆ್ಯಂಟನ ರಿಸೀವ್ ಮಾಡಬೇಕು. ಅದು ಪಿಕ್ಚರ್ ಪರ್ಫೆಕ್ಟ್ ಆಗಿರಬೇಕು. ಸೊ ನನಗೆ ಯಾವಾಗಲೂ ನಾನು ತುಂಬ ಪಬ್ಲಿಷ್ ಮಾಡ್ತಾಯಿದ್ದೆ ಪಬ್ಲಿಕೇಶನ್, ಬಿಕಾಸ್ ಐ ವಾಸ್ ಅ ರಿಸರ್ಚರ್. ಸೊ ಯೂಸ್ ಟು ಟೀಸ್ ಮೀ, ಪೇಪರಲ್ಲಿ ಬರೆದ್ಬಿಟ್ರೆ ಆಗಲ್ಲಮ್ಮ, ಮಾಡಬೇಕದನ್ನು ಅಂತ. ಬಿಕಾಸ್ ಇಟ್ ಇಸ್ ನಾಟ್ ಈಸಿ. ಆ ಸಿಗ್ನಲ್ ಬರಬೇಕು ಇಲ್ಲಿಗೆ ಆ್ಯಂಟನಾಗೆ ಅಷ್ಟು ದೂರದಿಂದ. So it was not easy, and years ಇವರೆಲ್ಲ ಕಷ್ಟಪಡ್ತಾಯಿದ್ರು. I didn't know what he was doing because I was also busy as a lecturer, professor in the college. ಏನು ಗೊತ್ತಾಗಲಿಲ್ಲ. So one fine morning the Chandrayaan satellite was launched, and ಮನೆ ಫುಲ್ ಸುತ್ತ ಟಿ ವಿ ಕ್ರೂ ಬಂದಿತ್ತು, ಎಲ್ಲರೂ ಬಂದರು. ನನಗೇನು ಅರ್ಥ ಆಗ, ಅದು ಬೇರೆ ಡಿಲೇ ಬೇರೆ ಆಯಿತು, ಸ್ಯಾಟಲೈಟ್ ಲಾಂಚಿಂಗ್ ಸ್ವಲ್ಪ ಡಿಲೇ ಆಯಿತು. ಆವಾಗ ನಮ್ಮಮ್ಮ ತುಪ್ಪ ದೀಪ ಹಚ್ಬಿಡಮ್ಮ, ಸ್ವಲ್ಪ ಸ್ಯಾಟಲೈಟ್ ಲಾಂಚ್ ಆಗಲಿ, ಇಲ್ದಿದ್ರೆ ಅಳಿಯ ಬೇಜಾರು ಮಾಡ್ಕೊಂಡು ಬರ್ತಾರೆ ಅಂತ. ಅಳಿಯ ಬೇಜಾರು ಮಾಡ್ಕೊಂಡು ಬರ್ತಾರೆ ಅಂತ ನಮ್ಮ ಅಮ್ಮ ಹೇಳಿದ್ದು. ಸೊ ಸ್ವಲ್ಪ ಡಿಲೇ ಆಯಿತು. ಇಟ್ ವಾಸ್ ಗಾಡ್ಸ್ ಡಿಸಿಷನ್, ಐ ಥಿಂಕ್. ಎವ್ರಿಥಿಂಗ್ ವೆಂಟ್ ಆನ್ ವೆಲ್. ನನಗೇನು ಗೊತ್ತಿರಲಿಲ್ಲ ಏನು ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಅಂತ, ಬಿಕಾಸ್ ಐ ವಾಸ್ ಸೋ ಆ್ಯಂಗ್ರಿ ವಿತ್ ಹಿಮ್, ಬಿಕಾಸ್ ಈ ವಾಸ್ ನಾಟ್ ಅವೈಲೇಬಲ್ ಮಂತ್ಸ್ ಟುಗೆದರ್. ಸೊ ಐ ವಾಸ್ ರಿಯಲಿ ಫೀಲಿಂಗ್ ವೆರಿ ಸ್ಯಾಡ್. ಆ್ಯಂಡ್ ಟಿ ವಿ ಕ್ರೂ ಎಲ್ಲ ಬಂತು ಮನೆಗೆ. ಆಮೇಲೆ ಏನು ಮೇಡಮ್ ಸರ್ ಇಷ್ಟೊಂದು ಒಳ್ಳೆ ಕೆಲಸ ಮಾಡಿದ್ದಾರೆ ಏನು ಅಂತ. ಸೊ ಐ ವಾಸ್ ಇನ್ ವೆರಿ ಆ್ಯಂಗ್ರಿ ಮೂಡ್. ಅವ್ರಿಗೆ ಸ್ಯಾಟಲೈಟ್ಸ್ ಎಲ್ಲ ಟ್ರ್ಯಾಕ್ ಮಾಡೋಕ್ಕಾಗತ್ತೆ, ಮನೇಲಿರೋ ಸ್ಯಾಟಲೈಟ್ಸ್ನ ಟ್ರ್ಯಾಕ್ ಮಾಡೋಕ್ಕೆ ಟೈಮ್ ಇಲ್ಲ ಅಂತ ಹೇಳಿಬಿಟ್ಟಿದ್ದು. ಸೊ ಸಚ್ ವಾಸ್ ದ ರಿಯಾಕ್ಷನ್. ಲೇಟರ್ ಐ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್. ಐ ಕಾಲ್ಡ್ ಹಿಮ್, ಏನು ಊಟ ಮಾಡಿದ್ರಾ, ತಿಂಡಿ ತಿಂದ್ರಾ, ಏನು ಎಲ್ಲಿದ್ದೀರಾ? ಚಿತ್ತೂರಲ್ಲಿ ದೇವಸ್ಥಾನದಲ್ಲಿದ್ದೀನಿ. ಐ ಆಮ್ ಸೋ ಹ್ಯಾಪಿ. ಸಿಗ್ನಲ್ ಬಂತಮ್ಮ, ಚಂದ್ರಯಾನ್ ಸಿಗ್ನಲ್ಲು ಆ್ಯಂಟನಾಗೆ, ಫಸ್ಟ್ ಸಿಗ್ನಲ್ಲು. ನಿಂಗೆ ಪಿಕ್ಚರ್ ಕಳಿಸೆ. ಫಸ್ಟ್ ನೀವೇನು ತಿಂದಿದ್ದೀರಾ, ಏನು ಮಾಡ್ತಾಯಿದ್ದೀರಾ, ನಿಮ್ಮ ಹೆಲ್ತ್ ನೋಡ್ಕೊಂಡು. I didn't even hear what he is telling. Later on, I realized what he has done. ಸೊ ಎಲ್ಲರೂ ನನಗೆ ಕೂಲ್ ಡೌನ್ ಮಾಡ್ತಾರೆ, ಮೇಡಮ್ ಕಂಟ್ರಿಗೆ ನಿಮ್ಮ ಯಜಮಾನ್ರು ಬೇಕು. ನಾನಂದರೆ ಮನೆಗೆ ಯಜಮಾನ್ರು ಬೇಕಪ್ಪ, ಕಂಟ್ರಿಗೆಲ್ಲ ಫಸ್ಟು ಅವ್ರ ಮನೇಲಿರಲಿ. ಸಿ ಲೈಕ್ ದಿಸ್ ವೈಫ್ಸ್ ರೋಲ್ ವಾಸ್ ವೆರಿ ಡಿಫಿಕಲ್ಟ್. ಯಾವಾಗಲೂ ಆಫೀಸು, ಯಾವಾಗಲೂ ಇವ್ರದ್ದು ಟೆನ್ಷನ್ಸು. ಈಗೆಲ್ಲ ಸ್ವಲ್ಪ ಸೆಟ್ಲ್ ಆಗಿದೆ ಸ್ಪೇಸ್ ಮಿಷನ್ಸ್, ಆವಾಗೆಲ್ಲ ತುಂಬ ಕಷ್ಟ. ರಾತ್ರಿ ಏನೋ ಸರಿ ಇಲ್ಲ ಅಂದರೆ ಇವರೆಲ್ಲ ಒಳ್ಳೆ ಸೋಲ್ಜರ್ಸ್ ಥರ ಎಲ್ಲ ನಮ್ಮ ಲೇಔಟ್, ನಾವಿರೋದೇ ಇಸ್ರೋ ಲೇಔಟು, ಕಾರ್ಗಳಲ್ಲಿ ದಡಬಡ ಅಂತ ಹೋಗೋರು, ಆ ಟೆನ್ಷನ್ನು. ಆಮೇಲೆ 2 ಬ್ಯಾಗ್ ಇಟ್ಟಿದ್ವಿ. ಬ್ಲ್ಯಾಕ್ ಬ್ಯಾಗ್ ತೊಗೊಂಡೋದ್ರೆ ಅವರು ಇಂಡಿಯಾದಲ್ಲೇ ಇದ್ದಾರೆ ಅಂತ, ವೈಟ್ ಬ್ಯಾಗ್ ತೊಗೊಂಡೋದ್ರೆ ಅವರು ಇದರಲ್ಲಿ ಇಂಟರ್ನ್ಯಾಷನಲ್ ಫ್ಲೈಟ್ ಅಂತ. ಒಂದಿನ ಐ ವಾಸ್ ಇನ್ ಚನ್ನಪಟ್ಟಣ, ಐ ವಾಸ್ ಟೀಚಿಂಗ್. ಸೊ ಐ ಗಾಟ್ ಎ ಕಾಲ್. ಹಿ ಸೆಡ್, ನಾನು US ಹೋಗ್ತಾ ಇದ್ದೀನಿ ಮಧ್ಯಾಹ್ನ 1:30. ನಾನು ನೋಡ್ತೀನಿ 10 ಗಂಟೆ ಕ್ಲಾಸ್ ನಡೀತಾ ಇದೆ, ನಾನು ಇನ್ನೂ ಅಡುಗೆನೇ ಮಾಡಿರಲಿಲ್ಲ, ಬಿಕಾಸ್ ಹಿ ಹ್ಯಾಡ್ ನಾಟ್ ಟೋಲ್ಡ್. ಸೊ 10 to 11 ಕ್ಲಾಸ್ ಮುಗಿಸಿ 11 o'clock ಬಸ್ಸಲ್ಲಿ ಬಿಟ್ಟು 12 o'clock ಮನೆಗೆ ಬಂದೆ. ಮನೆಗೆ ಬಂದು ದಡ ಅಡುಗೆ ಮಾಡಿ 1 ಗಂಟೆಗೆ ಅವ್ರು ಹೊಟ್ಟೆಯವರು ಎಲ್ಲಿಗೆ ಹೋದರು, ಯಾವ ದೇಶಕ್ಕೆ ಹೋದರು ಏನೂ ಗೊತ್ತಿಲ್ಲ. ನಾನು ಮಾತಾಡ್ತೀನಿ ಆಮೇಲೆ ಅಂದರು. ಆಮೇಲೆ ಅವ್ರು ಡ್ರೈವರ್ಗೆ ಫೋನ್ ಮಾಡಿದೆ, ಎಲ್ಲಪ್ಪ ಯಾವುದೇ, ಮೇಡಮ್ ನಮ್ಗೆ ಟೈಮ್ ಇಲ್ಲ, ಇವಾಗ ಡ್ರೈವ್ ಮಾಡ್ತಾಯಿದ್ದೀನಿ ಅಂದರು. ಆಮೇಲೆ ಅವ್ರು ಹೋಗಿ ಬಂದಮೇಲೆ ಗೊತ್ತಾಯ್ತು ಅವ್ರು ಯಾವ ದೇಶಕ್ಕೆ ಹೋದ್ರು, ಏನು ಮಾಡ್ತಾಯಿದ್ದಾರೆ. ಸಿ ಅ ವೈಫ್ಸ್ ರೋಲ್ ವಾಸ್ ರಿಯಲಿ ವೆರಿ ಡಿಫಿಕಲ್ಟ್, ಐ ಆಮ್ ಟೆಲಿಂಗ್ ಯು. ಸಿ ಇಟ್ಸ್ ವೆರಿ ಈಸಿ, ಹಂಕಿ ಡೋರಿ, ದೂರದಿಂದ ನೋಡೋವಾಗ ಅನಿಸುತ್ತೆ, ಬಟ್ ಯು ಗೋ ಥ್ರೂ ದ್ಯಾಟ್. ಬಟ್ ಐ ಫೆಲ್ಟ್ ಲೇಟರ್, ಐ ರಿಯಲೈಸ್ಡ್ ಮೈ ರೋಲ್, ಆ್ಯಂಡ್ ಎವ್ರಿಬಡಿ ವಾಸ್ ಕಾಂಪ್ಲಿಮೆಂಟಿಂಗ್. ನೀವು ಅವರು ಬಿಸೈಡ್ ಹೇಳ್ತಾ ಇದ್ದೀರಾ. ಸೊ ಐ ಡೋಂಟ್ ನೋ ವೆದರ್ ಇನ್ ಎ ಗುಡ್ ವೇ ಆರ್ ಬ್ಯಾಡ್ ವೇ, ಬಿಕಾಸ್ ಐ ಆಮ್ ಎ ವೈಫ್, ಆಫ್ಟರ್ ಆಲ್ ಐ ಆಮ್ ಎ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್. ಸೊ ಎಷ್ಟು ಮನೆ, ಫ್ಯಾಮಿಲಿ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ದಿಸ್ ಸಕ್ಸಸ್ಫುಲ್ ಪೀಪಲ್ ಅಂತ ಐ ಆಮ್ ಜಸ್ಟ್ ಕಾಷನಿಂಗ್ ಫಾರ್ ಆಲ್ ಲೇಡೀಸ್ ಹೂ ಆರ್ ಹಿಯರ್. ಸೊ ನಾವು ಎಷ್ಟು ಇಂಪಾರ್ಟೆಂಟ್ ಅವ್ರು ಸಕ್ಸಸ್ಫುಲ್ ಆಗೋದಕ್ಕೆ, ಎಷ್ಟು ತಾಳ್ಮೆ ಇರಬೇಕು ನಮಗೆ. ಸೊ ಇವತ್ತು ಅವರು ಸಾಧನೆ ಮಾಡಿದ್ದಾರೆ, ಆಮೇಲೆ ನನಗೆಲ್ಲ ನಿಧಾನವಾಗೆಲ್ಲ ಅರ್ಥ ಆಗ್ತಾ ಬಂತು. ದೆನ್ ಐ ರಿಯಲೈಸ್ ಹೌ ಗ್ರೇಟ್ ವಾಸ್ ಮೈ ಹಸ್ಬೆಂಡ್. ಬಟ್ ಬಿಫೋರ್ ಐ ವಾಂಟೆಡ್ ಟು ಸೆಲಿಬ್ರೇಟ್, he just left us. So this is also another tragic ending. So these are the things which we all should understand. Indigenous anta heladaga today I just remembered. So now let me go for my talk. I just went for this. Dr. Ramesh, CEO KSDA, Dr. Nagraj sir, Chief Mentor Prayoga, Dr. Shivaprasad sir, Associate Professor, Associate Dean NCBS, and then Dr. Manja Nayak and Dr. Vinay, Senior Faculty, and all the dignitaries on the dais, off the dais, and my dear students. So, ellarigu namaskara. Indeed, I am honored to be here today on this special occasion, Science Day. I thank KSDA for giving me this opportunity to say a few words. So today we have assembled here for Science Habba, Science Jatre or Science Festival. ಎಷ್ಟು ಖುಷಿ ಆಗ್ತಾ ಇದೆ ಎಲ್ಲರಿಗೂ. ವರ್ಷಕ್ಕೊಮ್ಮೆ ಈ ಸೈನ್ಸ್ ಜಾತ್ರೆ ಮಾಡ್ತೀವಿ, ಸೈನ್ಸ್ ಫೆಸ್ಟಿವಲ್ ಮಾಡ್ತಾ ಇರ್ತೀವಿ. ಈ ಸೈನ್ಸ್ ಬಗ್ಗೆ ನಾವು ಏನು ತಿಳ್ಕೋಬೇಕು? ವಾಟ್ ಈಸ್ ಸೈನ್ಸ್? ಕೆಲವರು ಸೈನ್ಸ್ನ ಫನ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಸೈನ್ಸ್ ಇಸ್ ಫನ್. Science is curiosity. Science is a way of thinking much more than it is body of knowledge. Some say science is the bridge that connects our dreams with reality and empowers us to shape the future we envision. And what did our Sir C.V. Raman say? He said the essence of science is independent thinking, hard work, and not equipment. So what is science? This science is derived from the Latin word scientia. This means knowledge. So science andre nothing but knowledge. So do you all know what is the full form of science? The full form of science is systematic, comprehensive investigation and exploration of natural causes and effect. So when you say the word science, it can evoke different emotions. I think all of you remember your fun time in classrooms. And some recall hours spent grappling with complex formulae and concepts. I think this is what for us the science is. Today we are celebrating Science Festival. ಇದಕ್ಕೆ ನಾವೆಲ್ಲರೂ ಸಾಕ್ಷಿ ಆಗಿದ್ದೀವಿ, ಅದೇ ನಮ್ಮ ಖುಷಿಯಾಗಿದೆ. National Science Day is a celebration of scientific inquiry and discovery that marks the anniversary of the discovery of the Raman effect by Sir C.V. Raman. ಸೊ ಅದ್ರ ಬಗ್ಗೆ ಡಾಕ್ಟರ್ ನಾಗರಾಜ್ ಸರ್ ಹೇಳೇ ಇದ್ದಾರೆ. The Indian government officially recognized the discovery of Raman effect and declared February 28th as the National Science Day in 1986. The proposal from the National Council for Science and Technology Communication was primarily intended to commemorate the discovery of the Raman effect and then to inspire the scientific inquiry among the general population of India. But the first National Science Day was held in 1987. National Science Day is celebrated. Why it is celebrated? It is celebrated to spread the message of importance of science and its application. At least ಆ ದಿನ ಆದರೂ ಕೂಡ ನಾವು ಸೈನ್ಸ್ಗೋಸ್ಕರ ಜೀವ ಕೊಟ್ಟವ್ರನ್ನ, ಸೈನ್ಸ್ಗಾಗಿ ದುಡಿದವ್ರನ್ನ ನಾವು ನೆನೆಸ್ಕೊಂಡು ನಾವೇನು ಮಾಡಬೇಕು ಅಂತ ತಿಳ್ಕೊಂಡು ಮುಂದೆ ಏನು ಮಾಡಬೇಕು ಅಂತ ನಾವು ಯೋಚನೆ ಮಾಡಬೇಕು. This is what the importance of Science Day. Each year, the celebration is anchored around a specific theme. Last year, theme ಯಾರಿಗಾರ ಜ್ಞಾಪಕ ಇದೆಯಾ? Anybody? Last year theme? Global Science and Global Well-being. But this year, the Department of Science and Technology has anchored a special theme, that is indigenous technologies for Viksit Bharat. So, the Government of India aimed at highlighting different facets of scientific endeavor and its relevance to the society. These themes are carefully selected to appreciate the scientific community's contributions while also spreading awareness about the importance of science in daily life and encouraging the youth to pursue scientific research. The theme for 2024 National Science Day is Indigenous Technology for Viksit Bharat. The theme Indigenous Technologies for Viksit Bharat resonates deeply with India's aspirations for a self-reliant and developed future. As we celebrate the remarkable achievements of Indian scientists and innovators, it's crucial to recognize the transformative potential of homegrown technologies. That is very, very important. I am stressing on homegrown technologies. Hittalagida maddalla anta. Hittalagida madde. The cornerstone of a vibrant Atmanirbhar Bharat. This theme is a big opportunity for people and scientists both in India around the world. ಮೊದಲು ವಾಲ್ಸ್ ಕಟ್ಕೊಂಡ್ಬಿಡ್ತಾ ಇದ್ವಿ ಒಂದೊಂದು ದೇಶಕ್ಕೆ. ಈಗ ವಾಲ್ಸ್ನ ತೆಗೆದು ದೇರ್ ಆರ್ ನೋ ಬ್ಯಾರಿಯರ್ಸ್. ಈಗ ಒಂದು ಕಡೆಯಿಂದ ಇನ್ನೊಂದು ಕಡೆ ನಾವು ನಮ್ಮ ನಾಲೆಜನ್ನು ಶೇರ್ ಮಾಡ್ಕೋಬೋದು, ತಗೋಬೋದು, ಮುಂದೆ ಹೋಗ್ಬೋದು. To work together, they can join hands to help India and humanity as a whole. The theme also aims to make people aware of the importance of using our technology and innovations. As you all know, today's science is tomorrow's technology. Indigenization of science, technology, and innovation is a topic that needs more public attention and respect. And this strategy is centered around the goal. Innovation, why it is necessary? Ivat nivella innovation ಮಾಡಿ ತಗೊಂಬಂದಿದ್ದೀರಾ, you have got the prize. Then why it is important? It solves the global challenges, improves lives, and ensures a sustainable future for all of us. So the strategy is we are indigenization of science, technology and innovation. ಮೂರನ್ನು ಜೊತೆ ಸೇರಿಸ್ತಾ ಇದ್ದೀವಿ, ಸೈನ್ಸು, ಟೆಕ್ನಾಲಜಿ ಆ್ಯಂಡ್ ಇನೋವೇಷನ್. ಇದು ನಮ್ಮ ಥೀಮ್ ಈ ವರ್ಷದ್ದು. ಇದನ್ನು ನೀವೆಲ್ಲರೂ ಗಮನವಾಗಿ ಕೇಳ್ಕೋಬೇಕು, ಎಸ್ಪೆಷಲಿ ಸ್ಟೂಡೆಂಟ್ಸ್. Creating Atmanirbhar India is a central focus of theme, encouraging people to think more practically, scientifically and technologically. People should adopt technology aptly, but they should not misuse it. It should not cause threats to the nation and its residents. That we all should take care. Embracing indigenous knowledge system is not just a step towards development. It is a reaffirmation of India's identity and commitment to building a better future for generations to come. Indigenous technologies in India are deeply rooted in its cultural heritage, passed down through generations. Practices like organic farming, Ayurveda not only promote environmental sustainability but also uphold ancient traditions that are integral to India's identity. By preserving and promoting these techniques, India can safeguard its cultural identity while fostering the sustainable development. Practices such as rainwater harvesting, traditional architecture, herbal medicine rely on natural resources without causing harm to the environment. That we should take care here. By integrating indigenous practices into mainstream development strategies, India can mitigate the adverse effects of climate change and work towards a greener future. Now, chemistry synthesis alla ಮಾಡುವಾಗ we use clean solvents. ಎಷ್ಟೋ ನಮ್ಮ ರಿಸರ್ಚಲ್ಲಿ ಟಾಟಾರಿಕ್ ಆ್ಯಸಿಡ್ ಇದೆಲ್ಲ ಯೂಸ್ ಮಾಡುವಾಗ ಹುಣಸೆ ಹಣ್ಣು ನಿಂಬೆಹಣ್ಣು ಎಲ್ಲ ನಮ್ಮ ಸಿಂಥಸಿಸಲ್ಲಿ ನಾವು ಯೂಸ್ ಮಾಡಿದ್ದೀವಿ. ಸೊ ಟು ಅವಾಯ್ಡ್ ದ ಹಾರ್ಶ್ ಆಸಿಡ್ಸ್ ಆ್ಯಂಡ್ ಬೇಸಿಸ್. The importance of celebrating National Science Day extends far beyond merely remembering the groundbreaking Raman effect, as Dr. Nagaraj said. It reflects India's continuous pursuit of becoming a world leader in science. Bari Raman avrna matra nenskobardu, Raman avrna inspiration agi tagondu we should become a world leader in science. ಅದಕ್ಕೆ ನೀವೆಲ್ಲರೂ ಕಾಣ್ಕತ್ರು, ಯು ಆರ್ ದ ಅಂಬಾಸಿಡರ್ಸ್ ಆಫ್ ಸೈನ್ಸ್. ಕುಂತಲ್ಲಿ ನಿಂತಲ್ಲಿ ಎದ್ದಲ್ಲಿ ಸೈನ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡಬೇಕು. ಡೈನಿಂಗ್ ಟೇಬಲ್ ಮೇಲೂ ಸೈನ್ಸ್ ಮಾತಾಡಬೇಕು. ನಾವು ಎಸ್ಪೆಷಲಿ ಪೇರೆಂಟ್ಸ್, ಗ್ರ್ಯಾಂಡ್ ಪೇರೆಂಟ್ಸ್ ಆಗಿರೋರು ಜಾಸ್ತಿ ಸೈನ್ಸ್ ಬಗ್ಗೆನೇ ಮಾತಾಡ್ತಾ ಇರಬೇಕು, ಇನ್ಸ್ಟೆಡ್ ಆಫ್ ಟಾಕಿಂಗ್ ಅಬೌಟ್ ಮ್ಯೂಚುವಲ್ ಫಂಡ್ಸ್. ಯಾಕೆಂದರೆ ಕಾಮರ್ಸ್ ಕಡೆ ಹೋಗ್ತಾ ಇದ್ದಾರೆ ಸ್ಟೂಡೆಂಟ್ಸ್ ಎಲ್ಲರೂ. ದೇ ಶೈಯಿಂಗ್ ಅವೇ ಫ್ರಮ್ ಸೈನ್ಸ್. ಆಲ್ ಆಫ್ ದೆಮ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಾಮರ್ಸ್ ಬಿಕಾಸ್ ಕ್ವಿಕ್ ಬಫ್ ಸಿಗುತ್ತೆ. ಸೈನ್ಸ್ ತುಂಬ ಲೇಟು, ನಿಧಾನವಾಗಿ, ಬಿಕಾಸ್ ನೇಚರ್ ಅಂದರೆ ನಿಧಾನ. 9 ತಿಂಗಳು ಕಾಯಲೇಬೇಕು ಮಗು ಬರಬೇಕಾದ್ರೆ. ಸೊ ಹಿಯರ್ ದ ಡಿಲೇ ಆಫ್ ಟೈಮ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಹಿಯರ್. ಸೊ ಎಲ್ಲರೂ ಫಾಸ್ಟ್ಗೆ ಕಾಮರ್ಸ್ಗೆ ಹೋಗ್ತಾ ಇದ್ದಾರೆ. ಸೊ ದಟ್ಸ್ ಹೌ ಸೈನ್ಸ್ ಕಾಲೇಜಸ್ ಎಲ್ಲ ಮುಚ್ಚೋಗ್ತಾ ಇದೆ ಈಗ ಬ್ಯಾಂಗ್ಳೂರಲ್ಲಿ. So notably it helps shape India's scientific advancement by remembering the past, appreciating the present and thinking about the future. Many scientists have given their lives to science. We should remember them and celebrate their lives. So this is the importance of National Science Day. By observing National Science Day, more people will be familiar with science and its concepts as well as the latest scientific discoveries. Along with this, what other people should do, what our society should do. Especially our radio and television programs should aim at promoting scientific ideas and thinking. ಯಾವಾಗಲೂ ಕ್ರೈಮ್ ಸ್ಟೋರೀಸು, ಪಾಲಿಟಿಕ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡೋ ಬದಲು ಸೈನ್ಸ್ ಪ್ರೋಗ್ರಾಮ್ಸ್ನ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ತೋರಿಸೋದು ಮಾಡಿದ್ರೆ ಮಕ್ಕಳಿಗೂ ಒಳ್ಳೆಯದು, ಎಲ್ಲರಿಗೂ ಒಳ್ಳೆಯದು, ದೇಶಕ್ಕೂ ಒಳ್ಳೆಯದು. ಎಜುಕೇಷನ್ ಇನ್ಸ್ಟಿಟ್ಯೂಷನ್ ಶುಡ್ ಹೋಸ್ಟ್ ಸೈನ್ಸ್ ಅಫೇರ್ಸ್, ಇಂಟರಾಕ್ಟಿವ್ ವರ್ಕ್ಶಾಪ್ಸ್ ಮಾಡಬೇಕು, ಲೆಕ್ಚರ್ಸ್ ದೇ ಶುಡ್ ಅರೇಂಜ್. The most recent discoveries and developments in variety of scientific domains should be displayed at research institutions. ಬೆಳಗ್ಗೆ ಬಂದ ತಕ್ಷಣ ನಮ್ಮ ಬೋರ್ಡಲ್ಲಿ ಇವತ್ತೇನು ಡಿಸ್ಕವರಿ ಮಾಡಿದ್ದಾರೆ, ಇವತ್ತೇನಾಗಿದೆ ಸೈನ್ಸು, ಅದ್ರ ಬಗ್ಗೆ ಒಂದು ವಿಷಯನ ನಾವು ಡಿಸ್ಪ್ಲೇ ಮಾಡ್ಕೋಬೇಕು. ಆ್ಯಂಡ್ ಆ್ಯನ್ಯುವಲ್ ಎಕ್ಸಿಬಿಷನ್ಸ್ ಶುಡ್ ಬಿ ಕಂಡಕ್ಟೆಡ್. Children are more likely to develop a love for science when they participating in exciting events like science fairs. ಇವತ್ತು ನೀವೆಲ್ಲ ಬಂದಿದ್ದೀರಾ, you are all excited. Workshops and exhibitions. Here it is worth mentioning our KSTA is doing remarkable programs to encourage an inquisitive spirit in the minds of our next generation. I congratulate KSTA for that. We as parents and grandparents should start talking about science more often at home. ಚಿಕ್ಕ ವಿಷಯ, ಸ್ಟಾರ್ಟ್ ದ ಸ್ಮಾಲೆಸ್ಟ್, ಪ್ರಾಕ್ಟೀಸ್ ದ ಈಸಿಯೆಸ್ಟ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಚಿಕ್ಕ ವಿಷಯ, ಇವತ್ತು ಯಾಕೆ ಇಡ್ಲಿ ಬೆಂದಿಲ್ಲ, ಯಾಕೆ ಏನಾಯಿತು, ಏನು ಹಾಕಿದ್ರಿ, ಯಾಕೆ ಸೋಡಾ ಹಾಕಿದ್ರಾ, ಸೋಡಾ ಹಾಕಿದ ತಕ್ಷಣ ಇಡ್ಲಿ ಹಾಕಿದ್ರಾ, ಅದಕ್ಕೇನ ಸಾಫ್ಟ್ ಬಂತು, ಇವತ್ತು ಯಾಕೆ ಉಪ್ಪಿನಕಾಯಿ ಉಪ್ಪು ಜಾಸ್ತಿ ಆಗಿದೆ, ಉಪ್ಪು ಯಾಕೆ ಜಾಸ್ತಿ ಹಾಕಬೇಕು ಉಪ್ಪಿನಕಾಯಿಗೆ, ಉಪ್ಪು ಜಾಸ್ತಿ ಹಾಕಿದ್ರೆ ತುಂಬ ದಿನ ಬಾಳಿಕೆ ಬರುತ್ತೆ, ಯಾಕೆ ಆಸ್ಮಾಟಿಕ್ ಪ್ರೆಷರ್ ಇದರಿಂದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಲ್ಲಿ ಬೆಳೆಯಲ್ಲ ಉಪ್ಪು ಜಾಸ್ತಿ ಹಾಕಿದ್ರೆ. ಈ ಥರ ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್ ನಾವು ಹೇಳ್ತಾ ಹೋದರೆ ಇಟ್ ವಿಲ್ ಬಿ ನೈಸ್. ಆ್ಯಂಡ್ ಮೊಸರಿಗೆ ಹೆಪ್ಪಾಗೋವಾಗ ಬಾಳೆ ಎಲೆನ ಹಾಕ್ತಾರೆ ರಾತ್ರಿ, ನಮ್ಮ ಅಜ್ಜಿ ಕಾಲದಿಂದನೂ ಹಾಕ್ತಾಯಿದ್ರು. ಸೊ ಯಾಕೆ ಅಂತ ಈಗ ಗೊತ್ತಾಯಿತು, ಬಟ್ ಆವಾಗ ಗೊತ್ತಾಗ್ತಿರಲ್ಲ, ಬಾಳೆ ಎಲೆ ಹಾಕಿದ್ರೆ ಮೊಸರು ಚೆನ್ನಾಗೇ ಆಗುತ್ತೆ ಅಂತ. ಸೊ ದೀಸ್ ಆರ್ ಆಲ್ ಇಂಡಿಜಿನಿಯಸ್ ಡೆವಲಪ್ಮೆಂಟ್. So we have a big onus of spreading knowledge called science to make our nation strong. Atmanirbhar India, better enabled India, self-reliant India. ನಮ್ಮ ಮೇಲೆ ದೊಡ್ಡ ಜವಾಬ್ದಾರಿ ಇದೆ, ಎಲ್ಲರ ಮೇಲೂ. ಇಲ್ಲಿ ಕ್ಯಾಸ್ಟ್, ಕ್ರೀಡ್, ರಿಲಿಜನ್, ದೊಡ್ಡವ್ರು ಚಿಕ್ಕವರು ಯಾರೂ ಇಲ್ಲ, ಎಲ್ಲ we all have great responsibility in propelling science. In conclusion, I feel that National Science Day should stand as a testament to India's enduring legacy of scientific exploration, and its dedication to celebrating and promoting scientific achievements and knowledge. Dear students, think like a proton. Always be positive in your life. Thank you, one and all.
Thank you very much, madam. And you shared your pain, how much you really got during Shivkumar sir work. And sir is a very dynamic personality. We are very fortunate. We had work. We are very lucky. KST employees, we worked under his leadership for 2 years. Unfortunate, we lost him. The great icons like Shivkumar sir and Yuvarrao sir. So thank you very much, madam, for your wonderful lecture. As madam told, success in the future is never possible unless the actions of present are perfect and flawless. So we must, without any creed, color, religion, we should work for the nation building and the prosperity for the future. So now I request Professor Shiv Prasad sir to please brief 2 minutes. Briefly address the gathering, and sir's session is there, that's what.
ಥ್ಯಾಂಕ್ ಯು ಫಾರ್ ದ ಕೈಂಡ್ ಇಂಟ್ರಡಕ್ಷನ್. ಎಲ್ರಿಗೂ ಶುಭೋದಯ. ಕನ್ನಡದಲ್ಲಿ ಮಾತಾಡಿದರೆ ಯಾರಿಗಾದರೂ ಬೇಸರ ಇದೆಯಾ? ಯಾರಿಗಾದರೂ ಕನ್ನಡ ಅರ್ಥ ಆಗೋದಿಲ್ವಾ? ನನಗೆ ಆನುವಂಶಿಕ ಶಾಸ್ತ್ರ ಅಂದರೆ ಜೆನೆಟಿಕ್ಸ್ ಮತ್ತು ಕನ್ನಡ ಅಂದರೆ ಭಾರಿ ಇಷ್ಟ. ಕನ್ನಡದಲ್ಲಿ ನಾನು ಅಟ್ಲೀಸ್ಟ್ ವಿಜ್ಞಾನದ ಬಗ್ಗೆ ಒಂದು 100ಕ್ಕೆ ಹೆಚ್ಚು ಕವಿತೆ ಬರ್ತಿದ್ದೇನೆ. ಯಾರು ನನ್ನ ಲೆಕ್ಚರ್ಗೆ ನಿಲ್ತೀರಿ, ನಿಮಗೆ ಅಟ್ಲೀಸ್ಟ್ ಒಂದೊಂದು ಶೇರ್ ಮಾಡ್ತೀನಿ. ಹೆಚ್ಚಿನವ್ರು ಯಾರು ನೋಡಿರ್ಲಿಕ್ಕಿಲ್ಲ ಇದನ್ನು. ಈಗ ನಾನು ಮರ್ತೋಗೋ ಮುಂಚೆ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಅವರು ಈ ವಿಜ್ಞಾನದ ಹಬ್ಬದ ದಿವಸ ನಮ್ಮನ್ನೆಲ್ಲ ಇದೊಂದು ವಿಜ್ಞಾನವನ್ನು ಕೊಂಡಾಡುವ ದಿವಸ ನಿಮ್ಮೆಲ್ಲರ ಜೊತೆ ಇರಬೇಕಂತ ನನ್ನ ಕೂಡ ಆಮಂತ್ರಣ ಕೊಟ್ಟು ಕರೆದಿದ್ದಾರೆ. ಅವರಿಗೆ ತುಂಬ ಧನ್ಯವಾದಗಳು. ನಾನು ಏನು ಹೇಳಬೇಕಂತ ಇದೆ ಅದನ್ನು ಹೆಚ್ಚಿನದನ್ನು ಡಾಕ್ಟರ್ ನಾಗರಾಜ್ ಮತ್ತು ಡಾಕ್ಟರ್ ಗಿರಿಜಾ ಅವರು ಆಗಲೇ ಹೇಳಿ ಆಗಿದೆ. ನಾನು 4 ವಿಷಯ ಹೇಳ್ಬೇಕಂತ ತಿಳ್ಕೊಂಡಿದ್ದೇನೆ. ಅದರಲ್ಲಿ 2 ನಿಮಿಷದಲ್ಲಿ ಏನು ಹೇಳ್ತೇನೆ ಅಂತ ನೋಡಿ 3 ಮುಗಿಸಿದ್ದಾರೆ, ಒಂದೊಂದು ಮಾತ್ರ ಹೇಳ್ತೇನೆ. ಈಗ ನಿಮಗೆಲ್ಲ ಗೊತ್ತಿದೆ ಇಲ್ವಾ, 1947 ನಮಗೆ ಸ್ವಾತಂತ್ರ್ಯ ಸಿಕ್ಕಿದಾಗ ನಮ್ಮ ದೇಶದಲ್ಲಿ ಜನಸಂಖ್ಯೆ ಏನು ಕಮ್ಮಿ ಇರಲಿಲ್ಲ, 30-40 ಕೋಟಿ ಇತ್ತು. ಅವರ ಜನರ ಜೀವನ ಎಷ್ಟು ಟೈಮ್ ಇತ್ತು ಗೊತ್ತುಂಟ ನಿಮಗೆ? 30 ವರ್ಷ. ಹೆಂಗಸರದ್ದು 26 ವರ್ಷ. ನಾವು ಅಲ್ಲಿಂದ ಇಷ್ಟರವರೆಗೆ ಬರಲಿಕ್ಕೆ ತುಂಬ ಹೆಚ್ಚಾಗಿ ಕಾರಣ ಅಂದರೆ ವಿಜ್ಞಾನ ಮಾತ್ರ. ನಮ್ಮ ಜೊತೆ ನಮಗಿಂತ ಮುಂಚೆ ಸ್ವಾತಂತ್ರ್ಯ ಸಿಕ್ಕಿದ ಎಷ್ಟೋ ದೇಶಗಳಿದ್ದಾವೆ, ಇನ್ನೂ ಎಲ್ಲೋ ಹಿಂದೆ ಇದ್ದಾವೆ. ನಿಮಗೆ ಗೊತ್ತಿರ್ಬೋದು, ಈಗ ಕೆಲವು ತೈಲದಿಂದ ತುಂಬ ಹಣ ಮಾಡಿದ್ದ ದೇಶಗಳೆಲ್ಲ ಈಗಲೂ ನಮ್ಮಲ್ಲಿಂದ ಒಂದು IIT ಬ್ರಾಂಚ್ ಮಾಡಿ ಅಂತ ನಮ್ಮ ಕಾಲಿಡ್ತಾ ಇದ್ದೀವಿ ಅಲ್ವಾ? ಹಾಗೆಲ್ಲ ನೋಡಿದರೆ ಆ ದೇಶಗಳಿಂತೆಲ್ಲ ನಾವು ತುಂಬ ಮುಂದೆ ಬರಲಿಕ್ಕೆ ಒಂದೇ ಕಾರಣ, ಇದ್ದ ಮೂಢನಂಬಿಕೆ ಅಪನಂಬಿಕೆ ಎಲ್ಲ ತೆಗೆದು ತಂತ್ರಜ್ಞಾನ ಉಪಯೋಗ ಮಾಡಿ ನಾವು ಇಷ್ಟು ಮುಂದೆ ಬರಬೇಕಾದ್ರೆ ವಿಜ್ಞಾನ ಮಾತ್ರ ಕಾರಣ ಅಂತ ಹೇಳಲಿಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಈಗ ಅದು ನಾವು ಮಾಡಿದ ಸಾಧನೆಯಲ್ಲಿ ಏನಾದರೂ ಕಮ್ಮಿ ಇದೆಯಾ? ತುಂಬ ಕಮ್ಮಿ ಇದೆ. ನಮಗಿಂತ ತುಂಬ ಚಿಕ್ಕ ದೇಶಗಳಾದ ದಕ್ಷಿಣ ಕೊರಿಯಾ ಅಂತ ನೋಡಿ, ಎಷ್ಟು ಚಿಕ್ಕ ದೇಶ, ಎಷ್ಟು ಮುಂದೆ ಬಂದಿದೆ ಅದು ಬರೀ ತಂತ್ರಜ್ಞಾನದಿಂದ. ಅದು ಯಾಕೆ ಅಂತ ಹೇಳಿದರೆ, ಈ ತಂತ್ರಜ್ಞಾನ ಅಂತ ಮಾಡಬೇಕಾದ್ರೆ ಒಂದು ಉತ್ತಮವಾದ ಕುತೂಹಲದಿಂದ ಬಂದಂಥ ಬೇಸಿಕ್ ಸೈನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಲ್ಲ, ಅದು ತುಂಬ ಅಗತ್ಯ ಇದೆ. ನಮ್ಮ ತುಂಬ ಈ ಸೀನಿಯರ್ಸ್ ಎಲ್ಲ ಅದಕ್ಕೆ ತುಂಬ ಕಷ್ಟಪಟ್ಟು ಆ ಥರದ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಎಲ್ಲ ತಂದಿದ್ದಾರೆ. ಅದು ತುಂಬ ಅಗತ್ಯವಾಗಿರುವಂಥದ್ದು. ಯಾವ ದೇಶವೇ ಇರಲಿ, ಮೂಲಭೂತ ವಿಜ್ಞಾನ ಇದೆ ಯಾವುದು, ಕುತೂಹಲ ಹಿಡ್ಕೊಂಡು ಅನುಸರಿಸಿಕೊಂಡು ಹೋದಂಥ ವಿಜ್ಞಾನ ಯಾವುದಿದೆ, ಅದರಷ್ಟು ಮಹತ್ವದ ವಿಜ್ಞಾನ ಯಾವುದೂ ಇಲ್ಲ. ಅದರಿಂದಾಗಿಯೇ ನಮಗೆ ಈ ಆನುವಂಶಿಕ ಅಪ್ಲೈಡ್ ರಿಸರ್ಚ್ ಯಾಕೆ ಬರ್ತದೆ. ಈಗ ಇನ್ನೋವೇಷನ್ ಮಾಡಬೇಕಾದ್ರೆ ತಂತ್ರಜ್ಞಾನದಿಂದಾಗಿ ಅದಕ್ಕೆ ಈ ಮೂಲಭೂತ ವಿಜ್ಞಾನ ತುಂಬ ಅಗತ್ಯ. ಈಗ ಈ ಮೂಲಭೂತ ವಿಜ್ಞಾನಕ್ಕೆ ಏನು ಅಗತ್ಯ ಇದೆ ಮನುಷ್ಯನಿಗೆ? ಈಗ ಕ್ಲಾಸಲ್ಲಿ ಯಾರು ಫಸ್ಟ್ ರ್ಯಾಂಕ್ ಬರ್ತಾರೆ ಅವರಿಗೆ ಇದು ಬೇಕಾಗಿಲ್ಲ, ಅವರು ಇದರಲ್ಲಿ ಹುಷಾರಾಗಿರ್ಬೇಕಂತಿಲ್ಲ. ಈಗ ನಾವೀಗ ಒಂದು 20 ವರ್ಷದಿಂದ ರಿಸರ್ಚ್ ಮಾಡುವಾಗ ನೋಡ್ದೇನೆಂದರೆ, ಅದು ಯಾರಿಗೆ ಇರ್ತದಂತ ನಮಗೆ ಗೊತ್ತಾಗೋದಿಲ್ಲ. ಈ ಒಂದು ಕೆಪಾಸಿಟಿ ಅದು ಯಾರಿಗೆ ಇರ್ತದೆ. ಕೆಲವೊಮ್ಮೆ ತುಂಬ ಹಳ್ಳಿಯಲ್ಲಿ ಬಂದವರಿಗೆ ತುಂಬ ಇರ್ತದೆ. ಅವರಿಗೆ ಏನು ಬೇಕಂತ ಹೇಳಿದರೆ ಒಂದು ಲಾಜಿಕ್ ಅಂತ ಬೇಕು, ತರ್ಕ. ಈ ತರ್ಕ ಅನ್ನೋದು ನಿಮಗೆ ನಳಂದ ತಕ್ಷಿಲ ಎಲ್ಲ ಹಳೆಯ ವಿಶ್ವವಿದ್ಯಾಲಯ ಬಗ್ಗೆ ಓದಿದರೆ ಅಲ್ಲಿ ಕೂಡ ಒಂದು ಕಲಿಸುವಂಥ ಒಂದು ವಿಷಯ. ಈಗ ತುಂಬ ಮಂದಿಗೆ ತರ್ಕ ಏನಂತ ಗೊತ್ತಾಗದಿದ್ದರೆ, ಈಗ ನಾಳೆ ಏನೋ ಬೆಂಗಳೂರಲ್ಲಿ ಸಂಜೆ ಮಳೆ ಬರ್ಬೋದು ಅಂದರೆ ಇವತ್ತೇ ನಾನು ನಾಳೆ ಬಿಳಿ ಬಟ್ಟೆ ಹಾಕೋದಿಲ್ಲ ಅಂತ ಯೋಚನೆ ಮಾಡ್ತಿರಲ್ಲ, ಅದೇ ತರ್ಕ. ಇದು ನಿಮಗೆ ತುಂಬ ಸುಲಭವಾಗಿ ಎಣಿಸ್ಬೋದು. ಆದರೆ ಇದಿಲ್ಲದೆ ಯಾವ ವಿಜ್ಞಾನದಲ್ಲಿ ಈ ಥರ ಬೇಸಿಕ್ ಸೈನ್ಸನ್ನು ಮಾಡಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ. ಇದು ಹೆಚ್ಚಾಗಿ ಯಾರು ಇರ್ತದೆ, ತುಂಬ ಮಂದಿಗೆ ಮಾತಾಡುವವರಿಗಲ್ಲಿ ಇರ್ತದೆ, ತುಂಬ ಓದುವವರಿಗೆ ಇರ್ತದೆ, ಅವರಿಗೆ ಆ ಜ್ಞಾನ ಬಂದಿರ್ತದೆ, ತುಂಬ ಮಂದಿ ಹತ್ರ ಚರ್ಚೆ ಮಾಡುವವರಿಗೆ ಆಟೋಮೆಟಿಕ್ಕಾಗಿ ಇರ್ತದೆ. ಅದಕ್ಕಾಗಿ ಯಾರು ರ್ಯಾಂಕ್ ಬರ್ತಾರೆ ಅವರು ಈ ಥರ ವಿಜ್ಞಾನದಲ್ಲಿ ಮುಂದೆ ಹೋಗ್ತಾರಂತ ನಮಗೆ ಧೈರ್ಯವಾಗಿ ಹೇಳಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ. ಕೆಲವೊಮ್ಮೆ ನನ್ನಂಥವರು ಕೊನೆ ಬೆಂಚಲ್ಲಿದ್ದವರು ಕೂಡ ಮುಂದೆ ಬರ್ತಾರೆ. ಹೌದಾ? ಅದಕ್ಕೆ ನನಗೆ ಒಂದು ಇನ್ನೊಂದು ಹವ್ಯಾಸ ಇದೆ. ಈ ಚಿಕ್ಕ ಚಿಕ್ಕ ಶಾಲೆಗಳಿಗೆ ಹೋಗಿ ವಿಜ್ಞಾನದ ಬಗ್ಗೆ ಏನಾದರೂ ಮಾತಾಡೋದು. ನನಗೆ ಒಂದು 6ನೇ ಕ್ಲಾಸಲ್ಲಿರುವ ಒಂದು ಚಿಕ್ಕ ಹುಡುಗಿ, ನಾನೊಂದು ದಿವಸ ವೈವಿಧ್ಯತೆ ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದ್ದೆ. ಆಗ ಏನು ಹೇಳಿದ್ಲು ಅಂದರೆ, ಈ ಎಲ್ಲಿ ವೈವಿಧ್ಯತೆ ಹೆಚ್ಚಿರ್ತದೆ ಅಲ್ಲಿ ಮಾತ್ರ ಇನ್ನೂ ವೈವಿಧ್ಯತೆ ಹೆಚ್ಚಾಗ್ಬೋದಲ್ವಾ. ಅದು ನಾನು ಏನು ಹೇಳ್ತಾ ಇದ್ದರೆ ಅಂದರೆ, ಈ ಕೀಟಗಳ ಸಂಖ್ಯೆ ಹೆಚ್ಚಿದ್ದರೆ ಕೀಟಗಳ ಪರಾಗಸ್ಪರ್ಶ ಹೆಚ್ಚು ಮಾಡಿ ಅದರಿಂದಾಗಿ ಹೆಚ್ಚು ವೈವಿಧ್ಯತೆ ಆಗ್ಬೋದು. ಆದರೆ ಕೀಟಕ್ಕೆ ಕೂಡ ಬೇರೆ ಬೇರೆ ಗಿಡಗಳು ಬೇಕು. ಈಗ ಹೆಚ್ಚು ವೈವಿಧ್ಯತೆ ಎಲ್ಲಿರ್ತದೆ ಅಲ್ಲಿ ಮಾತ್ರ ಹೆಚ್ಚು ವೈವಿಧ್ಯ. ಇದು ನನಗೆ 6ನೇ ಕ್ಲಾಸಲ್ಲಿರುವ ಒಂದು ಚಿಕ್ಕ ಹುಡುಗಿ ಹೇಳಿದ್ದು, ಒಂದು ಚಿಕ್ಕ ಹಳ್ಳಿ ಸಕಲೇಶ್ಪುರದ ಬಳಿಯದ್ದು. ಇದು ಏನು ಹೇಳ್ತಾ ಇದ್ದಂದರೆ, ವಿಜ್ಞಾನದಲ್ಲಿ ಮುಂದೆ ಹೋಗ್ಲಿಕ್ಕಾಗ್ತದೆ ನಮಗೆ ಅಷ್ಟು ಸುಲಭವಾಗಿ ಹೇಳಲಿಕ್ಕಾಗೋದಿಲ್ಲ. ಈಗ ರಿಸರ್ಚ್ಗೆ ಬಂದವರು ಕೂಡ 1 ವರ್ಷ 2 ವರ್ಷ ನಮಗೆ ಗೊತ್ತಾಗೋದಿಲ್ಲ ಅವರಿಗೆ ಆ ಒಂದು ಪವರ್ ಇದೆಯಾ ಇಲ್ವಾ ಅಂಥೇಳಿ. ಅದರಿಂದಾಗಿ ನಿಮಗೆ ಕೂಡ ಇರ್ಬೋದು. ದಯವಿಟ್ಟು ಯಾರಿಗೂ ವಿಜ್ಞಾನ ಇಲ್ಲ, ಇವರು ಫಸ್ಟ್ ರ್ಯಾಂಕ್ ಬಂದಿಲ್ಲ ಹಾಗಾಗಿ ಇವರಿಗೆ ವಿಜ್ಞಾನ ಕಷ್ಟ, ಆ ಥರದ ಯೋಚನೆ ದಯವಿಟ್ಟು ಮಾಡಬೇಡಿ. ನಿಮಗೆಲ್ರಿಗೂ ವಿಜ್ಞಾನದಲ್ಲಿ ಆಸಕ್ತಿ ಇದ್ದರೆ, ನಿಮಗೆ ಜ್ಞಾನದಲ್ಲಿ ಆಸಕ್ತಿ ಇದ್ದರೆ ನೀವು ಖಂಡಿತ ಈ ಥರದ ಕುತೂಹಲ ಚಾಲಿತ ವಿಜ್ಞಾನ ನಿಮಗೆ ಮಾಡಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇದೆ. ಅದು ಮಾಡದಿದ್ದರೆ ನಮಗೆ ಯಾವ ತಂತ್ರಜ್ಞಾನವೂ ಸಿಗೋದಿಲ್ಲ. ಸೊ ದಯವಿಟ್ಟು ನಿಮ್ಗೆ ಯಾರು ಪರಿಚಯ ಇದ್ದರೂ ಮನೆಯಲ್ಲಿ, ಈಗ ಮೇಡಮ್ ಹೇಳಿದರು ಮನೆಯಲ್ಲೆಲ್ಲ ನೀವು ವಿಜ್ಞಾನ ಮಾತಾಡಬೇಕಂತ. ವಿಜ್ಞಾನ ಅನ್ನೋದೇ ಒಂದು ಜೀವನದ ಒಂದು ವೇ ಆಫ್ ಲೈಫ್. ಅದನ್ನು ನೀವು ಅಳವಡಿಸ್ಕೊಂಡ್ರೆ ಸಾಮಾಜಿಕ ಪ್ರಾಬ್ಲಮ್ಗಳಿಗೂ ನಿಮಗೆ ಅದರಲ್ಲಿ ಉತ್ತರ ಸಿಗ್ತದೆ. ಆದಷ್ಟು ವಿಜ್ಞಾನವನ್ನು ಯೋಚಿಸಿ ಅಂತೇಳಿ ಹೇಳ್ಕೊಂಡು ನನ್ನ ಒಂದು 5 ನಿಮಿಷ 2 ನಿಮಿಷ ಹೆಚ್ಚಾಯಿತು, 3 ನಿಮಿಷ ಆಯಿತು, ಮಾತನ್ನು ಮುಗಿಸ್ತೇನೆ. ಧನ್ಯವಾದ.
ಧನ್ಯವಾದಗಳು ಶಿವಪ್ರಸಾದ್ ಸರ್, ತುಂಬ ಚೆನ್ನಾಗಿ ಮಾತಾಡಿದ್ರಿ. ವಿಜ್ಞಾನದ ಆಸಕ್ತಿ ಎಲ್ಲರೂ ಬೆಳೆಸ್ಕೊಳ್ಳಿ ಅಂತ ಹೇಳಿದ್ರಿ. ಥ್ಯಾಂಕ್ ಯು. ವೆಲ್ಕಮ್ ಟು ಪ್ರೊಫೆಸರ್ ಜಗದೀಶ್ ಸರ್, ರೀಜನಲ್ ಡೈರೆಕ್ಟರ್, ಕಾಲೇಜ್ ಆಫ್ ಹಾರ್ಟಿಕಲ್ಚರ್, ಆ್ಯಂಡ್ ವೆಲ್ಕಮ್ ಟು ಡಾಕ್ಟರ್ ದೊಡ್ಡಬಸಪ್ಪ ಸರ್ ಆಲ್ಸೋ ಫಾರ್ ದಿಸ್ occasion. ಈಗ KSTA ಇನೋವೇಷನ್ ಅವಾರ್ಡ್ ಪುರಸ್ಕೃತರು. ಮೊದಲಿಗೆ
KSTA ಇನೋವೇಷನ್ ಡಿಸ್ಕವರಿ ಅವಾರ್ಡ್ ಪ್ರತಿ ಪ್ರೈಸನ್ನು ₹10,000 ಮತ್ತು ಸರ್ಟಿಫಿಕೇಟ್ ಕೊಡ್ತೀವಿ. ಮೊದಲಿಗೆ ಪ್ರೊಫೆಸರ್ ಯು ಆರ್ ರಾವ್ ಪುರಸ್ಕಾರ. ಅನನ್ಯ ಟಿ, ಟೈಟಲ್ ಆಫ್ ದಿ ಪ್ರಾಜೆಕ್ಟ್ ವೇರಿಯೇಷನ್ ಆಫ್ ಫಿಶ್ ಸ್ಕಿನ್ ವೇಸ್ಟ್ ಇಂಟು ಕ್ವಾಲಿಟಿ ಲೆದರ್ ಆ್ಯಂಡ್ ವ್ಯಾಲ್ಯೂ ಅಡಿಷನ್, ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಆಫ್ ಫಿಶ್ ಪ್ರೊಸೆಸಿಂಗ್ ಟೆಕ್ನಾಲಜಿ, ಕಾಲೇಜ್ ಆಫ್ ಫಿಶರೀಸ್, ಮಂಗಳೂರು. Anand K. Joshi, innovative heat dispersion technique in lithium ion battery using nano enhanced PCM, President University, Bangalore. Nikhil N. Novel technique of complete plant regeneration of Morinda citrifolia, noni through leaves. College of Forestry, Sirsi. HR Samira, exploring the theoretical potential of Dunalina salina formulation against cardiovascular complication using marine and zebrafish model. Faculty of Pharmacy, MS Ramaya, University of Applied Sciences, Bengaluru. ಈಗ ಡಾಕ್ಟರ್ ಎಸ್ ಕೆ ಶಿವಕುಮಾರ್ ಪುರಸ್ಕಾರ. ಈ ಪುರಸ್ಕಾರನು ಪ್ರತಿಯೊಬ್ಬರಿಗೂ 10,000 ನಗದು ಪುರಸ್ಕಾರ ಮತ್ತು ಸರ್ಟಿಫಿಕೇಟ್ ಇರುತ್ತೆ. ಮೊದಲಿಗೆ ಸ್ವರೂಪ್ ಎ, ಫೇಸಿಯಲ್ ರೆಕಗ್ನಿಷನ್ ಸೆಕ್ಯೂರಿಟಿ ಸಿಸ್ಟಮ್ ಫಾರ್ ವಿಜುವಲಿ ಇಂಪೇರ್ಡ್ ಪೀಪಲ್, BMS ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಫ್ ಟೆಕ್ನಾಲಜಿ ಆ್ಯಂಡ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್, ಬೆಂಗಳೂರು. Some of you can come here, this side. Prashant S. Utkali, Smart Anti-Theft Control System using IoT. BLDs Vachana Pitamba, Dr. PG Halakatti, College of Engineering and Technology, Vijayapura, Karnataka. Mini M, a study of biodegradable sanitary pads using pineapple fibers, Maharani Kalstar University, Bengaluru. Siddharth Joshi, Biofuel Filter Sustainable Leap Towards Water Sanitation for Make in India Initiative, RV College of Engineering, Bengaluru.
this project is sponsored by KSTA, and this is of its own kind. My students have the examination today, so we are taking proud of it. Thanks a lot to KSTA for all the encouragement. Looking forward. Myself, Dr. Ashwani Sharma, who is the co-PI for this, and Dr. Nagashri N. Rao is the principal investigator for this.
Mayur V, D2D, Waste Management and Reward System, Vidyavartaka College of Engineering, Mysuru. Shashank CR, Life Saving Concrete, Vidyavardhaka College of Engineering, Mysore.
We have another 6 prizes, KSTA Innovation Award for teaching faculty and general public. First, Dr. Ummi Seema Hen for the development and assessment of polyherbal formulations for anti-obesity property in obesity-induced laboratory rats. College of Horticulture, Bangalore, and University of Horticultural Sciences, Bagalkot. Congratulations. Dr. P. Sudhir, SJSA Institute of Technology, Chikballapura, for the development of design and development of low-cost portable ventilator. Next awardee is Dr. Next award is Dr. Nagaraju Kottam from MS Ramaya Institute of Technology, Bangalore, for modified CDS nano composite for solar light driven hydrogen production. MS Ramaya Institute of Technology. Next award is Dr. Vinay B. Raghavendra from Teresian College, Mysore, for cellulose based biocompatible conductive fabric in converting sweat ions into electrolytes and use as electric energy. Next winner is Dr. Mallamma Reddy from Rani Chennamma University, Belagavi, for the artificial intelligence and information retrieval algorithms for cross language voice translation in natural language processing. Next winner is Dr. Srinivas Deshpande from University of Agricultural Sciences, GKVK, Bangalore, for the development of amylose sensor for assessing aging of rice. So congratulations to all the winners. Now I request
ಸಭಾ ಮರ್ಯಾದೆ ಅಲ್ಲ, ನನಗೆ ಅನಿವಾರ್ಯ ಕಾರಣಕ್ಕಾಗಿ ಇಷ್ಟೊತ್ತಿಗೆ ನಾನು ಇನ್ನೊಂದು ಕಡೆ ಇರಬೇಕಾಗಿತ್ತು, ಹೊರಡ್ತಾ ಇದ್ದೀನಿ, ದಯವಿಟ್ಟು ಕ್ಷಮೆ ಇರಲಿ. ನಮಸ್ಕಾರ.
ತಮಗೀಗ ನೀಡಿರುವ ಸರ್ಟಿಫಿಕೇಟಲ್ಲಿ ನಿಮ್ಗಳ ಗೈಡ್ಗೂ ಸರ್ಟಿಫಿಕೇಟ್ ಇದೆ, ಎಕ್ಸ್ಟ್ರಾ ಇರೋದನ್ನು ತಮ್ಮ ಗೈಡ್ಗಳಿಗೆ ತಲುಪಿಸ್ಬೇಕಾಗಿ ವಿನಂತಿಸ್ತೇವೆ. ಆಮೇಲೆ ನಿಮಗೆ ಈ ಕ್ಯಾಶ್ ಪ್ರೈಸ್ ಅಮೌಂಟ್ನ ನಿಮ್ಮ ಅಕೌಂಟ್ಸ್ಗಾಗಿ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತೀವಿ. ಈಗಾಗಲೇ ತಮ್ಮ ಹತ್ರ ಎಲ್ಲನೂ ಅಕೌಂಟ್ ಡೀಟೇಲ್ಸ್ ಎಲ್ಲ ತೊಗೊಂಡಿದ್ದೀವಿ. ಇವತ್ತು ಈವ್ನಿಂಗ್ ಒಳಗಡೆ ನಿಮ್ಮ ಅಕೌಂಟ್ಗೆ ಹಣ ಬರುತ್ತೆ. ಥ್ಯಾಂಕ್ ಯು.
Thank you, sir, and congratulations to all the winners. Now I request Dr. Anand R, Senior Scientific Officer of KSTI, to propose vote of thanks to all the dignitaries and all of you.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Esteemed dignitaries on dais and my dear participants, it's my privilege to propose vote of thanks on this occasion. I, on behalf of participants of this program and on my personal behalf, I extend a very heartful thanks to our beloved former member and chief patron Prayoga Education Research Center for gracing this occasion in his absence. I thank Dr. P.V. Shiva Prasad, Professor and Dean, National Center for Biological Sciences, Bengaluru, for accepting our invitation to be a part of this inaugural function and for accepting to deliver a lecture. I take this opportunity to thank Dr. C.R. Girija Shivkumar, former Professor of Chemistry, Government Science College, Bengaluru, for gracing this inaugural function and sharing her thought with us. We have among August gathering esteemed guests Dr. Manja Nayak, Dr. Vinay, Dr. Jagdish. I thank each one of you for joining this program. I, on behalf of KST and on behalf of all the participants of this program, I immensely thank Srimati S. Smita for all her support to the KSTA. She is the daughter of Dr. H.K. Shivkumar. I extend my thanks to Dr. A.M. Ramesh sir, CEO KSTA, for his support and encouragement to organize this program. I also thank all my colleagues and all those who are directly or indirectly involved in this conduction of this program. I thank all the participants and all the best for students who are participating in drawing competition. Thank you one and all.
Indeed, thank you very much, sir, for your effort for organizing this program, and also thank all the jury members for selecting the awardees. And we have 10 minutes tea break, and before going to tea break, I request all of you please gather in front of us for group photo. That's what our request, please.
ಕಾಪಿ ಮಾಡಿದ್ರೆ ಡಿಲೀಟ್ ಮಾಡಿ.
ಹೌದಾ ಸ್ವಲ್ಪ ಪೆಟ್ರೋಲ್ ಇಲ್ಲ ಸೋಲಾರ್ ಕೂಡ
ಇರುತ್ತೆ, ತುಂಬ ಸೈಜ್ ಇದೆ, ಅದರದ್ದು ದೊಡ್ಡ ಫೈಲ್ ಅದಕ್ಕೆ.
ಹೌದಾ
ಪರವಾಗಿಲ್ಲ, ಇದು ಆಫ್ ಮಾಡ್ತೀರಿ?
ಹಾಂ ಸರ್ ಯಾವುದು ಸರ್? ಇಲ್ಲ ಇಲ್ಲ, ಇದು ಪ್ರೆಸೆಂಟ್ ನೀವು ತೋರಿಸೋದಕ್ಕೆ.
ಹಾಂ, ಇದು ಸ್ಲೈಡ್ ಶೋ ಫಸ್ಟ್.
ಪ್ರೆಸೆಂಟ್ ಮಾತಾಡ್ಬೋದು.
ಓಕೆ. ಥ್ಯಾಂಕ್ ಯು. ಇದೊಂದು ಲೈಟ್ ಆಫ್ ಮಾಡಿ. Please tell them to come front. This will help. ಅದು ಬೇಡ, ಇದಿದೆ.
ದಯವಿಟ್ಟು ಎಲ್ಲ ಮುಂದೆ ಬನ್ನಿ ಅಂತ ಕೇಳ್ಕೊಳ್ತೀನಿ. ಪ್ಲೀಸ್ ಕಮ್ ಫ್ರಂಟ್. ದಯವಿಟ್ಟು ಮುಂದೆ ಬನ್ನಿ, ಮುಂದೆ ಸೀಟ್ಸ್ ಆಕ್ಯುಪೈ ಮಾಡ್ಕೊಳ್ಳಿ. ಹೇಳಿದ್ದೀನಿ ಸರ್. ಬರುತ್ತಾ ಬರೋದು ಅಲ್ಲಿ ಒಂದ್ಸರಿ ಇನ್ಫಾರ್ಮ್ ಮಾಡು ಎಲ್ಲರಿಗೂ, ಒಳಗಡೆಗೆ ಬರಲಿ. So welcome back. So we have with us today, as you already know, Dr. Shiv Prasad sir a renowned scientist at NCBS, to give a lecture on this occasion. So once again, welcome you, sir, and please, over to you, sir.
Good afternoon, everyone. Thank you for the kind introduction. Maybe those who are in the back can come front if you want to see some details in these slides. Okay? So, those who are in the back, can you see the slides? Yes, very good. You can all hear me? Okay, very good. So, I'm going to talk about this topic, but it will be broadly plant perspective. What is plant's perspective? So, I'm going to say it, and I'm going to say why am I saying plant's perspective. So, I'm at NCBS, which is under Tata Institute of Fundamental Research, one of the leading and oldest institutions for science, just like IIC, and TFR has contributed a lot to the country. We are maybe the first supercomputer, or for the nuclear energy, and so many other things. So, my lab in NCBS is now running 11th year. So, I used to work on Arabidopsis. Arabidopsis is a nice model system for plant biologists, but what we have realized over the years is that whatever people find in Arabidopsis cannot be applied to crops, because it is a very, very simple plant. It lives for a month and then it's done. But these are our crops, so we decided to work on all these. So now I said plant perspective. What is plant perspective? So look at this picture. Here, this guy is thinking, oh, he is stuck in an island. He is thinking there is now a boat coming. He is very happy about it. This guy, he is suddenly thinking there is a land. So these are the perspective from two of those people. Who is going to think about plant perspective? It's already lost the coconut. Maybe it will die when two people are involved. So what I'm trying to say is most of the time we just don't think about other organisms except mammals. Maybe most of the time it's we, just human being, nothing else. So this slide is mostly for the students. So if you convert Earth's history into 24 hours, so we are sort of like newborns. We are here less than 10 seconds, whereas plants are here at least for 6 hours. That will be several millions of years. And the first life is really even longer. So most of the bacteria, fungi, and plants, they evolved much before. They know how to deal with different things. All the climate change we are worried about, plants have seen it all. They had higher temperature, lower temperature, all kinds of dramas they have gone through. Only we are really not the ones who have the ability to cope with it. Even dinosaurs who could rule the world, they lived only for 1 hour of Earth's time. So it's important to see what is the perspective of other organisms. So it becomes very important. Here plants are, along with fungi, some of the most neglected organisms, and there is what is called as plant blindness. It means even if it is there, you are not going to see it. So you're all in the agriculture university. Most of the time, I don't know how many of you will be really looking at plants. You will be looking at humans or animals or something else, or the sky for the plants. So two researchers in the University of Missouri, they got fed up. They thought, why people cannot recognize plants, and they went to the doctors, and doctors said this is a medical problem, because when we evolved, we were only worried about animals that can come and kill us, so we lost the ability to recognize something that is not moving. So they gave this picture to see how many people have this problem. Gave this picture to 1,000 people, 999 people said, what do you see? They said they can see lion. One person said there is lion and acacia tree. So the percentage is 1 in 1,000. Now the problem for fungi is even worse. Only 1 in 10,000 can recognize a fungi. So the problem is so acute. If tomorrow, just like COVID, we get a disease made from a fungi, I think we don't have 10 people in the country who can deal with it. Not even 10, maybe 5. Okay? So that's why we should always think not beyond human, what else is there, and think about their perspective as well. So this is to bring this awakening. Somebody has to be taught or given examples when they are 3 to 9 years. So if you are any of your teachers, parents, you should give your kids or students examples of plants, fungi, and bacteria way early, so they are aware that other organisms exist. And without those organisms, we are not going to live even for a single day, and this is a ultimate truth. Now, why is that we don't really recognize plants? So I have just put some more like a fun facts for those who are not really into plants, so that you will get interested. Those who are really interested in plants, just to make you feel good about plants or yourself. So we think they can't move. Can they really not move? Sanjay Sane's lab, who is a colleague in NCBS, they showed that bladderwort has the fastest reversible reaction, way faster than how our neurons can work. And this is a tiny algae. It uses this pouch to catch and release the prey. So what about this? This is called tumbleweed. I know almost all of you are really fond of YouTube. If you go to YouTube and put tumbleweed, you will see tumbleweed can roll faster than the moving car, and there is no fuel in it. So the inputs we got from this helped to make rover, which could move in Mars without anything. So it can, any obstacle, it will just move, right? So this can move. There are many plants which can move way faster than animals can do. So what about, do our plants have sense? This is one of our biggest proud thing as human beings. We have all these panchendriya, 5 different senses. We can sense everything. We are very proud about them. Most of the time, we just don't care how much of this is really faulty. So for our brain to know that our tummy is full, it takes 20 minutes. By the time you would have heaped so much food, there will be a big bulge later, like sort of me. So, but plants are much more careful because they have been lived for such a long time. Even the simple Arabidopsis plants has 54 different sensors than our simple 5. Okay, so they can sense light. Even in the middle of the night, if somebody is going to switch on the torch, they know. And what kind of light, what frequency, what to do with it, how long it should be, or what to do beyond. So they have the chemosensory, touch, literally every sense. It is really much more amplified in plants than us, and most of these responses are way quicker than most of the animals. So usually we always think about our brain too much, because when we evolved, we put our money on 2 things. One on locomotion, second on consciousness, our brain. We think that plants and other organisms don't have a brain, so we don't have to really think about them. Let's see what Darwin had to say. Darwin had answers for a lot of things. Darwin thinks that the region which has shoots and roots joined, the tiny little bulbous region, is as good as the brain of a baby or a lower animal. And you know, dog has a brain equivalent of 7-year-old, and crow has a brain equivalent of 7-year-old. Plant's brain is as good as that, okay? So this is just to illustrate that one simple point. Glutamate is an amino acid we use in our brain to transfer signals. You put the same glutamate in Arabidopsis and have a sensor which will light up as blue, here green. In 2 minutes, it moved the whole plant. It knows that you have added the same glutamate, which we use for our own sensing. There are more fun facts emerging. All this is not just myth, not just make you all feel something. These are all the most recent research that I am highlighting. So Apple and Cockcroft, in a very famous paper, which is now close to 10 years, they showed that insects in the background, when they chew, any plant knows which insect is chewing and how far it is, how much it is chewing. It can separate out the background from, let's say, a leaf of a song. You are doing something or some machine noise. It knows exactly which insect and what to do for each insect, even though the insect is not actually chewing its own body. So it can sense, it makes noise. The noise can be heard by dogs, but not us. The most recent, this year's research shows that plants can really sense all this. They make noise, they can communicate to each other, even really further away. Now, most of the time, our sense is also this sense, okay? One is sense. Second is, do they have sense? We see sense in different ways. So plants have, almost look at this tomato, the seedling, its stem is filled with those hairs. Each of those hairs are like your electrical pole, which will amplify signal in few seconds. I'm sorry you can't see these numbers. In few seconds, here the GFP lights up, it spreads around if you touch. Even if there is a strong wind, it's going to make the same signal. the same calcium flux is involved in our signaling as well. So there is a transcription factor which is a very important set of protein which can go and do many, many things. Which gets activated, it can go and do. This is a paper in one of the famous journals called Cell. It came only 2 years ago. It shows that plants have this extraordinary network, just touch or sometimes blow, any of these they can go and sense and do. They know exactly what to do for each of those cues. So on the other hand, other type of sense, we don't have sense when we are in the trains or buses. We just want to do this. Look at the trees. If you watch those trees which are all grown same time, they have canopy shyness, which is nothing but giving the sensible gap for the other person. These gaps are usually 1 feet long, and in the edge of those branches, you will have a different leaf, different set of branching. Even the molecular mechanism of this we don't know yet. Okay, but plants can follow this. So just like our WWW, plants also have wood wide web. That means they use harness fungi, mostly mycorrhiza, to have a network throughout their region, not just one tree, from one tree to far away. And in a, let's say in a forest, if there is one tree has a phosphate deficiency, all trees know there is a phosphate deficiency because they will upregulate the proteins to now pick up more phosphate from the soil. So that's how sensible and what kind of brain they have. Now, plant biology usually is, I'm a big champion of plant biology. I think it really deserves much more, and so usually I make fun. Everybody says there is no, plant biology is not a hot topic. Let's see in a very little funny sense. Plants, unlike us, when we get fever, our temperature rises 1 degree, 2 degree, we are in ICU, right? So look at some of these plants. I'm sure you have seen all these plants. They have sterile male florets. The temperature in these florets goes to 50 degree centigrade, and in the night it acts like a big show for insects to come and find warmth for the night or to start doing pollination. So all these plants are completely fine. That's parts of their life. And many times they use in, let's say in a cold place, they can warm the neighboring region so that their constituents, that's DNA, RNA and protein, they are all remain intact. So the research on this really helped us because when people found out what exactly is happening in this plant, they found that in the mitochondria, you have to make this, you all remember mitochondria, right? Powerhouse, it will have chain. So there are enzymes which can shift this chain, and these enzymes are called alternate oxidase and uncoupling enzyme. So if you have it, the whole chain reaction stops, it shunts and suddenly makes a lot of heat. That's exactly what plants do. Now many of our pathogens have the same problem. Mammals have lost, humans don't have this ability to make the quick heat. We get angry, but we are not really becoming hot. But this enzyme, because it is absent in humans, it is the best candidate to tackle diseases like trypanosoma, which uses only alternate oxidase for its respiration. So sometimes I mentioned about basic science. So here the research on some other organism, completely different aspect, gave the best vaccine for us against all those organisms which have only this enzyme. Okay, in spite of all these, how many of you can remember or name 10 different grasses here? You all know 10 different dogs, 10 different cats or whatever, 10 different forms. How many of you know 10 different grasses? Very few or nobody, or how many sedges? So, but plants are really everyday part of your life, whether you want it or not, or whether you want to know it or not. So this is one such example. Okay, so which is our world's second longest man-made structure? Which is the first one? You are all hungry. Which is the longest man-made structure in the world? Chinese Great Wall, correct? So the second longest is in India. It is actually only one tiny piece is remaining. It is called Inland Customs Line. Okay, so this is where it is. It is roughly 4,000 kilometers long. Only thing is present day it is not there. So this was conceptual by this gentleman. Have you seen the picture of this person before? Anybody? If I say the name, maybe you will say it is Yeo Hume. Do you know what, where do you associate him? Exactly. He is the started Congress party, but his major role was he was the revenue inspector of the British East India, and he wanted to stop the salt from here moving to the populous region. So he built this live wall, which is the second longest wall. Though anybody from Mathura, if anybody is here, you can see a small fragment near Mathura, which is now only left 50 meters. So why am I presenting this? When they made this live whatever wall, they brought plants such as this in different parts, and now this plant is spread throughout the country. It hardly needs water, it can grow in salt, and it's called Cassia caranda. It is also called caranda. Have you seen this plant? So you're all eating this even though you may not know it, because what you think is cherry in your ice cream is actually the pickled version of undried fruit, which is actually a weed. Nobody cares, but that's what you think. You think it is coming from UK or Europe, but it is not. It's from here. Many of the essence from these are in your daily food. So I don't know how many of the habit to look into those E505 kind of names in your food packets. So what are those? Do you know? So many of those. Where are they coming from? So many a times nobody want to say it came from plant or fungi or some other source. So basically you are consuming all these. So whether we want to know plants or not, we just depend on them, continue to depend on them since our evolution for food, clothing, medicine, shelter. Even during COVID, first thing we did was to look for the library of 1.5 lakh compounds. Look for the library of 1.5 lakh compounds, which people have documented from plants to see whether any one of them can stop replication of CO2. So that's how much we depend on them, because every medicine has almost a plant origin. So they used to take from them, now we make in the company because it is more economical. So our urgent need is, we have messed up the climate, that's mostly our own doing. We want now plants to not bother with all the change in temperature or water scarcity and still be able to make a lot of yield for us in a sustainable manner. Most of the time we ourselves don't know what is meaning of sustainability. We want 3 cars in the house, we want to change dress every half an hour, whatever. But all that burden we want the plant to take up, and our idea is we should just have more yield and more stress tolerance in crop. Here incremental knowledge is almost not at all possible, so you have to think out of the box approaches. So first we have to think why plants make all this. So can somebody here say why plants make all this? Do they need this much for them? For whatever they make, even the carbohydrate they are making, it is several thousand-fold than what they need. Why do plants make all this? You will laugh when I give the answer, but what I'm going to tell you is the closest to the truth as we know it. Why do you think plants make all this? So plants make all this to have a nice collaboration with animals. Okay? I know you will smile. Many of you are smiling. But that's what we know, because if you simply look at the way how plants have evolved, 100 million years ago when we had the Jurassic time, when we had the dinosaurs, poor dinosaurs, they didn't look at flowers nor they enjoyed fruits because flowering plants were not there. They evolved because plants started making flowers to interact with animals for better pollination. They started filling all the nice things in their seeds and fruits so that they can get better dissemination. And in no time, in the Earth's history, 100 million years is nothing, right? And they have now conquered the Earth, any place, including here, 99% of the plant biomass will be flowering plants. So they really conquered. Even Darwin, who knew answer for everything, thinks it is abominable mystery. That means don't know how they came. Now, why this is important? This collaboration really helped animals too, because animals, until then, they are coming and going like people coming to the skit and running away, right? You have one role, just go. But once there was a food security for animals, it helped almost every animal form to evolve and ultimately leading to the evolution of us, right? So this is what happened. So for millions of years, this collaboration really worked well. But there is one catch here. So I mentioned animals have put money on two things. One, locomotion. Second, thinking, too much thinking, right? So because we are capable of those movement, animals could come, just eat whatever plants have produced and run away, and that means plants have now come with a way to stop all these nonsense animals will come about or do to them. So they were already the best chemists world has seen. They can break water and make oxygen, which even in the lab condition we don't know how to do properly or in an economical way. They could make very complex molecules from mere sunlight. In sunlight, all we know is to hold an umbrella. We don't know what to do with it. But they can make all this. That means they can make millions of compounds. Okay? So, and because they also don't have an excretion system, they made evolved enzymes that can convert one to another. They ended up making millions and millions of compounds. Sometimes one plant can have few thousand individual molecules. And this is how they started interacting, sort of fighting back with the animals. Of course, they provided a lot of carbohydrate and protein because this is the bride, so that the animal will do the bidding. But nicotine. So anybody smoking here or chewing palm, you don't have to feel shy. Nobody, right? So if those who use nicotine, they are going to destroy their collagen, which is part of the muscle. Not for us. Every animal that consumes nicotine will have a weak muscle. That's why Usain Bolt or any sportsman, have you ever seen them smoking for advertisement? No, because they are gone if they do it. And that's exactly what happens to most of the animals. They can't keep chewing this plant. What about caffeine? We just had a bit of it. It stops hunger, not just in us. That's why you will be able to tolerate my talk without getting too much hungry, because you just add some caffeine. And that's exactly what caffeine does for most of the animals. What about scopolamine? Scopolamine in low doses is medicine for Parkinson. Slightly higher dose, it causes madness. This is Datura plant, and this is true for most animals too. I don't have to say much about cyanides. Even low dose can stop life cycle as not just us, many, many animals. What about flavonols? These are interesting compounds. This is, what is this? Gooseberry. How does it taste? Bitter. After that, whatever you eat, sweet. There are also flavonols which are actually very sweet while eating. After that, everything is bitter. So they alter our taste buds, not just us, of other animals, so that you don't go and eat and consume everything that is possible. Photosensitizers, some of the plants have more toxins than anything we can prepare in the lab. These are anthocyanins and flavonoids, part of the bribe in flowers and fruits. And I put these plants. I don't know how many of you can recognize them. These plants have psychoactive substances. Roughly 10% of all plants have psychoactive substances. Now, why plants make them? Anybody? They don't have much nutrition to offer, but why is that plants make them? There is hardly any difference. Most of the time they are okay. So what we know is that this is cannabis. There are two species of cannabis. One grows in Mississippi, where there is too much water. Plant grows very nicely, and it has very low THC, which is the active compound. The one grown in Afghanistan, there is less water, you all know, has very high level of THC. Again, you know from the newspapers. And what people notice is that any goat that eats the cannabis plant in Afghanistan is not going to find the same plant ever again. It has no idea. So this is why, because animals put all their energy in the consciousness, we all want to improve our consciousness. See the tablets people eat to improve their memory and all that. Even our kids, if you watch, kids will rotate to get visual hallucination, right? They are very happy. They will be giggling after rotating, but adults cannot. We will fall down and we will have chakkar. But why are we doing it? Because we think we have to improve our consciousness. We have to improve we like that hallucination feeling. That's what plants offer. That's why most of our thinkers, all the writers, famous people, They were all under the influence of these substances. Now, no, this is not curcumin. This is the active substance in cannabis, opium. Cannabinol. Okay, so this sort of was a nice balance because plants could fight back, really need whatever animals need to do, they can only do that much. Helped animals because animals had nutrition. Earth was fine until some plants targeted us. We are the ones who have all kinds of nice, too much thoughts. And these are plants, curiously, they don't have colorful flowers, but they have carbohydrate. This is very important because protein source will go bad in a week, whereas carbohydrate you can store for years or thousands of years. There are rice grains 2,000-year-old which can be still consumed. You can still get some ATP out of it. So this gave the ultimate sort of food security for animals, especially humans, and humans could not resist. We started clearing the whole world just to pamper these plants, and these are all cereals. And here rice is probably the most abundant macroorganism on earth. Sometimes it is wheat some years, sometimes it is rice, because each plant is one seed coming from one single seed. Now in this co-evolution here is both have evolved nicely, shared each whatever resources. There is more grain for us, but actual dominance goes to plants. And there is also pampering for plants. So what I mean by saying pampering? Have anybody seen cauliflower or cabbage fields anywhere? Do you see people, do you see those fields without people? There's always somebody there, just to do something or the other. So this is called, ultimately this is called domestication syndrome, where even if you provide everything, nutrition, water, plants still won't be able to produce much. That's why you see many times our greenhouses are way better than our own dwelling. They have light control, they have water, they have moisture, they have this. So basically this is called pampering. This is the last stage of domestication. Okay, so now rice is the most important plant for us as well. Most of Southeast Asia, most of Asia. And how did rice evolve to this nice form where there is nice stem, lot of grain which sticks in the plant, literally needs much less water. We still complain saying it is 3,000 liters of water to make 1 kg of rice. But its ancestor plant is a semi-aquatic weed which is sometimes floating on the edge of lake. From this, in 10,000 years, this is how plant changed. Okay? So these are called wild relatives or wild species. These are called cultivated species. So indica rice that we all eat comes from Oryza nivara. This is like, for those of you who are not at all getting connected to plants, think that this is like monkey and what we eat is like human. Okay? So this is our ancestor rice, and Rufipogon is ancestor for indica and japonica and javanica rices. These plants, as I mentioned, can still be found in edge of lakes, especially in the coast either side of the peninsula. So what changed during this domestication of rice? So this is one of the problems we picked in our lab, and the reason was we first wanted to see what changed everybody wanted to see what has changed, so we went back to sequencing the genome. They took the wild species, cultivated species. Surprise, they don't see much change in ATGC. That is the digits which make up our code, right? Our recipe book of every organism. So just to give one example, I don't know how many of you have heard the term QTL. Anybody didn't hear QTL before? So when breeders are working, they want to associate one trait, like big grain, to many regions in the genome. Genome is full of DNA of different type, and one region contributes to, let's say, big grain. So they call that quantitative trait loci. These were identified for decades, and we know from many research experiments, 200 rice wheel related QTLs are known. But we know identity of only 8 genes. So I'm sure you are all heard about genes. I'll give some examples. Only 8 genes. That means what else is there? So we thought there is role of what we call it as small RNA and epigenetics. You don't have to get frightened with this term at all. I'm going to say Very simple words what it means. Okay, so it all starts here. So DNA is your book of recipe. Each of us have, each cell has, each living organism has. And in a book of recipe, you copy only some of them in a given time. You make some of them like rice every single day, some of them when guests come, some of them really once in a real blue moon. But this copied recipe, when you convert into a dish, that is your protein. So you have make DNA, DNA makes RNA, RNA makes protein. And I said only few of the dishes are made all the time. What happens to the others? Sometimes they are not even opened, the recipe is closed, or sometimes the copied recipe is just destroyed. You don't really go and make, you are fed up in the middle itself. So how this happens? This is done by small RNAs. These are set of molecules, bits of RNA. They can go and degrade, cut these copies, or they can do what is called as epigenetics. So what is epigenetics is, it can go and modify DNA or histones around which DNA is coiled around. So people got Nobel Prize for doing one really small experiment, and they got Nobel Prize within 4 years of doing it, so you can see how important it is. Most of the time people wait for Nobel Prize till forever. You can see everybody will be white, right, when they get Nobel, but these people, they still have black hair. They got in no time a Nobel Prize to show you how important this field is. Now, the pathway looks little complicated, but I don't know how many of you have already seen something similar. So, in a genome, you make RNA. RNA will be cut into small RNA bits, and that's done by a protein very nicely named. It's called dicer. Somebody who cuts, dice. In fact, the graduate student who coined this term, graduate in the sense PhD student, she was cooking, she was cutting carrot. She thought the protein is doing the same, why not call it a dicer? So the name stuck. So it's called dicer protein, which makes small RNA. The small RNA will have two strands. One gets degraded in a protein called argonaute. Now this one has all these GCAT, right? This can find complementarity in a cell with anything that has Watson-Crick complementarity. What is Watson-Crick complementarity? G should pair with G should pair with C. Very good. So that's Watson-Crick complementarity. If there is a complementarity in this 21-bit small RNA, it can go and cut the RNA into two. Okay? So this cutting is so precise, the target is cut between position 10 and 11 of the complementary region. And this is how most of the small RNA looks. You all must be wondering, what is the use of this? You just make RNA, you chop it, it's like a negative thing, everything. But this is a very positive thing for every organism that we know of, because without this, you end up copying different recipe, making different dish, and the organism is gone. So why we work with plants is, so some of these organelles can go and talk to the DNA. They can make this modification in DNA called DNA methylation, which is a small chemical modification, or it can go and make modification in the histone protein around which DNA is coiled, and together this is called epigenetics. Now what is special in plants is that, or why we work with plants is that, once you change this, the whole property, the recipe book is completely changed without changing the DNA sequence, and this can go to the next generation. Unlike humans and other animals, our germ cells are decided before, so whatever we go through, our kids are not going to have the same thing, right? Let's say we lose some organ, it's not like our kid will born without that organ, it's not the same case. But in plants, every cell maintains this and passes this information to the next generation, just like what Lamarck suggested, but Lamarck used the wrong model system. What did he use? Giraffe. He should have used a plant, right? If he uses a plant, whatever he thought, which you must have really loved when you studied, is exactly correct if he uses plants or certain species of yeast. So this here you need additional polymerases and really a machinery which can memorize this and pass exactly what goes into the next generation. Is very important if you think why, even being in sessile plants which don't move much, they are still able to manage everything. They manage 6 hours of Earth's lifetime because they pass this information really nicely to the next generation. If a plant went through, let's say, dehydration, less water, its offspring, the seed, already knows what to do under dry condition. So unlike us, so whatever we go through, it's not going to be possible because our germ cells are designed, decided even before we are born. It's decided when we are in our mother's womb. So this is why we work with plants, and we thought maybe such a mechanism might be responsible for how rice got devolved or developed. In the last 10 years, this is what we have done. We have identified how small RNAs are made, what do they act on, how do they go and change all this recipe book, and how do they keep this memory in the next generation and subsequent generation, and related work on how plants can cope with stress. So I mentioned about epigenetics. Here is a bit more detail. Those who are really interested in DNA, this is cytosine, one of the ATGC. You get one chemical modification, that is methylation, is already going to make methyl cytosine. Now this is a methylated DNA. One cytosine in right place can now make the recipe closed or make one single cytosine. You all know how many digits are there in the genome. So one of them can have that much effect. Whereas in the histones, these are like octamers, 8 bits of protein. They will have these tails, that is, the protein is a long chain, one chain is protruding out, that chain is N-terminal end, and this can again have chemical modification. Depending on this modification, the DNA will be either red or not, so that is what we call it as epigenetic change. Now, to study if rice went through such a change, we took 400 different land races. I don't know how many of you know what land race is. These are the land races which seniors, many of us, were eating until 1970. What we now eat is called cultivated, high-yielding green revolution lines. The ancestral species are 2 wild species, Oryza nivara and Oryza rubicoga. We collected all these, did the phenotyping, did the analysis to find what has changed in DNA methylation level, histone modification level, which kind of recipes are read, that's through RNA sequencing, what kind of genetic changes changed through DNA sequencing. So I'm going to give you one example of what we found here. And the work was done by Shweta Chenna, who is doing a postdoc now in University of Copenhagen. What she found is that this small RNA typically should be of size 21 and 24. So this is a simple graph. You have a lot of abundance of 24 and 21, right? Take any plant, you will get this kind of ratio. Lot of 21 and lot of 24, nothing else. But in wild species of rice, called Oryza nivara, you got much more of 22 nucleotide, which was a big surprise to us. So if you, in the graph, you can see more in nivara, little less in rubipogan. These are the 2 ancestral species. Landrace is intermediate, almost nothing, very low in cultivated species. So rice has 12 chromosomes. Chromosomes are regions where a lot of genes are all packed into different numbers in different organisms. Rice has 12, and when we try to find where is the source of this small RNA, we picked one region in chromosome 2. Here, reads of 140 million reads. So here the reads are next generation sequencing reads. The number tells how abundant they are. This runs in millions. So this is a new technique which has transformed all kinds of biology. And you can see in wild species very high, landrace intermediate level, and cultivated rice lines that we all eat, nothing. There is no such small RNA. So between certain markers, we found a new microRNA. This is one type of small RNA which can go and cut the target into 2 bits. If you run a northern analysis, which is simply take all the RNA, your copied recipe, check what is present. This density tells how much is abundant. This microRNA very high abundant in nivara. That's what we see with this analysis. Intermediate in land races. I don't know how many of you can recognize some of the land races which you might have heard or eaten when you're small. And white ponya and pusa basmati, which are the high yielding cultivated lines. This small RNA is completely lost. Okay, so just to give a recap, wild species gave rise to land race. After 70, we have the high yielder lice, and there is a gradual change in the small RNA. Now the question is, what are the target of this small RNA? Now the targets of small RNA should be having that Watson-Crick complementarity, very high for this 21, 22 nucleotide small RNA. It turned out to be enzymes called lactases. Lactases come in a pathway called phenylpropanide pathway, very important pathway for all organisms, especially plants and fungi. So here monomers of alcohol are converted into polymer called lignin. Lignin is the second most abundant organic polymer on earth after, which is the most abundant? Cellular, very good. So you need these enzymes to make lignin, and lignin provides mechanical strength. That's how plants conquered from aquatic system, they went to terrestrial system. So most of these are targeted by the small RNA. That means there are 24 different enzymes, different such genes in rice. Nobody had studied any of these. We found almost everything is chopped, cut into bits in wild species of rice. We used many different techniques. All those details you don't have to know. But believe me, most of the time in wild species, the result is that there is hardly any expression of these enzymes. Out of 24, 3 barely expressed. So expression here means how much of copies of RNA is present. This can be assayed by a technique called RNA sequencing. Here all the bits means how much is present. Cultivated species, very high abundance. Land races, intermediate. Wild species, almost nothing. So this enzyme is completely lost. And because of this, now you have less of enzyme, you get less lignin accumulation in the wild species compared to cultivated lines which have very high accumulation. So if you make stem sections in the plant, so rice has a stem, I don't know whether you thought only leaf is there is also a stem. If you stain for through fluoroglycin or for lignin, low lignin here, high lignin in the cultivated lines. So these proteins are glycoproteins, one category of proteins. So we used a column called concanoline to purify this protein, check how much they are active, which one is active, how they are active. All we found is that cultivated lines have lot of lactase to make lot of lignin, whereas wild species didn't have any. So this is what we think. Is this a key domestication event? Wild species of rice is a semi-aquatic weed, hardly any grain, and here microRNA is really abundant. Targets are all chopped, lactases are not there. The result is you get low lignin, plants are prostrate, like python-like, they are not really standing up, and they have closed panicles and seed shattering. All these are connected to low lignin accumulation. During domestication, this is the magic, we have lost the microRNA. Now enzyme is accumulating much more, so plants have high lignin to support panicle. Sometimes we measured half kg grains in single plant. In the wind it will move, but it is not going to fall unless you have rain or really strong wind. So plants have really good now developed this ability to hold much more grain. Now what has changed in this? Now we have identified a transcription factor which is now differently activated in the cultivated lines compared to wild species. So how to check this? This is still a correlation. See, most of the research is just a correlation and of no use unless you test and find what happens. So one of those indigenous technology, what we are talking about, should be the technology to really nail and say this is how things happen, which most of the time, which is a drawback of our scientific methods, especially when either poor resources or not really able to go far. This is what happens. But it's not good research or it's not research. Correlation is something our forefathers have established. Farming has been established without modern scientific methods. So it's all correlation. So you should always separate out correlation from active research. So here it is still correlation, no matter what we have done. How do we prove this? To prove this, we brought this high expressing microRNA here. Now what do we expect? Anybody can say? If you bring the high expressing microRNA into cultivated line, what is that we should be expecting? Or how is that we can prove that this microRNA is really responsible for all the yield related changes? So the plant should be like this because it is now not having any more mechanical strength. That's exactly what we got. We made plants which make a lot of this small RNA. Plant is just falling off, no mechanical strength. and it made total of 0 seeds, no yield at all. And the plants don't have much lignin compared to the control. But now if you overexpress this enzyme, if you make the enzyme more in the plant, plant grows much better and plant makes much more yield. So we thought, how come people missed such an important finding? Because there are, I don't know, thousands and thousands of rice researchers all over the world. So we went back to those 200 QTLs that I mentioned. So only 8 genes were known. Now we mapped all these microRNA and the lacases back to those unknown QTL, and we got in one go 26 different QTL mapping to these enzymes. So which is 1/8 of all the unknown QTL, 1/8 of the reason for rice yield in one study. Okay? So if you look at some of those QTLs, some of them have only lacase. One of these QTL was identified in GKVK. India made only 2 out of 200, just 2 from India. So these were the known genes. This was already known. This is what we added in one study. So we obviously got a lot of good comments from people, and our paper for 2.5 months, the top rated in Plant Cell, which is our premier journal. Not many, 8, maybe there are 8 papers on this journal from our country. Okay, so I'll just take few more minutes. I have probably 10 minutes left. I'll finish in 10 minutes. So we were also interested in how this color is formed, which I already dealt with in one of the slides, right? So we have this small RNA data from around 120 plants. We are simply comparing which plant makes what, how much. And what we found is that grape makes very high abundant small RNA of specific type. We target a gene called MIP, and this was very abundant in reproductive tissues, that is flower and fruit. So we thought, why not study in grape, because nobody established grape as a system other than in breeding. So we took Bangalore blue. So those Bangaloreans, you don't have to feel too proud. This is a Iranian variety. We just named Bangalore blue. So it has not developed in Bangalore. So this is called Dilkush. Good for heart supposed to be, because it makes lot of flavonols. Okay, so this makes anthocyanin. This is Dilkush, again not really Indian, it came from Persia. And Red Globe, thankfully it is a modern variety which makes also high anthocyanin. So we wanted to check what changed this coloration. This coloration is really, no matter what, very important, because we say it is antioxidant, anti-whatever. Every anti seems like to be working, but please be aware, if you take in this syrup or in tablets, which are sometimes 100 rupees per tablet, it's of no use. Your grandmother was right, you should be chewing and eating, otherwise you are going to burst the vacuole and these molecules are not going to be absorbed by you. Okay? So all this high added anthocyanin, added flavonol is all just rubbish. Okay? So you have to eat to get this. So what is the use for us? So we think they are useful because we eat fruits not for sugar. Sugar, you could just take some sugarcane juice. We use for all these molecules, which are the ones which help us to recover, drive our growth, stop injuries, or recover from wounding, etc., etc. So, but for plants, if they are in the flower, they are going to help with pollination. If they are in the fruit, that is going to help with fruit dispersal. So, these also decorate in flowers. Can somebody say what this flower is? There is no, not this one. There was an article only last week in Indian Express. So this is called Semper Augustus. This is a tulip which created what we call it as tulipomania. Dutch really became crazy and they really sold the whole land just for a few seeds of this tulip. And this is, so basically humans and all animals are fond of clover. I don't know, I am sure there are many men here. Men are equally fond of flowers like women. And do you know why? Why men are fond of flowers? So it's in our genes. When we evolved, we were hunter-gatherers. That means if we know there is a flower, it is a promise for food. So you'll go back to the same place where there was flowering to pick up the fruits. Even today, San people in South Africa, they know, even if it is 20 kilometers away, which plant flowered when. so they know when to collect the fruit. So that is in our blood. That's why men also are interested. Don't take it wrongly. I'm just saying what I read. Okay. So we wanted to find how this color forms. Without giving much molecular details, what we find is that these need one specific small RNase to chop a protein called MIB repressor. This is a new category of gene that we identified. It's called 114 class. If this is gone, you get color. If you allow this to make the recipe and make the protein, you get flavonol. Seems like all this is, most of the fruits when they evolved, they made only flavonol. Just because we and other animals, even birds like the colored fruits, most mammals like colored fruits, so even small kids. So we had a project for high school kids. We kept 20 different fruits. Only 2 were colored. And everybody picked only the colored fruit. Nobody wanted the colorless fruit. You all know the race to find the cabbage should be colored, or even anything should be colored. That's in our sort of brain. We want more colored things, except in our skin. Everywhere we want high color. And that's what we have done. So we have again domesticated in such a way that we like color. And this seemed to be the same mechanism, because when Varsha's thesis was last year, we checked the literature. After our paper came, we see 25 papers across different crops, from bengan, from potato, from strawberry, apple, everywhere the same molecule is giving rise to color. So the use of this is, we could make hell lot of flavonols, which are again very important medicinally, way more than anybody has ever done. In plants like tobacco, we can make a factory of flavonol, which we are trying to patent. Okay, so that's what we find across many of our studies. You need small RNA or epigenetics to make useful traits of any different type. And because I work in NCBS, till recently I was the only plant person, everybody will ask, how does this work in animals, right? Because everybody is an animal researcher. So in animals too, this seems to be the same reason. You have to get rid of myostatin through a small RNA to make what is called as double muscle phenotype. For you all know how the wild chicken to the cultivated chicken looks. They are all double muscled, and this is what happens when people are really pushing the iron and becoming bodybuilders. So all you need to do is get rid of myostatin. The research came from Yale. They made the patent, but somehow the information leaked out. Now we know that somehow if you reduce myostatin, you are going to get double muscle phenotype. So next time when you watch a bodybuilder, you should think whether he has taken myostatin drug or really worked out to get the bus. Okay. So I think my time is almost up. So I'm not going to the details. I'm going to say 2 things in the end and stop. Okay. So just like the mobile phones, we are in the 4G stage. This is for breeding. So this is what happened in the initial days. This is the pre-green revolution, which is simply prefer and start making growing. Now 1900 onwards, we started mating the plants, making breeding. So started making hybrid. Literally all what we eat now are hybrids. So we are all hybrids, and all what we eat are hybrid. We are hybrids. I'm not talking about the recent hybridization. I'm talking about an Homo sapiens evolved. We are crosses between Neanderthals and, I think you are all really too hungry. Desonians. Okay, so these are the crosses we have made, and we are all offsprings of this hybrid, and that's what we eat from really long time. And in 1980 onwards, we have the transgenic technology and so many techniques, so we are able to make much more yield. We are so far okay, but most of the food we are making, so let's say from US, it goes to feed the pigs for meat, right? So we really have equal amount of food, but the food is not coming to humans because it is going to feed animals, so we are for biofuel preparation. So we have really less food already, okay? And what is going to happen in the next few years, we are nowhere really sure how to feed the world, because many of these techniques, such as genome editing that we use, but it needs a lot of expertise. So we are really far away for this, what is called as third green revolution, which will be the 4G design breeding stage. So some of the experiments I mentioned can contribute to this change. So all this is possible only if we can mitigate the climate change effects. That again, we are really far. That's why each one of you have the job to really pick up research and go and attack this problem. Now in the country, we have exhausted all the arable land, and actually we have destroyed most of the arable land into nothing. Anybody who has really watched the countryside, you know that the regions which are green ones, they are not green. Regions which could hold a lot of grains, now they don't, except reservoir-driven rice cultivation. It's only coming from reservoirs. So already we have sort of a food security issue, which is not coming in the newspaper, but you all know we don't really produce all the pulses that we need. It comes from Canada and Australia. Sometimes every day you are eating pulses which are made from other country. So by 2050, even if we don't diverge and take food for, let's say, bioethanol, we need twice the amount of what we are producing, and we can do many things. We can crop diversify. That means you start eating which are nutritive food, but we are neglecting them. Improvement of all these needs political will. We definitely need to develop climate resilient crops, which some labs like ours are doing. But these are also need a lot of expertise, but what everybody can do is this. So this is the food you are all eating. It needs 4,500 liters of water to make, okay? And nutritionally, it's one of the poorest food you can think of. You can move to this. This needs only 400 liters of water to make, and some of them I am sure you can recognize. They are local vegetables. Sometimes they have some smell. You have ragi here, which many of you don't like to eat. But I just want to say, in food and in health, literally every human related information, there is 99% of misinformation than the real information, which is because people want to make money, they end up saying all kinds of things, such as here. I just want to highlight this one first. So apple a day keeps doctor away. How many of you believe this? Nobody. This is really amazing audience. Usually everybody will say, yeah, it is what is happening. I think either you are too hungry or you are not listening, one of those. But apple a day keeps doctor away is a 110-year-old lie, which is still being propagated. If you keep on eating apple, you are going to get diabetes, because it was a myth. So till 1850s, 90s, US was growing a lot of apple. They were converted into alcoholic drinks. Then church said prohibition law. No, nobody should drink, or kids should not drink, and so on. What do the poor growers do? They said, let's start a slogan, apple a day keeps doctor away. And in that kind of urge, we have forgotten all our crops. Just go to Google and Wikipedia. Your own poor guava is 20 times more nutritive than apple, 20 times. So I don't have to even say most of the apple you are eating coming from New Zealand from the last year, it was with zero oxygen condition, and it doesn't actually mature at all. You are eating almost a rotten apple. So, but again, yeah, that kind of misinformation is really ripe. You are the best judge. You can do all this in Wikipedia and find out. So the ragi, this is called teff. This is the closest relative of ragi, okay? And this we are now importing from Ethiopia and Kenya to feed our marathon runners. Teff is super good, almost like our ragi, okay? So you all know who wins gold medal in marathon. It's always those very thin people, because they get nutrition from teff. And that's why now we are importing without realizing that many of our own grains have the same capability. So these things, even if you don't want to come to research, or even if you don't want to be part of research, you can still do this and help everyone, okay? With that, I thank my lab. So we call ourselves epipepis, and I don't know why. So I had a lot of cryobiome structures which came from all these friends. So we identified a first histone H4 variant in any organism. So there is no time to discuss all that. And these are my source of inspiration. Weltham is the pioneer biotechnologist in the country. He has trained 643 people. I am one of them. So one of those people who will never come to the limelight but do lot. And that's plenty in our country. And Sir David Balcom was my mentor. He is the discoverer of small RNAs. And I'm in a fantastic place called NCBS, and anybody who want to really do research, real research, with logic, we can join us. And thank you for listening. I'd like to also thank Dr. Ramesh for the kind invitation to share with you all what we are doing. You all should ask questions. I'll tell you, okay, you're all very hungry, but I'll tell one thing. How much knowledge a Nobel laureate has? It's 1% of any given topic. How much knowledge a fool has? It is 0.5% of the knowledge. So we are all in between. So it is somewhere between, 99 point something. So what you know, I won't know. What I know, you need not know. So that should be your idea when you ask question. And I know there is a very poor response now across the board in our country to ask question. You're all thinking yourself, but not ready to ask a question. Because when you ask a question, you will be formulating something. This is exactly what I call logic. You have heard something, you have brought some memory from something else, maybe from newspapers, maybe from TV, maybe from somebody, and that logic is the basis for curiosity-driven research, so you must ask questions. So plant doesn't feel the pain because it doesn't need to feel the pain, because each cell is not having the same nerves like us to do something else. Why we need that brain? Because we have to coordinate a lot of things. We have different organs everywhere. We have to walk, so walking was a big problem. So we need a central organ to do this, and when something happens, that organ need to be told something is wrong. For plant, nothing. Plants, each cell is like a brain. Okay? So what we find is that each cell can make the same decision, like part of our brain. So and it is not at all did not put money on sentiments. It's many times okay to cut a tree, and it will now make what is called as copicing, more shores, and that means more leaves, that means more flowers, that means more seed. So if somebody is looking at reproductive ability as the success, it is going to be happy if you cut it, rather than not. So anybody has grown guava? Nobody. None of you are growing anything. Oh, good. So if you keep the guava plant without cutting, you're going to get 1 or 2 bitter fruits which will never ripe. But you keep cutting, you will activate floral meristem, which will make much more fruits which are now light colored, softer, and have more seeds. So why? So plants are okay with they don't feel pain, unlike what you might hear in the internet. I said most of the time the information is wrong. They don't need to feel a pain because their body design is such that they make local decisions. That means they are not worried what else happened in the other branch. So one branch, if it gets lot of light and nutrients, it's going to go better. It's not going to think, oh, other branch is now poor branch, what to do about it, nothing. So the only connection is when there is a defense attack, it will take local decisions too. So within a plant, if one part is cut, other part is now ready for the same attack from animal, herbivore, or human. So it's done nicely without having the pain component. So that is what Pitil Chaudhary has shown. But what we now know, so I said something in the initial slide. So plants can sense smallest noises and changes. Some of them, they are going to respond. And that response can help with growth. So that's why the people who touch, move the plants, it's going to respond in such a way that it is going to make much more of chemicals in its stem, which will help with better branching. See, you touch the plant, what it is going to think, okay, now there is probably wind, so I have to make now more callows, in the right cells, so I have to be ready for the next thing, so it will activate all the meristems, so it will make better branching, which is ultimately quite helpful for the plant. So the growth or even any, any noise of any kind plants can sense, but they are not really enjoying the music. They are just responding to as a cue. That's how they are designed. MIB1, yeah. Yeah. So what we know is that its expression is high in every fruit that has a color. So finger millet has a related microRNA. In animal monocots it is connected slightly differently, but the same enzymes are there. Even in rice. Rice has also kavuni called red black grain, which is actually strong violet, which looks like Bangalore blue, grown in near Tanjore and south near Trichy area, where it is called, our Chettiars are fond of it. It's a black rice. So same mechanism. Yeah. Yeah. So it's the same nutrition. Only thing is now plant has ability to make both. Only thing is it will neither make tomatoes as much as a tomato plant, nor it will make potato as much as a potato plant. It will make both little smaller, and it's fine. So plant is, whatever the resources it get, it will make a more seed or more tuber or more flower. So it's actually selfless service. So the more nutrition means it can help, but tomato plant especially, it comes from grafting, tomato and potato. So its ability will be reduced, but it will make both. You don't have to do anything for it. Health is, again I mentioned, it's a dicey word. It is a myth that we all read and check. So sometimes, see, now we want to go back to all those food which is rich in fiber. For 30, 40 years, we wanted maximum processed food because everybody said those food is bad, they have anti-nutrient capabilities, you should not be eating that, that is bad. So it is only driven by money. Now people want to go for that because they think there is more money in millets, more money in that, more money in that. So that's completely different aspect. So it is, even by having less inputs, it is possible to get very nutritive food. Only thing is we should have as diverse food as possible. That's why people say you should have 20 different vegetables, 10 different fruits per week and all that, because they are all going to help with our microflora, and our flora is the one which helps us to absorb, helps us to stay healthy, reduce all kinds of damages we do ourselves. So most of them are synthetic. Who is going to have that much plant, no? So all the henna is not now coming from your favorite henna plant. So most of that color which you add in chicken and gobi Manchurian, it is now coming from one insect powder. So you can read all again in Google. So it's not from plant. So for plant, if you want, you need a plant called anato, which is a lipstick plant. I'm sure some of you have heard. That is very hard to grow. How many places you can grow and how much you can extract. You can make the same in lab in no time, but it is highly carcinogenic. So that's what we end up. So best is not to add any color or don't go behind any color. Every food now what we eat has very high levels of all the wrong things which you should not be eating. And anybody who is careful with health, you can see the number of increase in cancer patients in the country. So you just go to site care just for a visit, you will see very young people. You will feel like crying. They are 10-year, 8-year-old. How many thousand you can see? You will think they are gone there for a picnic. No, they are all patients. So just it's very important to know what is Truth is there in the books, in the internet. You can pick the right truth, the real truth, and do accordingly. So most of the food industry, most of what we do are actually injurious to ourselves. Sorry, I might sound too this one for many of the young people, but I'm supposed to say this irrespective of, no? So it is very important not to get the wrong information from anywhere. So most of these is, so if you don't believe, just go to Sidecare. It's in the airport road. So just have a visit just to see how people are. Any other from students? Students don't want to ask anything. So there is a transporter called of a sodium potassium transporter. So what it does is it changes the turgor pressure immediately and stops. So the pressure is now gone. So the water is removed, so now it will bend down. So when it fills, it will now come back to its original pressure. Everything plants and other organisms do, though we think they are all dumb, they are very clever. They are now exposing what is that? All those needles in the touch me not, so that your poor cow or somebody cannot come. See, cows are very clever. Most cows don't try to even eat those. But goats are not that clever. They'll still eat it even if it is with nails. And those anybody reared goat, you will see them having problem. They'll be twisting their tongue after eating for a long, long time. Yes. No such is known. It is unlikely. I think most of the plants' ability is to, no matter what, we will cope with it. Right? So I removed some slides. See, many times we are worried about fire, but plant is completely fine. It will, its own part will be dead and it will become a nutrient and it will germinate as soon as the next rain comes. So most of the calamities have nothing to do with plant because it is not having an organized brain like ours. But it has local brain, it will deal with it, it has enough ways to cope with literally anything. That's why it is there from last 6 hours of Earth's history. And if you give space, or what we are doing is we are removing its space, right? If you give space, I don't think any plant will just get extinct, unlike animals. The plants know how to do it. So now we are doing a lot of cropping in a different way. Most plants, instead of cross-pollination, they are now doing self-pollination. So they are, the androecium is, filament is smaller or coming closer to stigma. So there's a happening in last 10 years in nature, okay? So plants which are not having too many in the neighborhood, they are already becoming self-fertile rather than waiting for the insect because insects are not there. So all this suggests that plants are actually quite capable. Yes.
So in that, how can we identify which RNA is isolated, whether it is an mRNA, rRNA, tRNA?
Very good question. So this is called northern hybridization. Supposed to be like southern hybridization. Southern, as a great scientist in 1975, he used southern to find DNA. How does he find DNA or RNA? You label the other copy, Watson-Crick copy. So this is your normal strand. You make another strand which will go and hybridize. ATGC should pair. And this you label with radioactivity or some chemicals such as digoxygenin, and this will emit light. So you can in a gel find out if, see, this sequence specificity is really nice, right? So really the Crick and Watson deserved not one, many Nobel, because it makes that complementary, right? A pairing with G and T and G pairing with C. So same way if you make a stretch which can complement a given sequence in the cell or in no matter how much of RNA you have thrown or DNA you have thrown, it will go and find the right one which has complemented, not something else. In a stretch of 21, even if there is one base difference, it is not going to hybridize. So it is really pakka zipper kind mechanism. So all you have to do is your probe, what we call it as probe, label with radiation or chemical, put it, and then you can check where the light is coming from or how much light is coming from. With the X-ray sheets or with the different methods, then you will know how much is present and which size, because you are separated in the gel.
So depending upon the size, we get to know which type of RNA is as well.
Yes, because your RNA, you know which one. You have designed an RNA which can make pairing only with one other RNA. That is what you are checking, whether it is there or not. So in the northern I showed, the RNA sequence we know. So we simply make a complementary RNA, label it with something which can emit light. Here it was P32 radioactivity.
Thank you, sir. So is peptide grown in India?
It is being tried in Rajasthan when I last heard, and I don't know whether anybody, it's possible that people will start growing in the greenhouse just like we are fond of every Western vegetable and fruit just for getting money. How many of, huh?
What is so special about it?
It has very high levels of nutrients, so both amount of protein, it has amino acids, mineral content, everything is really optimal for a person who has the right kind of muscles for long distance run.
Amino acids, is it essential amino acids?
Absolutely, but it has the right ratio. So see, the person who is running marathon will not have muscles like me, right? What kind of muscles they have? Thin, striated, this is like, I don't know what kind of, I don't know about humans much. I know more about plants than humans. So these muscles need constant source of energy, but not at bulk at once. They don't store too much, but they release very slowly for really long time. And that kind of nutrition comes from teff and other grains.
Thank you, sir. Sir,
Yeah.
in grafting process, some plants are growing. Some plants are not growing well. Which gene is responsible?
It's a very, very good question, and most up-to-date research came only last week. You need what is called as a PAX gene, okay? If that gene is there, you can fuse any two plants. So, for a long time we knew that only within the species, within the genera, you can graft, it will come as nice, like the pomato he mentioned. But now we know a gene which can make a bridge as long as cambium cells, which is one type of cells which can grow, divide and make connection. If you have a gene called PAX, and this came in Nature Plants last week, and it's my friend who has published, so I know well. So you need many different things to happen. Basically, cambium cells should attach and not have defense response to kill each other, but have a compatibility to establish new cells growing and make the bridge.
As we told, molecular basis of epigenetics, DNA is DNA is red or not?
DNA is?
DNA is red or not, as you told, sir.
Red or not? Yeah, so red.
How to identify any fluorescent dye is used?
So there is, I thought it's time for food, so I didn't say, but I'll take 1 second to answer this, okay? Yes. Okay, so this is the stain. This is DNA which cannot be read. This is called K9 methylation. Such DNA will look like this, okay? You can stain, this is a one nuclei. You can see those regions will never be read. This is the unread recipe. This is such a region which will be like a guest. When the guest comes, that recipe, sometimes it will make, sometimes it will not make. That you look for this mark. This is a small chemical mark in one of the histones. And on that we make antibody, we make probe, we can check. Literally we can walk through the genome, though it is many MB, go through each region to see which one region looks what. And we can also now edit it. We can modify single bases, use CRISPR, many other methods are possible. ಓಕೆ.
So, sir is available during lunch time. If you have any more questions, we can discuss it. Already we are running out of time. And once again, thank you very much, sir, for your interesting lecture. Please give me a big round of applause. Thank you, sir. Thank you very much. And few announcements. We have half an hour lunch break. 2:30, please come back. And those the winners wanted to claim for TA, please go to the reception after dinner. Okay. So please, sorry, lunch. Please yeah, please have lunch and and assemble back at 2:30. And we will.
Those who are participating in painting should come back early.
Yeah, please. Thank you. Please come, sir.
A very good evening to everybody. Respected dignitaries on the dais and all the participants who are sitting off the dais. I am extremely honored, overwhelmed, and ecstatic to be here as a part of valedictory function of KSTA sponsored program today. In the morning, one of my students, Miss Ananya, she was a recipient of Padma Bhushan, Professor UR Innovation Award for his innovative PG research she has conducted under my supervision at the College of Fisheries, Mangalore. One thing I would like to say, being a mentor or advisor, certainly we have to give a lot of opportunity to the students, and we have to motivate the students, the youngsters. They need to come up and they do a lot of innovative work irrespective of their field, and this is how the country needs. And today the KSTA under the chairmanship of Dr. Ramesh MR done today. I am extremely happy to be here as a chief guest. And this is the chief guest remarks. And at the end, I wish all the very best, all the participants who have participated in this program, the great success. Thank you so much.
Thank you, sir. ಈಗ ನಮ್ಮ ಮಾನ್ಯ ಅಧ್ಯಕ್ಷರಾದಂಥ ದಿವಂಗತ ಡಾಕ್ಟರ್ SK ಶಿವಕುಮಾರ್ ಸರ್ ಅವರ ಒಡತಿಯಾದಂತಹ ಡಾಕ್ಟರ್ CR ಗಿರಿಜಾ ಮೇಡಮ್ರವರಿಂದ ಒಂದೆರಡು ನುಡಿ.
So, good evening all of you. I think we have come to the end of this program. ಬೆಳಗ್ಗೆಯಿಂದ ಭಾಳ ಅರ್ಥಪೂರ್ಣವಾಗಿ ನ್ಯಾಷನಲ್ ಸೈನ್ಸ್ ಡೇನ ಆಚರಿಸಿದ್ದೀವಿ. ಬೆಳಗ್ಗೆ ಇನೋವೇಷನ್ ಡಿಸ್ಕವರಿ ಪ್ರೋಗ್ರಾಮ್ದು ಪ್ರೈಸಸ್ ಡಿಸ್ಟ್ರಿಬ್ಯೂಷನ್ ಆಗಿತ್ತು, ಟಾಕ್ ಇತ್ತು, ಆಮೇಲೆ ತುಂಬ ಜನ ಸ್ಟೂಡೆಂಟ್ಸ್ ಎಲ್ಲ ಬಂದಿದ್ದರು. ನೀವೆಲ್ಲ ಪೇಂಟಿಂಗ್ ಕಾಂಪಿಟೇಷನ್ಗೆ ಬಂದಿದ್ದೀರಾ. ಐ ಥಿಂಕ್ ಯು ನೋ ದ ಥೀಮ್ ಆಫ್ ದಿಸ್ ನ್ಯಾಷನಲ್ ಸೈನ್ಸ್ ಡೇ. ಗೊತ್ತಲ್ವಾ ಎಲ್ಲರಿಗೂ ಈ ವರ್ಷದ ಥೀಮು? ವಾಟ್ ಈಸ್ ದ ಥೀಮ್? ಇಂಡಿಜಿನಿಯಸ್ ಟೆಕ್ನಾಲಜೀಸ್ ಫಾರ್ ವಿಕಸಿತ್ ಭಾರತ್. ಸೊ ಇಂಡಿಜಿನಿಯಸ್ ಟೆಕ್ನಾಲಜೀಸ್ ಅಂದರೆ ಗೊತ್ತಾ ಎಲ್ಲರಿಗೂ? ಯು ಆಲ್ ವುಡ್ ಹ್ಯಾವ್ ಪೇಂಟೆಡ್, ಇವತ್ತು ಪೇಂಟಿಂಗ್ ಮಾಡಿದ್ದೀರಾ, ಬಟ್ ಥೀಮ್ ಫಾರ್ ದ ಪೇಂಟಿಂಗ್ ಇಸ್ ಡಿಫ್ರೆಂಟ್ ಅಲ್ಲ? ವಾಟ್ ಈಸ್ ದ ಥೀಮ್? Sustainability. So innovations, they are important for solving global challenges and sustainable development. So innovation is very, very important. So ಈ ವರ್ಷ ಸೈನ್ಸ್, ಟೆಕ್ನಾಲಜಿ, ಇನೋವೇಷನ್ನ ಮೂರನ್ನು ಸೇರಿ ನಾವು ಮುಂದೆ ವಿ ಶುಡ್ ಡೂ ದ ಥಿಂಗ್ಸ್ ಇನ್ ದ ಫ್ಯೂಚರ್ ಅಂತ ಹೊಸ ಥೀಮ್ ಮಾಡ್ಕೊಂಡಿದ್ದಾರೆ. ಇಷ್ಟು ದಿನ ಸೈನ್ಸ್ ಆ್ಯಂಡ್ ಟೆಕ್ನಾಲಜಿ ಮಾತ್ರ ಇತ್ತು, ಈಗ ಇನೋವೇಷನ್ ಕೂಡ ಸೇರಿಕೊಂಡಿದೆ ಅದರಲ್ಲಿ. ಸೊ ಇನೋವೇಷನ್ ಈಸ್ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ವಿಕಸಿತ್ ಭಾರತ್, ಅದು ಇಂಡಿಜಿನಿಯಸ್ ಇನೋವೇಷನ್ಸ್. ಸೊ ಐ ವಿಶ್ ಯು ಆಲ್ ಗುಡ್ ಲಕ್. ಯು ಆರ್ ಆಲ್ ಅಂಬಾಸಿಡರ್ಸ್ ಆಫ್ ಸೈನ್ಸ್. ಸೈನ್ಸ್ನ ನೀವೇನು ಸೀರಿಯಸ್ ಆಗಿ ಯೋಚನೆ ಮಾಡಬೇಡಿ, ಯು ಟೇಕ್ ಇಟ್ ಆ್ಯಸ್ ಎ ಫನ್, ಲೈಟಾಗಿ ತೊಗೊಳ್ಳಿ. ಡೈಲಿ ನಿಮ್ಮ ಡೈಲಿ ಲೈಫಲ್ಲಿ ಸೈನ್ಸ್ನ ಅಳವಡಿಸ್ಕೊಳ್ಳಿ. ನಿಮ್ಮ ನೀಸು ನೆವ್ಯೂಸು ಯಾರ ಜೊತೆ ಮಾತಾಡುವಾಗಲೂ ಸೈನ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡಿ. ಚಿಕ್ಕ ಚಿಕ್ಕ ವಿಷಯನೂ ಯಾವ್ದಾರ ಭಾರದ್ದು ಒಂದು ಸೂಟ್ ಕೇಸ್ ತೊಗೊಂಡೋಗುವಾಗ ನೀವು ಸುಮ್ಮನೆ ತೊಗೊಂಡೋದ್ರೆ ಭಾರ ಆಗತ್ತಲ್ವಾ, ಅದನ್ನು ಎಳ್ಕೊಂಡೋದ್ರೆ ಭಾರ ಆಗಲ್ಲ. ಸೊ ಸ್ಮಾಲ್ ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್, ವೈ ಇಟ್ ಈಸ್ ಹ್ಯಾಪನಿಂಗ್. ಸೊ ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್ ವಿಲ್ ಟೇಕ್ ಯು ಟು ಬಿಗ್ ಹೈಟ್ಸ್. ಸ್ಮಾಲ್ ಲಿಟಲ್ ಡ್ರಾಪ್ಸ್ ಮೇಕ್ಸ್ ಎ ಮೈಟಿ ಓಷನ್ ಅಂತಾರಲ್ಲ, ಆ ಥರ ಚಿಕ್ಕ ಚಿಕ್ಕ ವಿಷಯಗಳು ನಿಮ್ಮ ಸೈನ್ಸ್ ವಿಷಯಗಳನ್ನ ಡೈಲಿ ಲೈಫಲ್ಲಿ ನೀವು ಅಪ್ಲೈ ಮಾಡಿಕೊಂಡು ಅದ್ರ ಬಗ್ಗೆ ಥಿಂಕ್ ಮಾಡಿಕೊಂಡು, ಇಫ್ ಯು ಕೀಪ್ ಆನ್ ಥಿಂಕಿಂಗ್ ಅಬೌಟ್ ಸೈನ್ಸ್, ಒನ್ ಡೇ ಯು ವಿಲ್ ಬಿಕಮ್ ಎ ಗ್ರೇಟ್ ಸೈಂಟಿಸ್ಟ್. ಸೈಂಟಿಸ್ಟ್ ಆಗಬೇಕು ಅಂತ ಸೀರಿಯಸ್ ಆಗಿ ಲ್ಯಾಬಲ್ಲಿ ಕೂತ್ಕೊಂಡು ಕೆಲಸ ಮಾಡೋದಲ್ಲ, ಈವನ್ ಅಟ್ ಹೋಮ್, ಈವನ್ ದೊಡ್ಡವರಾದಮೇಲೆ ಇಫ್ ಯು ಗೆಟ್ ಮ್ಯಾರಿಡ್, ಗರ್ಲ್ಸ್ ಎಸ್ಪೆಷಲಿ, ನೀವೇನು ಅಯ್ಯೋ ನಾನೇನು ಸೈನ್ಸ್ ಮಾಡೇ ಇಲ್ಲ, ಇನೋವೇಷನ್ ಮಾಡಿಲ್ಲ ಅಂತ ಅನ್ಕೋಬೇಡಿ. MTR ಪ್ರಾಡಕ್ಟ್ಸ್ ನಿಮಗೆ ಗೊತ್ತು, ಎವ್ರಿ ಟೈಮ್ ದೇ ಆರ್ ಇಂಪ್ರೂವಿಂಗ್ ದ MTR ಪ್ರಾಡಕ್ಟ್ಸ್. ಈಚ್ ಟೈಮ್ ದೆರ್ ಈಸ್ ಸಮ್ ಇನೋವೇಷನ್. ನಮ್ಮ ಸ್ಟೂಡೆಂಟ್ಸ್ ತುಂಬ ಜನ ಇದ್ದಾರೆ ಆ ರಿಸರ್ಚ್ ಗ್ರೂಪಲ್ಲಿ. ಸೊ ಹೇಗೆ ತುಂಬ ರುಚಿ ಮಾಡೋದು, ಟೇಸ್ಟ್ ಮಾಡೋದು. ಸೊ ಡೈಲಿ ಅಡುಗೆ ಮನೇಲಿ ಹೆಣ್ಮಕ್ಳು ಅಂದರೆ ಹೆಣ್ಮಕ್ಳು ಯಾವಾಗಲೂ ಅಡುಗೆ ಮನೇಲಿ ಇರಬೇಕು ಅಂತಲ್ಲ. ಸೊ ದೋಸ್ ಹು ಕಾಂಟ್ ಗೋ ಅಹೆಡ್ ಇನ್ ಸ್ಟಡೀಸ್, ದೋಸ್ ಹು ಕಾಂಟ್ ಡೂ, ಅವರೆಲ್ಲ ಬೇಜಾರು ಮಾಡ್ಕೊಳ್ಳೋ. ಸೈನ್ಸ್ ಎಲ್ಲ ಕಡೆ ಮಾಡ್ಬೋದು, ಲ್ಯಾಬಲ್ಲೂ ಮಾಡ್ಬೋದು, ಮನೆಯಲ್ಲೂ ಮಾಡ್ಬೋದು, ಕಾಲೇಜಲ್ಲೂ ಮಾಡ್ಬೋದು. ಥಿಂಕಿಂಗ್, ಬರೀ ಥಿಂಕಿಂಗೇ ಇನೋವೇಷನ್. ಹೊಸ ಹೊಸ ಥಾಟ್ ಪ್ರೋಸೆಸ್ನ ಮಾಡಿಕೊಂಡು ಯು ಕ್ಯಾನ್ ಅಪ್ಲೈ ಇನ್ ಯುವರ್ ಡೈಲಿ ಲೈಫ್. ಸೊ ಎಲ್ಲರೂ ಸೈನ್ಸ್ ಬಗ್ಗೆ ನೀವು ಜಾಸ್ತಿ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡಿ. ಯು ಶುಡ್ ಪ್ರೊಪೆಲ್ ಸೈನ್ಸ್. ಸೈನ್ಸ್ ಬಗ್ಗೆ ಜಾಸ್ತಿ ಮಾತಾಡಬೇಕು, ಬರೀ ಫಿಲ್ಮ್ ಬಗ್ಗೆ, ಯಾವುದೋ ಇನ್ಸ್ಟಾಗ್ರಾಮ್ ಬಗ್ಗೆ, ವಾಟ್ಸಪ್ ಯೂನಿವರ್ಸಿಟಿ, ಗೂಗಲ್ ಯೂನಿವರ್ಸಿಟಿ ಇದೆಲ್ಲ ಮಾಡೋ ಬದಲು ಸೈನ್ಸ್ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಡೈಲಿ ವಾಟ್ಸಪ್ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಕಳಿಸೋ ಬದಲು ಯಾರೋ ಒಬ್ಬ ಸೈಂಟಿಸ್ಟ್ ಪಿಕ್ಷ್ನರಿ, ನಾನು ಮೊನ್ನೆ ಯಾವುದೋ ಕಾಲೇಜಲ್ಲಿ ಪಿಕ್ಷ್ನರಿ ಮಾಡಿದ್ವಿ, ಗೆಲಿಲಿಯೋ ಫೋಟೋ ಹಾಕಿ ಹುಡುಗರನ್ನ ಯಾರಿದು ಅಂತ ಕೇಳಿದ್ರೆ ಐನ್ಸ್ಟೀನ್ ಅಂದರು. ಐನ್ಸ್ಟೀನ್ ಫೋಟೋ ಹಾಕಿ ಯಾರಿದು ಅಂತ ಹೇಳಿದ್ರೆ ಸ್ಕ್ರಾಡಿಂಜರ್ ಅಂದರು. ಅಂದರೆ ಸೈಂಟಿಸ್ಟ್ ಮುಖಾನೇ ನೋಡಿಲ್ಲ ನೀವು. ದಯವಿಟ್ಟು ಗೂಗಲಲ್ಲಿ ಡೌನ್ಲೋಡ್ ಮಾಡಿಕೊಂಡು ಸೈಂಟಿಸ್ಟ್ ಯಾರು, ಫ್ಯಾರಡೆ ಅಂದರೆ ಹೆಂಗಿದ್ದಾರೆ, ಐನ್ಸ್ಟೀನ್ ಅಂದರೆ ಹೇಗಿದ್ದಾರೆ, ಅಟ್ಲೀಸ್ಟ್ ನೋಡ್ಕೊಳ್ಳಿ ಯಾರು ಸೈಂಟಿಸ್ಟ್ ಇಷ್ಟು ಕಷ್ಟಪಟ್ಟಿದ್ದಾರೆ, ಇಷ್ಟೆಲ್ಲ ಮಾಡಿದ್ದಾರೆ. ಸರ್ ಸಿ ವಿ ರಾಮನ್ ಫೋಟೋನು ತುಂಬ ಜನ ನೋಡಿಲ್ಲ. ಸರ್ ಸಿ ವಿ ರಾಮನ್ ಫೋಟೋ ಹಾಕಿದ್ರೆ ವಿಶ್ವೇಶ್ವರಯ್ಯ ಅಂತ ಹೇಳ್ತಾಯಿದ್ರು, ಬಿಕಾಸ್ ಇಬ್ಬರು ಪೇಟ ಹಾಕ್ಕೊಳ್ತಾರೆ. ಸೊ ಪ್ಲೀಸ್ ಅದೆಲ್ಲ ನೋಡಿ. ಸೈಂಟಿಸ್ಟ್ ಫೋಟೋ 101 ಸೈಂಟಿಸ್ಟ್ ಅಂತ ಯಾವುದೋ ಒಂದು ಬುಕ್ ಇದೆ, ಅದರಲ್ಲಿ ಓದಿ ಅವ್ರು ಏನೇನು ಮಾಡಿದ್ದಾರೆ ಅವ್ರ ಲೈಫಲ್ಲಿ, ಏನು ಕಷ್ಟಪಟ್ಟಿದ್ದಾರೆ. ಹೂ ಈಸ್ ದ ಫಾದರ್ ಆಫ್ ಸೈನ್ಸ್, ಹೂ ಈಸ್ ದ ಮದರ್ ಆಫ್ ಸೈನ್ಸ್ ಗೊತ್ತಾ? ಮದರ್ ಆಫ್ ಸೈನ್ಸ್ ಯಾರು? ಮೇರಿ ಕ್ಯೂರಿ. ಫಾದರ್ ಆಫ್ ಸೈನ್ಸ್ ಯಾರು? ಗೆಲೀಲಿಯೋ. ನೋಡಿ, ಸೈನ್ಸ್ ಬಗ್ಗೆ ಒಂಚೂರು ನೋಡ್ಕೊಳ್ಳಿ. ಎಲ್ಲರೂ ಇನ್ಸ್ಟಾ ಯೂನಿವರ್ಸಿಟಿನಲ್ಲಿ ಫೇಮಸ್ ಆಗಿಬಿಟ್ಟಿದ್ದೀರಾ, ರೀಲ್ಸ್ ಮಾಡೋದು ಹಾಕೋದು ಎಲ್ಲ. ಈ ರೀಲ್ಸ್ ಮಾಡಿ, ಪಿಕ್ಚರಿ ರೀಲ್ಸ್ ಮಾಡಿ, ಡಿಫ್ರೆಂಟ್ ಫೋಟೋಸ್ ಹಾಕಿ, ಸೈಂಟಿಸ್ಟ್ಗಳದ್ದು ಫೋಟೋ ಹಾಕಿ ಆನ್ಸರ್ ಹೇಳು ಅಂತ ಹೇಳಿ, ಈ ಥರ ಡೈಲಿ ಒಂದೊಂದು ವಾಟ್ಸಪ್ ಮೆಸೇಜ್ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಕಳಿಸೋ ಬದಲು ಒಂದಿನ ಗೆಲಿಲಿಯೋ ಫೋಟೋ, ಒಂದಿನ ಇದು ಬೋರ್ ಫೋಟೋ, ಸ್ಕ್ರಾಡಿಂಜರ್ ಫೋಟೋ, ನ್ಯೂಟನ್ ಫೋಟೋ, ಈ ಥರ ಎಲ್ಲ ಹಾಕಿ. ಸೊ ದ್ಯಾಟ್ ಎಲ್ರಿಗೂ ಗೊತ್ತಾಗುತ್ತೆ ಯಾರು ಸೈಂಟಿಸ್ಟು, ಏನು ಕೆಲಸ ಮಾಡಿದ್ದಾರೆ, ಎಷ್ಟು ಕಷ್ಟಪಟ್ಟಿದ್ದಾರೆ ಎಲ್ಲ ಗೊತ್ತಾಗುತ್ತೆ. ಆ್ಯಂಡ್ ವಿಮೆನ್ ಸೈಂಟಿಸ್ಟ್ ಎಷ್ಟು ಜನ ಇದ್ದಾರೆ, ಏನೇನು ವಿಮೆನ್ ಸೈಂಟಿಸ್ಟ್ ಕೆಲಸ, ಐ ಸಿ ಲಾಟ್ ಆಫ್ ಗರ್ಲ್ಸ್ ಹಿಯರ್, ಎಷ್ಟು ವಿಮೆನ್ ಸೈಂಟಿಸ್ಟ್ ಏನೇನು ಮಾಡಿದ್ದಾರೆ ಅದೆಲ್ಲ ಸ್ವಲ್ಪ ಡೌನ್ಲೋಡ್ ಮಾಡ್ಕೊಳ್ಳಿ, ನೋಡಿ, ಓದಿ. ದಟ್ ಈಸ್ ಸೈನ್ಸ್. ಬರೀ ವರ್ಷಕ್ಕೊಂದ್ಸಲಿ ರಿಚುವಲ್ ಜಾತ್ರೆ ಮಾಡ್ದಂಗೆ ಮಾಡಿಬಿಟ್ಟು ಮನೆಗೆ ಹೋಗ್ಬಿಟ್ಟು ಬಾ ಆಯ್ತಪ್ಪ ಸೈನ್ಸ್ ಡೇ ಅಂತ ಸುಮ್ಮನೆ ಕೂತ್ಕೊಳ್ಳೋದಲ್ಲ. ಯು ಶುಡ್ ಕೀಪ್ ಆನ್ ಡೂಯಿಂಗ್. ದಿಸ್ ಇಸ್ ಎ ಕಂಟಿನ್ಯೂಯಸ್ ಪ್ರೋಸೆಸ್. ಡಿಡ್ ಯು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್? ಇಟ್ಸ್ ಎ ಕಂಟಿನ್ಯೂಯಸ್ ಪ್ರೋಸೆಸ್. ಇದನ್ನು ಒಂದಿನ ಗೋಕುಲಾಷ್ಟಮಿ ಮಾಡಿಬಿಟ್ಟು ಸುಮ್ಮನೆ ಇರೋಕ್ಕಾಗಲ್ಲ. ಡೈಲಿ ಕೃಷ್ಣನ್ ಜ್ಞಾಪಿಸ್ಕೊತಿರೋ ಇಲ್ವೋ? ವರ್ಷಕ್ಕೊಂದೇ ಸಲಿನ ಕೃಷ್ಣನ್ ಜ್ಞಾಪಿಸ್ಕೊಳ್ಳೋದು ಗೋಕುಲಾಷ್ಟಮಿ ದಿನ? ಇಲ್ಲ. ಡೈಲಿ ಯು ಥಿಂಕ್ ಆಫ್ ಗಾಡ್ ಲೈಕ್ ದ್ಯಾಟ್. ಥಿಂಕ್ ಸೈನ್ಸ್ ಆ್ಯಸ್ ಗಾಡ್. ಸೊ ಟ್ರೈ ಟು ರೀಸನ್ ಔಟ್ ಥಿಂಗ್ಸ್. ಅಂದರೆ ಜಗಳ ಆಡಬೇಡಿ, ಡೋಂಟ್ ಕ್ವಾರಲ್ ವಿತ್ ಯುವರ್ ಪೇರೆಂಟ್ಸ್, ಅದೇನು ಇದೇನು ಕ್ವೆಶ್ಚನ್ ಮಾಡಬೇಡಿ. Instead, you go to Google, go to encyclopedia, find out the answers, and enlighten and educate your parents and grandparents also. ಗ್ರ್ಯಾಂಡ್ ಏನಾದ್ರು ಹೇಳಿದಾಗ ಇದೆಲ್ಲ ಹಿಂಗೆ ಅಂತ ಕಾಮಾಗಿ ಅವ್ರ ಜೊತೆ ಮಾತಾಡಿ, ಜಗಳಾಡೋದಲ್ಲ. ಯು ಶುಡ್ ಟಾಕ್ ಟು ಯುವರ್ ಎಲ್ಡರ್ಸ್, ಸ್ಪೀಕ್ ಟು ದೆಮ್, ಟೆಲ್ ಅಬೌಟ್, ರೀಸನ್ ಔಟ್, ಇದೆಲ್ಲ ಹೀಗೆ ಹೀಗೆ ಅಂತ ಎಲ್ಲ ಹೇಳಬೇಕು ನೀವು. ಯು ಶುಡ್ ಟೆಲ್, ಟಾಕ್ ಅಬೌಟ್ ಲಿಟಲ್ ಸೈನ್ಸ್. ಅಂದರೆ ಡೋಂಟ್ ಥಿಂಕ್ ಆಫ್ ಬಿಗ್ ಇನೋವೇಷನ್, ಅಪ್ಪ ಡಿಸ್ಕವರಿ ಮಾಡಬೇಕು ಆ ಥರ ಅಲ್ಲ. ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್ ಮೇಕ್ಸ್ ಎ ಮೈಟಿ ಓಷನ್. ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್ ಇನ್ ಯುವರ್ ಲೈಫ್. Did you understand? So I don't want to give a big lecture, but I just want all of you to have the scientific temper in your life. ಎಲ್ಲನೂ ಸೈನ್ಸ್, ಒಂದು ಆಕಾಶ ನೀಲಿ ಯಾಕಾಗತ್ತೆ, ಗಿಡ ಒಂದೊಂದು ಗಿಡದಲ್ಲಿ ಒಂದೊಂದು ಕಲರ್ ಗ್ರೀನ್ ಯಾಕಿರುತ್ತೆ, ಕ್ಲೋರೋಫಿಲ್ ಏನು ಮಾಡುತ್ತೆ, ಫೋಟೋಸಿಂಥಸಿಸ್ ಏನು ಮಾಡುತ್ತೆ, ಯಾಕೆ ಈ ಗಿಡ ಇಂಡೋರ್ ಪ್ಲಾಂಟ್ಸ್ ಆಗಿ ಹಾಕ್ತಾರೆ, ಏನಕ್ಕೆ, ವಾಟ್ ಈಸ್ ದ ಗ್ಯಾಸ್ ಇಟ್ ಈಸ್ ಗಿವಿಂಗ್, ವಿಚ್ ಈಸ್ ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್ ವಿಲ್ ರಿಯಲಿ ಯು ಫೀಲ್ ವೆರಿ ಹ್ಯಾಪಿ, ಇಸ್ ಇಟ್ ನಾಟ್? ಸೊ ದಯವಿಟ್ಟು ಎಲ್ಲರೂ ಈ ಸೈನ್ಸ್ ಬಗ್ಗೆ ಥಿಂಕ್ ಮಾಡಿ ಆ್ಯಂಡ್ ಟಾಕ್ ಟು ಯುವರ್ ನೀಸಸ್, ನೆವ್ಯೂಸ್ ಆ್ಯಂಡ್ ಯುವರ್ ಪೇರೆಂಟ್ಸ್. ಡೈನಿಂಗ್ ಟೇಬಲಲ್ಲಿ ಸ್ವಲ್ಪ ಸೈನ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡೋದನ್ನು ಅಭ್ಯಾಸ ಮಾಡ್ಕೊಳ್ಳಿ. ಫ್ಯಾಮಿಲಿ ದಟ್ ಸ್ಟೇಸ್ ಟುಗೆದರ್ ವಿಚ್ ಟಾಕ್ಸ್ ಸೈನ್ಸ್ ಟುಗೆದರ್ ಅಂತ ಈಗ ಹೊಸ ಇದನ್ನು ಹೇಳ್ತಾ ಇದ್ದೀವಿ ವಿಚಾರ. ಮೊದಲು ಪ್ರೇಸ್ ಟುಗೆದರ್ ಅಂತ ಹೇಳ್ತಾ ಇದ್ವಿ, ಈಗ ಹೂ ಟಾಕ್ಸ್ ಟುಗೆದರ್ ಅಂತ ಹೇಳ್ತಾ ಇದ್ವಿ. ಯಾಕಂದರೆ ಮಾತಾಡ್ತಾ ಇಲ್ಲ, ಮಕ್ಕಳು ಮಾತಾಡ್ತಾ ಇಲ್ಲ, ಬರೀ ಮೊಬೈಲ್ ಮೊಬೈಲ್ ಮೊಬೈಲ್. ಪ್ಲೀಸ್ ಗೆಟ್ ರಿಡ್ ಆಫ್ ದಟ್ ಅಡಿಕ್ಷನ್ ಆಫ್ ಮೊಬೈಲ್, ಗೆಟ್ ಅಡಿಕ್ಟೆಡ್ ಟು ಸೈನ್ಸ್ and good science, not bad science. So with this small advice to you, I wish you all good luck. So I think you're all waiting to receive the prize. So we have come to the end. I thank Dr. Ramesh for giving me an opportunity to talk a few words in the valedictory function. Thank you.
ತಾವೆಲ್ಲರೂ ವೈಜ್ಞಾನಿಕ ಮನೋಭಾವವನ್ನು ಬೆಳೆಸ್ಕೊಳ್ಳಿ ಎನ್ನುವಂತಹ ಜೀವನ ಸತ್ಯವನ್ನು ತಿಳಿಸ್ಕೊಟ್ಟಂಥ ಡಾಕ್ಟರ್ ಗಿರಿಜಾ ಮೇಡಮ್ರವರಿಗೆ ಧನ್ಯವಾದಗಳು. ಈಗ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕ ಅಧಿಕಾರಿಗಳಾದಂಥ ಡಾಕ್ಟರ್ ಎಂ ರಮೇಶ್ ಸರ್ ಅವರಿಂದ ಅಧ್ಯಕ್ಷೀಯ ನುಡಿ.
Good evening. Respected Girija Madam, my friend Dr. Manja Nayak, Mr. Srinivas, Mr. Madhavegoda, Umesh, Bharat, Pandu, Chandru, Satish sir, Ashok, Siddhegoda, Ramesh, and all students from different colleges who have participated in the painting and drawing competition. In fact, when we now ಮೊದಲು ಅಡ್ವರ್ಟೈಸ್ ಮಾಡಿದಾಗ some 91 entries were there for painting and drawing competitions, but later only 35 or 40 came. Now around 20 students are there, among them 16 are girls, 17 are girls, 3 are boys. So it is not exaggeration that in most of the universities and colleges, best top will be girls. This is the reason. Boys will go out, girls will stay and do the work properly. Congratulations to all of you. There are only 6 prizes to be given. So 20 of you are there. Among you, 6 will get the prizes. But those who did not get need not have to worry. Many programs like this will happen in KSTA. My teaching colleague, doctor is from Jyoti Nivas is there. He will bring you again to compete. So don't be disheartened. As they say, success is not final. Failure is not fatal. So you should always keep trying new things. Only then you will get success. Madam Eldro, as science students, science should become the way you live, the way you talk. Only then science can spread to many people across the state. ಈಗ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಸೈನ್ಸ್ ಓದ್ದವ್ರು ಮಾತ್ರ ಸೈನ್ಸ್ ಮಾತಾಡ್ತೀವಿ, ಬೇರೆ ಆರ್ಟ್ಸು ಕಾಮರ್ಸ್ ಓದ್ದವರು ಸೈನ್ಸ್ ಬೇಡ, ನಮಗೆ ಆರ್ಟ್ಸು ಸಾಕು, ಕಾಮರ್ಸು ಸಾಕು ಅಂತಾರೆ. ಅದಕ್ಕೆ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್, ಸೈನ್ಸ್ ಲಿಟ್ರಸಿ ಈಸ್ ಲೆಸ್. ಈವನ್ ಇನ್ ಅಮೇರಿಕಾ ದೇ ಸೇ science literacy is less than 20%. Imagine India. So what should we do? You all should be ambassadors of science. As Madam told, even in dining table you should talk about science instead of cricket or film. Only then science can be, so you bring scientific awareness among people. ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಹೇಳ್ತೀನಿ. ನೀವು ಸೈನ್ಸ್ ಓದ್ತಾ ಇದ್ದರೆ ಯು ಸ್ಟಡಿ ಓನ್ಲಿ ಫಿಸಿಕ್ಸ್ ಆರ್ ಕೆಮಿಸ್ಟ್ರಿ ಆ್ಯಂಡ್ ಯು ಡೋಂಟ್ ಸ್ಟಡಿ ಬಯಾಲಜಿ. ಬಟ್ ಬೆಳಿಗ್ಗೆ ನಮ್ಮ ನಾಗರಾಜ್ ಸರ್ ಹೇಳಿದ್ರು, ಬಯಾಲಜಿ ಆಲ್ಸೋ ರಿಕ್ವೈರ್ಸ್ ಕೆಮಿಸ್ಟ್ರಿ, ಮ್ಯಾಥಮೆಟಿಕ್ಸ್, ಫಿಸಿಕ್ಸ್. ಈ ಗುಡ್ ಎಕ್ಸಾಂಪಲ್ ಈಸ್ ದಟ್ ಆಫ್ ವೆಂಕಿ ರಾಮಕೃಷ್ಣನ್, Nobel laureate in 2009 for ribosome discovery work, or how structure and function of ribosome. In fact, he was a physicist. He did his PhD in physics. After doing physics, he thought physics is boring. He should do something in biology. So he tried to do one course in molecular biology. No university accepted. Only at the end, California University accepted him as a student to do a graduation degree for 2 years. After doing graduation in molecular biology, he pursued research in molecular biology. Finally, he got Nobel Prize in ribosome. Why I would like to highlight this is that you should not stick on to only one subject. You should have a T-shaped knowledge curve. In the sense, you should be an expert in one subject, whereas you should also have the knowledge of other subjects. Only then you will succeed. Second thing is, you should work in a group, not individually. If you are a physicist, you can't work alone as a physicist. You should also collaborate with chemists, because we are not in silos. As Madam was mentioning in the morning, science has no borders. The science done in India should reach to Africa also. So there is no restriction. So you should be able to collaborate well with your own colleagues and teachers. Only then success will be yours. So with this, I wish you all a great success in future. And those who did not get prize need not have to worry. After April, we will conduct one more program like this related to science. You can always compete and get the prize. I wish you all a great future. Thank you.
ಧನ್ಯವಾದಗಳು ಸರ್. ಈಗ ಒಂದು ಚಿತ್ರಕಲಾ ಸ್ಪರ್ಧೆಯನ್ನು ಏರ್ಪಡಿಸಲಾಗಿತ್ತು. ಅದರಲ್ಲಿ ವಿಜೇತರಾದಂಥವರಿಗೆ ಬಹುಮಾನ ವಿತರಣಾ ಕಾರ್ಯಕ್ರಮ ಇದೆ. ಅದರಲ್ಲಿ ಮೊದಲನೇ ಬಹುಮಾನ 5,000 ರೂಪಾಯಿ, ಎರಡನೇ ಬಹುಮಾನ 3,000 ರೂಪಾಯಿ, ಹಾಗೆ ಮೂರನೇ ಬಹುಮಾನ 2,000 ರೂಪಾಯಿ. ಇದರ ವಿವರವನ್ನು ಓದಲಿದ್ದಾರೆ ನಮ್ಮ ಶ್ರೀನಿವಾಸರವರು.
ಮೊದಲಿಗೆ ಯು ಜಿ ಪ್ರೋಗ್ರಾಮ್ ಅಲ್ಲಿ ಗೆದ್ದಂಥ ವಿದ್ಯಾರ್ಥಿಗಳು. ಥರ್ಡ್ ಪ್ರೈಸ್ ಗೋಸ್ ಟು ಪವನ್ ಕುಮಾರ್, ಜ್ಞಾನ ಡ್ರಾಮಾ PU ಕಾಲೇಜ್. ಪವನ್ ಕುಮಾರ್. Second prize goes to Sahalala Sherin from College of Horticulture, Bangalore. ಹೆಸರು ಬರಿತೀವಿ, ದುಡ್ಡು ಕೊಡ್ತೀವಿ. So don't worry. The first prize goes to again the College of Horticulture from Hemant Kumar Vyar. Under post-graduation category, the third prize goes to Rajalakshmi B from Maharani Cluster University. And the second prize goes to Manya M from Maharani Cluster University. Same. Manya M. Manuja. Manya M ಅಲ್ಲೇ ಹಾಕಿದ್ದಾರೆ. ಮನುಜ ಎಂ ನಾಡ್ದೇರಾ? And first prize goes to Sadhanjali Sahu from East West First Grade College. PG category. PG. And thank you and congratulations to all the winners. Thank you, dignitaries, for presenting the prizes.
ಬಹುಮಾನಗಳನ್ನು ತಮಗೆಲ್ಲರಿಗೂ ವಿತರಿಸಿದಂಥ ಮತ್ತು ಸ್ವೀಕರಿಸಿದಂಥ ತಮಗೆಲ್ಲರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸ್ತಾ ಈಗ ವಂದನೆಗಳನ್ನು ಸಲ್ಲಿಸ್ತಕ್ಕಂಥ ಸಮಯ. ಈ ಒಂದು ಸಮಾರಂಭದಲ್ಲಿ ಬೆಳಿಗ್ಗೆಯಿಂದ ಇಲ್ಲಿಯವರೆಗೂ ಕೂಡ ತಮ್ಮೆಲ್ಲ ಕೆಲಸ ಕಾರ್ಯಗಳನ್ನು ಬದಿಗೊತ್ತಿ ನಮ್ಮ ಜೊತೆಯಲ್ಲಿ ಉಪಸ್ಥಿತರಿರ್ತಕ್ಕಂಥ, ಹಾಗೆಯೇ ಈ ಒಂದು ಸಮಾರಂಭದ ಮುಖ್ಯ ಅತಿಥಿಗಳಾಗಿ ಉಪಸ್ಥಿತರಿರ್ತಕ್ಕಂಥ ಡಾಕ್ಟರ್ ಮಂಜು ನಾಯಕ್ ಸರ್ ಅವರಿಗೆ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ಹೃತ್ಪೂರ್ವಕವಾದಂಥ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸ್ತೇನೆ. ಹಾಗೆಯೇ ನಮ್ಮ ಗಿರಿಜಾ ಮೇಡಮ್ ಅವರು ಕೂಡ ಬೆಳಿಗ್ಗೆಯಿಂದ ಇಲ್ಲಿಯವರೆಗೂ ಕೂಡ ನಮ್ಮ ಜೊತೆಯಲ್ಲೇ ಇದ್ದಾರೆ. ಈ ಸಮಾರಂಭದಲ್ಲಿ ಮುಖ್ಯ ಅತಿಥಿಗಳಾಗಿಯೂ ಕೂಡ ಉಪಸ್ಥಿತರಿದ್ದಾರೆ. ಅವರಿಗೂ ಕೂಡ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ, ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ಹೃದಯಪೂರ್ವಕ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸ್ತೇನೆ. ಹಾಗೆ ಈ ಸಮಾರಂಭದ ಅಧ್ಯಕ್ಷತೆಯನ್ನು ವಹಿಸಿರ್ತಕ್ಕಂಥ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಅವರಿಗೂ ಕೂಡ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹೃದಯಪೂರ್ವಕವಾದಂಥ ಧನ್ಯವಾದಗಳು. ಹಾಗೆಯೇ ಈ ಚಿತ್ರಕಲಾ ಸ್ಪರ್ಧೆಯಲ್ಲಿ ಭಾಗವಹಿಸಿದಂಥ ತಮ್ಮನ್ನು ಆಯ್ಕೆ ಮಾಡಿದಂತಹ ಜೂರಿ ಮೆಂಬರ್ಸ್ಗಳಾದಂಥ ಅಶೋಕ್ ಮತ್ತು ಸತೀಶ್ ಸರ್ ಅವರಿಗೂ ಕೂಡ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ, ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸ್ತೇನೆ. ಹಾಗೆಯೇ ತಮಗೆಲ್ಲರಿಗೂ ಸ್ವಾಗತವನ್ನು ಬಯಸಿದಂಥ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ವೈಜ್ಞಾನಿಕ ಅಧಿಕಾರಿಗಳಾದಂಥ ಶ್ರೀನಿವಾಸರವರಿಗೂ ಕೂಡ ವಂದನೆಗಳನ್ನು ಅರ್ಪಿಸ್ತೇನೆ. ಹಾಗೆಯೇ ಈ ಒಂದು ಸಮಾರಂಭಕ್ಕೆ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಎಲ್ಲ ಅಧಿಕಾರಿ ಸಿಬ್ಬಂದಿ ವರ್ಗದವರು ಪ್ರತ್ಯಕ್ಷವಾಗಿ ಪರೋಕ್ಷವಾಗಿ ತಮ್ಮ ಕೆಲಸ ಕಾರ್ಯಗಳನ್ನು ತುಂಬ ಅಚ್ಚುಕಟ್ಟಾಗಿ ನಿರ್ವಹಿಸಿದ್ದಾರೆ. ಅವರೆಲ್ಲರಿಗೂ ಕೂಡ ನಿಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹೃದಯಪೂರ್ವಕ ಧನ್ಯವಾದಗಳು. ಇಲ್ಲಿಗೆ ಇವತ್ತಿನ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮ ಮುಕ್ತಾಯ ಆಗ್ತಾ ಇದೆ. ಈಗ ರಾಷ್ಟ್ರಗೀತೆ, ತಾವೆಲ್ಲರೂ ಎದ್ದು ನಿಂತು ಗೌರವವನ್ನು ಸೂಚಿಸಬೇಕು. ನಮಗೆಲ್ರಿಗೂ ನಗದು ಬಹುಮಾನವನ್ನು ಇಲ್ಲಿ ಕೊಟ್ಟಿಲ್ಲ. ಅಲ್ಲಿ ನಮ್ಮ ಶಾಂತರಾಜು ಅಂತ ಅಕೌಂಟ್ಸ್ ಆಫೀಸರ್ ಇದ್ದಾರೆ, ಅವರು ಕೊಡ್ತಾರೆ. ನಿಮ್ಮ ಸರ್ಟಿಫಿಕೇಟ್ಸ್ ಏನಾದ್ರು ತಪ್ಪಿದರೆ ಅಲ್ಲಿ ಮತ್ತೆ ಕರೆಕ್ಷನ್ ಮಾಡಿಸ್ಕೊಳ್ಳುವಂಥದ್ದು. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು.